interact to increase the risk of prostate cancer. So we have the genetics, but what is the role that the environment play? And can we see signals to show that there is a translation of the social experience into the biological nature? And so our goal, our aims in the pilot, I'm sorry, I keep saying pilot project, full project. We started phase 1 as pilot project, is to link genetic factors, which is what we call the germline DNA repair variation that we found from the work that we did in CAPTC, to the plan we can use to source, and also health-related quality of life. And so we sort of spread it out in 5 legs. We have the study, the understudy population, the related biology, the dynamics of the biochemical state, the functional, analysis of the tumor, and then we're looking at how we use that to impact survivorship. So next slide, I'll be presenting our approach. I thought about just mixing the method that we're using with the findings, and then we can wrap it up later. So one of the challenges that we have in doing this type of work in Africa is that a lot of things you have to build afresh. It's like if you want something that is not in the market, you have to find a way to create those things. So we started by recruiting so far, and the data that we have, I'll be presenting to you with data from Benin Republic, Ivory Coast, she's here, Ghana, Kenya, and Nigeria. But right now we have additional samples coming in from Sierra Leone and Burkina Faso, and other parts of Africa that are still reaching out to us. And so of course we extract the DNA, we did the sequencing, and we thought that is all. We realized that no, you have to build the bioinformatics line, and we're very adamant on doing everything indigenously. So rather than exporting part of it to the US and asking our colleagues who are very always generous to support, we decided to build this locally, and we see the importance of that because that allows us to train indigenous scientists. So we build it locally, and in building it locally gives us the opportunity to really have a high on what we're building and seeing that, wait a minute, what we are seeing in our population is different from even what have been reported in African America. And so we kept on tweaking. An example of that was that when we first of all analyzed the data, and I'll show you, we found a gene that was sort of mutated. I'm trying to use not to get that. It's kind of changed in all the samples that we have, almost 100% of the data. And this is a gene that predisposed to Lynch syndrome, which is a risk marker for colorectal cancer, breast cancer, of course now prostate cancer. And we're concerned. So we realized that actually in our population we have what we call the pseudo gene, which is a copy of that gene but it's inverted. Okay, and if we had used the common pipeline, we're just going to call it that way. So we had to look for a way to rewrite that one. And so that's the further process. In fact, that's actually what slowed down that publication. And so this is the result that we're seeing. In the 180-something participants that were sequenced so far, you look at the upper part, you will see genes like ATM, PMS2, that is almost in everybody. And I will explain that APC, ALK, BRCA1, BRCA2. Now, if you look at the BRCA1, BRCA2, ATM, APC, these are genes that we had called before in our CAPTC publication, and we call so many other mutations that we have. Again, these mutations that we're calling are not in the tumor, but in the germline, which means things that are transmitted from parents to offspring. And so the first slide you see, they're just general variation. It doesn't mean that it's going to cause anything. It doesn't mean, it could just, most of them don't even mean anything, okay, variation. But these are pathogenic variations that are well documented, okay, that have been well documented, and you could see that out of that 180-something, we're able to identify 34 individuals that actually have mutations that are well documented to be pathogenic. And don't be alarmed by that. I think what is more concerning is what we don't know. We have high rate of what we call variants of unknown significance. We know this is a change. It has never been reported as normal. There's also no evidence that is abnormal. And this is the problem that we have when we're doing genomic studies in African population. The reason why we have this problem is that when we read out this sequence, we have to compare it to a reference. That reference is grossly deficient in African population. So he's saying I'm seeing something that has never been reported before, but we don't know whether it's pathogenic or it's not. And so imagine patients in Africa that have been doing genetic studies, tests, and they're going to call a lot of variants of unknown significance. So that creates a lot of distress. And that's a major gap that we need to fill. That's almost all the participants that we study. We identify important genes, changes in important genes, but we just don't know whether they are pathogenic or not. So we call it variants of unknown significance. The good thing is that for breast cancer, we working with Mayo Spore, and they have a project that actually now doing functional study on the ones in breast cancer. Hopefully, they will be able to help us out with this, with prostate cancer. But it's very similar to what we have in breast cancer. So we'll have high rate of variants of unknown significance. Again, just showing you our previous data from CAPTC that we've seen this BRCA2 before APC, ATM, BRCA1. But what we didn't call very well that time was the PMS2. It's somewhere there, that panel 1, that panel A is the one is for Nigerian data. Okay. Okay, I'll skip over this. So one thing about genetic studies that is good, but it really doesn't tell us the full picture, because if you look at the flow of what we call the biological information, at the base of the pyramid is the DNA that we studied in genomics. But at the top of it, that really are the messenger that provide us the phenotype, the variation that we are seeing are the metabolites. And so in this phase, we are also not just studying the DNA, which is the upper phase, we are also studying the upper realm of the pyramid. I say, what is going on there that is really interacting with the environment? Those are the messengers that do the work. So when we run this data, each sample gives us at least 6,000 metabolites. And we look into the process of analyzing it. Again, we really didn't find anything that can address the kind of parameters that we have. And I will tell you the reason. What we set out to look out for were what we call endogenous metabolites, things that are part of us. In the samples that we got a lot of things that are not endogenous. We forgot, we didn't realize that in Africa we take a lot of other things. And as you've seen the data, these other things just clouded everything. So we had now to develop our own algorithm to analyze this data. We call it multimodal classification system for metabolomics data. We're yet to finalize the name, but we're developing it, and I'll show you. So this is how it works. So this is, we call it, my guys just call it X2. And what we've done is that we've developed an AI, driven end-to-end algorithm for metabolomics intelligence pipeline. And we take it from the point 1 to the end, that we can even, using AI APIs that will be integrated to the system, begin to tell us some important features. So these results come out in what we call chemical structure smiles. And for those of us that did basic chemistry in undergraduate, typically in analytical chemistry, they give you this one compound and it take about 1 hour to find the structure and all of that on paper. You put it on ordinary computer, maybe it will take about 10 minutes or so to run through. Now we're talking of 6,000. So that was more than what just ordinary computer can power. So, thank God we have high-performing computer in Covenant University. So we embedded on that our AI system that can now quickly, like this morning, I just said to them, no, I need more clarification on this. And they ran through it within 30 minutes, able to run through the whole pipeline that has been developed locally. It's not that the system to analyze this didn't exist, but here is a slide showing examples of approaches that have been reported in literature to analyze this type of data. But realize that everywhere you see X means that those pipelines that are existing are deficient in those areas. But we need something that will be more robust. So again, doing this indigenously, locally at home, gives us the opportunity to develop what we need that can address our own peculiarity. Okay, so this is what you have, just a global look at it. Most of those things that you are seeing in red on top, most of them, and I will show you further. But then the PLS and PSD were able to use AI, machine learning approach to classify, and then you can see clearly there's a decision between cases and control. Okay. This is exogenous, the thing I told you about. And I think there's a typo here. This is endogenous, actually. I'll come back to this. This is exogenous, the one I told you about that clouded everything. So we separated endogenous from exogenous. This exogenous means that compounds that are not naturally part of human body, from environment, drug, medicinal plants, food, and all of that. And what you will see, so I decided to give a few examples on that table. So these are things that we found in the samples that are like medicinal plants. Some of them have anti-cancer property. One of them actually is showing indication of arsenic toxicity. Okay, I think it's the last one. It's actually an organic arsenic form. And these are things that we found that we were not planning to see before. But at the same time, I think that's my concluding part. This is the endogenous part that is helping us to really understand how do environmental exposure and social experience translate to biological information. And I'm giving this to example one, porphyrin 9, and then ipucine. What they do, which we have seen in these plasma samples that we analyze, is that we're beginning to see markers that we can use to measure how social and live experience impact the genomic of the populations that we're studying. And put all of this together in that while we have the social experience, social determinant of health, and while the biological determinants of health on the other hand, with our metabolomics analysis, we're able to see signature of what we can call the bridges or bridge molecules that are able to take this information from the environment and use it to control the genetic information and changes that we're seeing, whether it's epigenetics or gene expression. And moving forward now, we're now designing projects to study this further in the laboratory. So to conclude, we've seen evidence that prostate cancer in our cohort is linked to variation in what we call generally homologous repair pathway and mismatch repair pathway, the sub-pathway of that. The good thing about that is that we have drugs now, the PARP inhibitors, that individuals that have this variation can benefit from should they develop prostate cancer. And in Nigeria, we're talking to AstraZeneca that markets olaparib, and discussing with them to see how they bring this for the population. I think it's very concerning that we have high rate of variants of no significance, 99% of them largely driven by PMS2. But then we've seen indication that protoporphyrin 9 and ipucine appear to be plausible biochemical bridges, molecules that can translate social exposure to biological drivers of prostate cancer risk and aggression. And for our population in Africa, xenobiotics, things that people eat, and swallow, people believe so much in, medicinal plants, seems to be very important drivers of the phenotype that we're seeing. That's an area that has not been explored before. I think this data that we have is giving us indication that that's something that we should look in the direction. I'm just doing the talking. These are people, and I just grabbed some pictures of them. Up at the campus is the ESI, Bimbo is the postdoc, trained under me as for a PhD. Without iCCaRE, she too should have run away. There are many of them like that. But then with funding from iCCaRE, we're able to engage as postdoc. Tola trained for a master's. She's now continuing as a research assistant. Sylvester and Freeman, they are the guys that are developing the AI to Dr. Che met them, and very good guy. Bumi and Elizabeth are two people that just came in for their masters. Bernard is doing residency in Ghana, come to our lab for training. Gbolo Cooper is from Liberia, come to our lab for training. Michael is under our training in Ivory Coast. And Sidona Fatima is a new PhD that's part of the project that we just expanded to Burkina Faso. Thank you very much. Just to acknowledge the DoD for this funding. Thank you.
Thank you so much, Professor Rotimi, for shedding light on all that pathogenic variations and environmental exposures to prostate cancer, especially in Africa. And we'll take questions for Professor Rotimi now. Any questions for him? Girls?
Yeah, there's a question over there.
Oh, okay.
Dr. Rotimi, thank you very much for the presentation. My question to you is, can you elaborate, because I know in Africa, Nigeria, and in Africa, people take so much herbal medication. They prefer to even taking the normal pills. So, and then the food too. Can you just elaborate one or two that you found out that affected your study in what you were doing? Thank you.
Yeah, thank you very much, sir. I agree with you. So, so the joke we had in our lab is where you said in Africa that this patient that you are recruiting is treatment naive. What does that mean? Because this person has taken a lot of hybrid drugs. Some of them were fortified with pharmaceuticals. We saw all of that. In fact, if you look at these ones that we're showing, they are not even nutrients. They are not like common, flavonoids or all of that. I would say, oh, this is just, things that are antioxidants, from vegetables and all of that. We've extracted those ones that we consider to be xenobiotics, like strange compounds, external compounds that could have, that could alter biochemical processes. There are many of them, they run into, I don't have the exact number, but I would not be surprised if they run into 1,000 out of the 6,000 metabolites that we identified. But here I just listed 1, 2, 3, 4, 5. One of them, number one, for example, is from onion. Number two, and we take a lot of onions, Number two is, I think one of them is from cola. I think I think that dropped off. That should be number 6 or so. It's a long list. So what we're doing now is that I've asked Dr. Docampos is now working and refining it. And actually that's the abstract that she submitted to iCCaRE. We want to really make something out of that. Say, what is it that we, and it's called exposomes. Okay, it's part of environmental exposure now that we have to be able to describe. What are the herb-derived potentially toxic compounds? There's a general theme that once it's from herb, it's safe. That's what they used to market it. Say, oh, this herbal product is from plant, is safe. Is that we need to show this and say, look, the fact that it's from herb does not mean that it's safe. In fact, one of the challenges that NAFDAC is having now is that NAFDAC tried to ban, there's something, these things, herbal, something that they sell in, but infused with alcohol that they sell in sachets. There's a lot of political pressure against NAFDAC from banning it. I think some people even protested and all of that, and that it should be allowed and all. So, yeah, I agree with you. People take a lot of herbal drugs. And I think it's the same thing in Burkina, in Cote d'Ivoire. Yes. And I think the other one that is also important is this arsenic, because there's a lot of waste, a lot of dumping of electronic products in the, coastal area in Africa. I think there was even a report of, I don't know which country from Europe that dumped waste, which, can you remind me? In Ivory Coast, it's all over. And it seems it's getting into the sea food, and that's the product that we picked out here as that number 5.
I'll be with you.
Good morning. My question to you, why Black men affected with the prostate cancer all the time? That's my question. Can you explain to me why?
Oh, okay. Well, so that is what brought me to this research. So I won't go that storyline. But what we're seeing is an interplay between the genetics and the environment. We still don't have the full information about how, but we think for every science that we do, we're adding more information to it. So there's the genetic component of it, as I showed from the twin study. But then the environment is very important. All of these things that we're seeing, that people are eating, the change in lifestyle, the stress, all of it come together. Thank you.
Thank you, Professor Rotimi. 2 quick questions for you. Given that so many of us historically, as Black men and even non-Black men, share genetic stock of African men, because if you think about it, we have been brought all across the world and interbred, so to speak, and therefore we all share this genetic stock. And so if we share this genetic stock, how important is it that concurrently, while you are doing this work on men who reside in Africa, that you do this kind of work as well on men who are outside of the continent of Africa? Because even though we share genetic stock, the genetic stock has been watered down or changed by interbreeding, as well as the social determinants that we face are a little different than what exists inside of Africa. So that's the first question. How important is it to concurrently study? And then the second thing is, as we look on the social determinants, if you were to do some modeling to estimate attributable factors, so how important is the pulling of the trigger versus the loaded gun? In epigenetics, we talk about the gene being the loaded gun and the determinants like the social and environmental stuff being the finger that pulls the trigger. How important is either? And can we do something? Is it more malleable to do something on the social side as opposed to the genetic side?
Thank you very much. I'll start from the second one. You see this slide? The challenge so it's more malleable to address the social side, but because we didn't we're yet to fully understand how the social translates to the epigenetics. That's why we don't have the window of opportunity for intervention. But the data that we are now getting from our study is not showing us the window of that intervention, that there are signal molecules that you can study that will show up. In fact, part of what we are picking up in this signal right now is that, and I was discussing that with Mr. Badimos, he stepped out yesterday, that from our data, there are those controls that you could see out of the 6,000 points, you could see some, like 100, that are already showing indication tending towards the cases, but they're still controls, right? And so the follow-up project that we're doing now is to zero in on these two, and some of them that I've identified, and really in the laboratory study them and be able to model and show that, look, if we focus on this, we can begin to see that, look, this guy that is exposed to this is already showing signal that this is going to affect his epigenome, because these molecules actually influence the changes in the epigenome. We know epigenome changes, but we really didn't know what changes the epigenome. So that's number one. So that's the second question. So for your first question, yes, we were together yesterday, so we discussed about doing this other part of this study in the United States, which is very important. Yes. But you see, the great thing is that the evidence is coming from Africa, okay, rather than it coming from outside Africa and now trying to translate it to Africa, which is very, very difficult. Would you, Dr. Adichie, okay.
Yeah. So a follow-up question to that.
Oh.
Who is asking the question?
Dr. Osi.
Okay. I can always ask.
Professor Rotimi,
Yes, sir.
please forgive me if you've answered this question already. I would apologize. Is there any discrete information on the genetics and the epigenetics of those people who live urban as opposed to a group that truly would be what you would consider rural or agrarian, or they're just not exposed to McDonald's and cigarettes and all the other things we're exposed to? It would be interesting if there was such a study to look at the incidence and the severity and survivorship of prostate cancer, or does it even exist in the Khoisan Bushmen or people who really live like our ancestors of old?
Yeah, thank you very much. We're working on that. There's a whole project coming up now there's an ongoing project that we're looking into that. We didn't factor that into this, but there's a follow-up project that we're working on with our sister consortium to really look at, what we call zoning, Prof. Hospital, let it. Okay. So we're stratifying our population into what we call the blue and red zones right now. That where you live, whether you're living rural or in the urban, and even within the urban, there are urbans that are really, really polluted. There are urban aspects that are affluent and well-structured and all of that. So we're classifying across Africa now, different countries, and I think it's also going on in the United States, where we're mapping what we call the blue zone and the red zone, and we're collecting the social determinant of health, biological samples that will allow us to answer that question. Thank you, sir.
Any more questions? Okay, please another round of applause for Professor Rotimi. Okay,
Thank you, the great presentation, and good morning, everyone. I'm Ting Yuzhou. I'm a research fellow at the Mayo and in Dr. Dina's lab. And welcome to the plenary scientific session 2, social and structural factors driving prostate cancer disparities. And our speaker is Dr. Ernest Kaninji. He's an associate professor of public health at Georgia College and State University, and a multiple principal investigator in the iCCaRE Consortium. His research examines how immigration shapes prostate cancer risks and outcomes, especially among sub-Saharan African immigrants in the United States, and explore important differences within men of African ancestry. And thank you for joining us. The floor is yours.
Hello, good morning. I am delighted to be here with you all to talk about social determinants of health that is driving prostate cancer disparities. And I will touch a little bit, add some more layers into what Dr. Rotimi just presented a short while ago. But my presentation is actually in two halves. The first half will be broad. So I'm going to bring everybody together, picking up on some of the conversations we had yesterday, the advocates. We're going to talk more broadly. What are these determinants? What do we mean? How does geography, touching a little bit on the question that Dr. Willis was just asking, using more data from the US and geography, what can geography tell us about these determinants? And then the second half of my talk will go more deeply into my study, what we're finding among sub-Saharan African immigrants. So that's the way I plan to approach my talk, is in 2 halves. So this is the classic definition of social determinants of health. So the conditions into which people were born. Did you know that your zip code has a high influence on how long you live? So the conditions in which people are born, the conditions in which people grow, where they live, where they play, where they go to school, the education that they have, the social support or lack thereof. Also, housing. Housing is a big one. And then, most importantly, access to care, especially in the US system. So this is a slide by the Commission on Social Determinants of Health, just showing you the importance of social structures, policies, the governance on your left there. Do we have a universal access to care or not? How do people access care? How about work conditions? Do you work in a healthy environment? Do you even have health insurance through your job? So all of that interact with social hierarchy. What you see there in the box there are conditions that interact with each other, creating social structures, creating social determinants that becomes an intermediary. And the reason we focus on social determinants is because it impacts well-being. It impacts quality of life. So we're not just interested in social determinants of health for the sake of it. It's because it does impact quality of life and well-being. So we talked about disparities yesterday. We talked a little bit about incidence, mortality, morbidity. Here's how it looks like. When we talk about disparities, here's how it looks like. The blue is showing you data on incidence, new cases of prostate cancer in the United States as of 2025. This is data from the American Cancer Society. You could see Black race in blue leading in terms of new cases for prostate cancer. About 198 per 100,000 people. You see whites are second. You see American Indians and Alaska Natives are third. You see Hispanics. So it's all broken down by race and ethnicity. The red is where the big story or the bigger story is. That's mortality. Who dies from prostate cancer? What are the race? You could see Black there. It's overwhelmingly 36.9. Look at American Indian and Alaska Native. They represent a small proportion of the population, and yet they bear a huge burden, about 20%. And then look at white, about 18%. So when you look at the difference between Black and white, it's 2 to 1. The difference in mortality between Black and white, as you can see here, is 2 to 1. 18.4 versus 38.6. So when we talk about disparities, this is what we're talking about, the differences in health outcome. So what is driving it? So we're going to focus now more broadly. What are some drivers that we know has been shown in the literature? So I'm going to show you some evidence in the literature about factors that are driving this. So the first one is access to care. How do we get care in the United States? We don't have universal insurance. So you get access to care through insurance, mostly through your job or privately. So this slide is showing you the percentage of a population that does not have insurance. Focus on the green at the top. So the green at the top is showing you uninsured percentage by race or ethnicity. You can see whites there, about 6.5% are uninsured. Hispanic, very high, 17.9. Blacks, 9.7. You can see Asians, the lowest at 5.8. Look at American Indian and Alaska Natives, very high, 18%. So you could see some populations have disproportionate numbers of lack of insurance. And this has a bearing in terms of health outcomes. All right? So let's talk a little bit about screening. So screening rates are declining across all races and ethnicity. All right? Here's the problem. While Black men are screening, they're not screening at the rate that is commensurate to the problem that we just saw in the red. They're screening, but they're not screening to the degree that will close that gap. So we'll talk about disparities in terms of screening. This is a great study that just came out recently. It was a study that had a large population, over 606,000 people. A very huge Black men were represented in this study. It's a study across 3 states, over 900 clinics, 900 sites. And we could see here that Black men, the good news is Black men were more likely to be screened and to get a referral to a urologist. But again, you will see here the levels are not where they should be. On the top slide, you could see screening by different age groups. So 40 to 49, 50 to 59, 60 to 65. You could see the pattern, the trajectory has been going down. Again, this is just not one state. We're talking about North Carolina, South Carolina, Georgia, 900 different locations, huge population of Blacks. You could see, regardless of the age bracket, it's been going downwards. And then below, you could take a look at the Black versus white. White is in blue, the gold is Black. So basically, they're almost tied in terms of screening rates. Well, we just saw a couple of slides before, the mortality rate for Black was twice as much as white. So we're saying it's declining. It's a bigger problem among the Black population because we know we're more genetically disposed to this. All right? So it turns out geography can tell us a lot about prostate cancer risk. Geography can. I'm going to show you quite a few slides. So geography, again, is a key determines where you live, your zip code. Providers tend to be clustered in urban areas and limited in rural regions. So the greater the distance it is to travel to care, the lower the likelihood that people will get the care or the treatment that they need. That's a determinant. Just because you live in a rural area doesn't mean you shouldn't have care. But in reality, that's what's happening. All right, so here's a good study about the influence of geography. They use Medicare database. They took a look at people who reported that they had prostate cancer and where the providers were from 2011 to 2014. So they mapped the location for different treatments like prostatectomy, intensity modulated radiation therapy, and I'll show you some data of that. So they use regression to estimate what's the effect of if you have to travel more than 10 miles to go get care, what is the likelihood that you get care or treatment? So you can see it's just showing you a map of where the incidents were reported. And the good news was that 90% of people were located in urban areas where they had a lot of treatment. But when you go past that, we realize in the next slide, where on the left side, they're trying to estimate the effect. If you have to travel more than 10 miles, they're showing here that for the top one, for prostatectomy, if you have to travel more than 10 miles to go get treatment, the likelihood of go getting your treatment is less than 2%, lower probability. It was even higher for intensity modulated radiation therapy, about 3.8. So the longer the distance to go get treatment, the less likely that people will get that treatment. So geography influences access also to advanced diagnostic imaging. All right, again, rural and low-income populations have delayed imaging and less access to advanced techniques. So these delays lead to worse treatment times and worse outcomes. Also, imaging disparities are also a key upstream factor, key structural factor that needs to be addressed. All right? So here's an interesting study, also looking at geography. Here's an example where geography and your occupation conspire to make you more likely to face adverse health issues. A study from Georgia, we know that Georgia has consistently been having rates of prostate cancer higher than the national average. Also, the Peach State is known for its agriculture. So it's a lot of agriculture that's going on. So what this study did, they took a look at age-adjusted prostate cancer incidence. But they did some unique 3 things. They looked at counties where they're spending on agriculture. They look at spending on agriculture in terms of fertilizers and chemicals, and then also try to determine what was the incidence rate among counties where the proportion of the Black population was the state median. The state median Black population was 27%. And so what this study did, you can see right here, to the left, you can see counties where the African American population was less than 27%, so less than the county average. To the right, you could see counties where the African American population was at least 27% or higher. And what this study showed that when you look at the amount of dollars that was spent on agricultural chemicals, it was statistically significant in terms of the incidence at 0.04. When we take a look at the same thing, the same scenario, but comparing this to commercial fertilizers, it was marginally significant at 0.7. So geography, where you live, environmental factors, as was mentioned before, also impacts that. And then here's the last one I'll focus on. In terms of individual level factors, income, housing, all of that, this is a great study, a meta-analysis that looked at 47 different studies, over 1 million participants, patients, comparing Black versus white, looking at the association between social determinants of health and prostate cancer specific mortality. So what they did, they created a scoring system based on how those studies adjusted for social determinants of health. And what they found out was that in studies where social determinants of health was low, all right, you can see the hazard ratio there. Blacks or African Americans was 29% higher risk for prostate cancer-specific mortality. So this is showing you when you go to intermediate, the risk was a little bit lower. Overall, Black people were 8% more likely, all right, to have the prostate-specific mortality rate just based on their social determinants of health. So I'm going to skip this. You could talk to Dr. Bonner about comorbidities. He works a lot in that space. But I'm going to focus the rest of my presentation taking a look at the study that we worked on. We did a qualitative study looking at social determinants of health factors impacting African immigrants in the United States, trying to understand some of the key drivers that's impacting this population. I could see our participants, the investigators listed there. So we interviewed about 16 participants. Here you can see the different countries where they come from. You can also see the different age brackets. So we had a good mix from East Africa, West Africa, people who stayed here for a long time. We also have folks who've been diagnosed. Luckily, we saw many of them were diagnosed at early stage, stage 1. We still had a few who had late stage diagnosis at 4. In terms of their educational level, many of them had university degrees. In terms of income, many of them made less than $50,000. Insurance, you can see the breakdown of different types of insurance that they had. And the majority of them were employed. So this is the interesting findings. In terms of the treatment, what type of treatment were they taking? So you could see all the different types of treatment options that our participants opted for. You could see right here that the prostatectomy, almost half of the participants were electing for that. Now, part of this could be the stage at which it was diagnosed, but we wanted to drill down deeper. We wanted to drill down deeper. How are these folks making decisions about their treatment type? And so this diagram kind of gives you the substance of what we're finding in the data, all right? So right at the point of diagnosis, there's a range of emotions that these men go through. There's fear, there's fatalism, there's shock, but majority of them was solution-oriented. They wanted to take action right away, all right? Now, some of them, they'll listen to the doctors, what the doctors was offering in terms of treatment options. A few would seek a second opinion. And when they sought a second opinion, most of them were using informal care teams, people that they know from back home who are now in the US, perhaps not part of their treatment team, but getting information from them, using them as sounding boards. Now, when they did make a decision for treatment, you could see right that the different treatment types, but it did come with side effects. Some of those side effects was incontinence, social isolation, all right, erectile dysfunction, personality change, mood change, all right? They cope with this using faith, family support. Some of them were very resilient, all right? And we also see that there were some instances of mistrust of the healthcare system. That mistrust came mostly because of communication issue, lack of communication, or not quite understanding the way the system here works. When they went to meet with the doctor, instead of having their most pressing issue come up front, they were delaying, all right? And then we had decisional regret as one of the key outcomes that we're seeing in this study. So a lot of the men regretted the treatment option that they took, all right? So what are we doing to fix that? So in the area where we come, there's a high rate of uninsurance. So we're trying to form strategic partners with our local health department to do screenings to take away the barrier of lack of insurance, providing them screening at no cost. We also work with different fraternities, all right? We also try to meet men where they are, all right? We take a mobile clinic, we take a mobile bus. Dr. Francis is right here with IV. They've been using this for doing other chronic diseases. For the last 2 or 3 years, we've been doing that also with prostate cancer, engaging the men, having them get tested in the mobile clinic at no cost. Also, what we've been doing is sending messages, trying to educate those men about the importance of taking action about their health instead of waiting for someone to do that, to be empowered to make those decisions by themselves. You see, I have no hair left because my community partner is right here. Don, can you wave? You may have seen some of the videos on the barbershop conversations. So he's a strong advocate. He's a strong advocate in sharing, letting team members know what they could do. So we try to use trusted people to get the messages out to our people. So in terms of takeaway, disparities in prostate cancer stem from a myriad of factors, all the way from screening, all the way to survivorship. Now, addressing this means addressing some of the root causes. All right, how can we make sure that people have access to care? How can we make sure that treatment options are not just in the urban areas, but also in the rural areas as well? So I am done. So that's the team. And questions?
Thanks so much for sharing so many insightful points about the social and structural factors driving the prostate cancer disparities. And I wonder if any questions or comments from our audience. Please feel free to stand up and. When we give audience more time, we do have some questions from some audience. So what do you see as the most actionable structural factors that we should be addressing right now?
So right now, we should be empowering our men. There's a lot of stigma about this disease. We all know October, Pink October, even the NFL, they wear pink cleats. It's so customary for women to talk about mammography, but when it comes to men, to talk about prostate cancer, for some reason, there is some stigma. So we have to take away that stigma. We have to normalize, we have to mainstream conversations about prostate cancer. So people feel it's okay to talk about prostate cancer. It's okay to have conversations with your doctor. If we allow the stigma to continue to dominate, men will still be shy, and then we're still having advanced stage, people presenting with advanced stage. So it's a disease that, if found early, it's a 97% survival rate. So the question, the urgency that I see, and which is what Don and I were trying to do, is to get the word out. Get yourself checked up, especially if you are from African descent, where we know we die twice as much as other ethnicities.
Thank you.
Hey, Ernie. I know you and I work together at the barbershop on this project. And I want to thank you so much, because when I went through prostate cancer, I didn't have no information. I think when you first came to the shop and I told you I went through, I didn't have no information. So now we are trying hard in small town, rural town. We are now trying to get more African-American men to come out and be tested. And you know, we are at a roadblock right now as far as trying to create ideas, get people to come in. But I just want to give him his flowers now. You know, he's really helped in the community, because we didn't know anything about this. I mean, one thing I credit him for is that when I went, I didn't get no second opinion. The doctor said, hey, you got prostate cancer. And as I said in the video, listen, I said, okay, because council came up and I said, it's time for me to get it done. But when he came in and started more information, I'd be trying to give the guys at the shop now, because no one gave me that information. And so I thank God for him for coming in the barbershop and just relaying the messages and everything to us, because we were lost. I mean, I was just lost. So now at the shop keep pamphlets and everything, and all we got is phone number so I can call him. But people don't understand, in a rural town, we don't have no help. You know, here we don't have no help. I mean, if you have some churches sometimes might say something about, but most of the time we don't have no help. And I've been at that shop for 18 years, and my father-in-law passed from prostate cancer, and we had no information, no one relayed the message to each other. So we were just lost. I mean, you all don't understand what I mean. We were lost. I mean, we had nothing. We go to a doctor and basically they say, hey, you know, the first thing they say, you know, we're going to remove your prostate. That's the first thing he said to me. I had a wife. She's not, well, I got a wife. I got one more. Let me clean that up. My wife like, no, it's got to be some other treatment side. But the point I'm making is, from my personal experience, being in a rural community, they give you just one alternative. They say, remove your prostate. And so he have helped out so much down there trying to get information out to God. I just want to thank him so much for it. Thank you, Ernie.
Dr. Nagafor?
We'll get to your question
right.
afterwards.
All right, Chair, you and I can talk after. So,
Yes. Now it is time for our break. We're going to come back at 10:35.
Can we take one more question? For sake of time, we take one last question.
Yeah. Yes. Yeah. This is not a. It's just. I want to advise on something. Because of the part of the body where this prostate cancer affects, Black men most of the time are not willing to come out. So I think if we can empower the women, the wives, the girlfriends, to pressurize their husbands or their spouses to go to the doctor for this test. I'm saying this because when I was having my radiation, when I went to a doctor's office, the white men who were there always came with their wives. So when you go there, they don't.
Even at the time when you're being diagnosed, mentally you're going through a lot. You need somebody there who can take notes, listen, calm. So we're working with the women. You saw one of the slides I showed. We're working with the sorority because they really help to drive grassroots participation. But you're right, it's about educating the young men early, because the old generation may be facing that stigma, but we're finding out the younger generation are not so stigmatized. They're okay talking about some of these things. So there is hope. If we focus on the younger generation, some of that stigma no longer persists. Thank you.
on research and grant writing. The other section focused on research training, and the last section focused on publications. So while we are here and does anybody have the mic? Professor Rotimi, can you just give us a brief highlight of the next steps for yours? Either come on stage or can he have a mic? Thank you.
Yeah, thank you very much, Prof. So we discussed about the evidence, the science that we've done in iCCaRE phase 1 and phase 2, and the current data that we have. And the idea is to step back a little bit, look at the big picture, see where we have data to fill in that picture, and where do we go from here. So our group was to focus on the research aspect and clinical trial. And we did a little bit of we did a space at time available to look at basically what iCCaRE is about, which is the social determinant of health and the biological determinant of health, and how those two factors come together, including access to care, to influence prostate cancer risk and overall outcome of the Black patients in iCCaRE. But we quickly realized during our conversation that we should look at all men, while we know where we're going eventually. And so that's a very important conversation that we had. So one key thing that came out yesterday in our talk was our outreaches have been such that we're using the metrics to know how much people we're reaching out to, we're touching. But we've not taken into cognizance how many we are converting. And so moving forward, we look forward to designing research that will measure our outreach in the way that we can determine conversion. And where my people, I don't have my computer here. All of those C's. Mr. Meriwether, do you remember? I have it written on my computer.
Yeah. Oh,
Mr. Meriwether.
The slides are ready?
Oh, okay.
Thank you. No, go finish your thought.
So yesterday, we talked about conversion approach in our outreaches.
I think Dr. Omar knows it.
Yes. Conversation, conversion, completion, and scalability.
Okay, thank you.
And we're looking for the C word to replace scalability. So if you have any idea, please share with us. Thank you very much.
Thank you, Professor Rotimi. Dr. Bolajoko, could you come and stay here so that you can tell me when it's 5 minutes more? I really want us to get on track. So full project 1 is focused on improving quality of life through the point of prostate cancer diagnosis. Okay, now it's not, okay, what's going on? Okay, no relevant conflicts of interest. This is the power team for full project 1. As you can see, myself and my co-PIs, we have multiple principal investigators. So this project is led by myself, Mr. Meriwether, and Dr. Ngufor. We have really outstanding clinicians and other survivors that are working with us. So I'm going to really do my presentation in a way that talks about the problem, and it's really easy to kind of follow on how we are really implementing the solution for this problem. So we have all understand from Dr. Kaninji's presentation, thank you, Dr. Kaninji, that the prostate cancer disparities is really a problem. It's not only about the burden, it's also about the significant mortality, it's only about also being aggressive, the fact that there's an upturn now in advanced disease because a lot of people are not doing screening. But another thing that is starting up, and we are writing a grant on this, is the fact that we are now having early onset of prostate cancer in people who are less than 50. In fact, we have a grant that is due this week that is going to allow us to set up a registry for early onset of prostate cancer. So men of African ancestry feels a disproportionate burden. We developed this, many of people who participated in the research are in this room. We develop a community-based model, not a theoretical scientific model of the status of Black men at the point of prostate cancer disparity, what they need at that point, prostate cancer. So our study is really based on addressing this. So let me kind of set the stage. Just kind of think about it. Just imagine that you are Mr. Johnson, a 58-year-old man, recently told that you have prostate cancer. You are kind of hearing Gleason score, PSA, active surveillance, radiation. The things that we learned from the study that we did is like at that time you feel shocked, you feel fear, you are uncertain. Within minutes, you must begin to think about the solutions, about the treatment. Maybe you have a follow-up an appointment, and you're thinking about, okay, what am I going to choose? You know, to be able to treat something that I just learned a few minutes ago about what is going on. So the question is that, and this was not something that we asked, this came from a grant that was funded in 2013 about where should we begin the intervention. And so the men said, start the intervention at the point that we are told that we have prostate cancer. So at this point, there is a lot of challenges that men face, emotional distress, information overload, complex treatment choices. Dr. Kaninji, spread out what he's finding among the immigrants. They report, one of the things that we are finding that after making these choices, people are reporting decisional regrets. We regret the treatment that chose, maybe because we did not have time to be able to do it. And there's mistrust and there's needs to support them by cultural or emotional needs. So what we focus on is that how do we support these men, if you look at the bottom rank, to make sure that we eliminate the decisional conflict, to make sure that we improve the trust in the medical system, to prevent them with access to what is going to help them to do what they need to do. So this is the solution. Now, here's where you pull up your phone. The solution is right there. Do a screenshot. Don't play it. Just do a screenshot. You can play it later on. Because no, don't play it. I'm going to play it for you. Don't play it, people. Okay, why is it not playing? It's not playing. IT.
Prostate cancer diagnosis.
Okay, that's fine.
Fears, anxiety, and moments when the world feels heavy.
I guess there's no volume.
Prostate cancer diagnosis brings questions, fears, anxiety, and moments when the world feels heavy. From the point of prostate cancer diagnosis, the trans.
Okay, maybe you play it during the break. That's okay. You have the solution on hand. Let's play it during the break. I'm trying to get
to prostate cancer survivor.
Yep. Okay. So you're going to see what the solution is. You have it on your phone and you can play during the next break. So the goal for us is to empower newly diagnosed prostate cancer patients. And so in doing this, what they have identified that they need at the point of prostate cancer diagnosis is oncological support, emotional care, navigating them through the social determinants of health factors, and building trust through digital empathy. So what we decided to use is what is called immersive technology. And why are we using the immersive technology to support men at the point of prostate cancer diagnosis? Because the traditional pamphlets and short consultations that I had, they fail to support the comprehension because there's such a lot of information. The immersive environment allows you to go into 3D. Sorry, you could have seen it in the video, but you can look at it later on. The immersive environments allow you to go into the 3D environment in which you are actually there making the right decision and seeing all the things that you need to make decisions about right there. And also, it can create a calming experience for you. When you go in at the beginning, it calms you down to be able to process everything that needs to be processed. The VR enables experiential learning rather than passive information, and it provides the opportunity for you to personalize the information to you directly as well. So what are the specific aims? I'm going to talk about what we've done in phase 1. I'm not going to report on phase 2 for this project. And please do not photograph because right now we're still, I think we got one paper accepted and we're working on another paper. Yes, so we want to make sure that we are published before we put this out. So our aim for phase 1 was to plan and develop the PPC, a point of prostate cancer diagnosis VIRA that provides social determinants of health navigation services, psycho-oncology support, and emotional support using qualitative formative research, and to establish that Black men, especially prostate cancer patients and survivors, they accept it and they feel that they can easily use it. That's what we focused on. This is the methodology that we use for the VIRA development. You know, in addition to the fact that we had prostate cancer survivors on our team, we also did focus group with more consumer advocates. We did interviews with clinicians to figure out when is the best way to use it and how is the best way to use this. We had qualitative analysis, coding, and everything. So this was really very rigorous design. Then we developed, this is one of the things that a lot of people miss when they are doing intervention for individuals or community. They do not develop the communication strategic framework. So after gathering all this data, we developed the communication strategic framework, which is what, where, when, how, what does it need to be, including summarizes all the information that we gathered, not only from prostate cancer survivors, but also from clinicians as well. Then Dr. Ngufa appropriately led the development of the VIRA, which was quite interesting. All the filming, all the things that went behind it. Is going to talk about it when he talks about the digital health core. And then we did quality control care. So just kind of think about this, and you can see more about it when you go to that QR code that I gave you, because it gives you the insight into what exactly the VIRA looks like. So this is the VIRA workflow. You know, is where, just kind of imagine that you launch the VIRA and you complete a questionnaire assessment form. And this assessment form gets to what do you need for psycho-oncology support? What do you need for emotional support? What are the social determinants of health challenges that you currently have? The questions, when you answer those questions, they are put through an artificial intelligence engine to make personalized interventions for you. So if Mr. Gainey goes in, the interventions that comes out from Mr. Gainey is going to be different from if Mr. Merriweather goes in, because everything is personalized to the way that Mr. Gainey has already answered those questions. And then, you know, we have it in 3D, the first one that we did. 1 minute, look at, with all that technology problem that you all have, I'm going to finish this thing. So you put on the headset and you are taking in an immersive journey, and you have the option to interact as much as you can interact. I'm saying I have more minutes. You can read the methodology sometime else, but this is a VRTD at a glance. This is kind of what you go through, the different things that you have to be done. This is the 2D at a glance, and I hope that Dr. Ngufa will talk a little bit more about that, because in addition to the immersive environment, we want to have it to be app-based as well as to be website-based, so that you can actually do that as well. So since I have 1 more minute, I'm only going to focus on what is important. But you know, overall on the result, it shows that a lot of people, we didn't have anybody who was dissatisfied or neutral. They love it. They love it. They think that is excellent, very good, or good. You know, these are some of the things that the men are saying, which is even much more important than the quantitative data that we have. You can see someone saying, what it did for me is made me realize I am not the only one. There's the emotional support component that you can literally go in and you are in a barber shop and you have other men, and these are real men. We have former Senator Tony Hill, we have Pastor McCall, we have Mr. Arnold Merriweather, we have the late Mr. Aguala on there, and they're chatting, and you can go on into that environment in 3D, and you're sitting down and you're with them on the barbershop. And based on what the questions that you have answered in pre-assessment, they're talking about the things that matter to you, and you're able to hear the things that matter to them. I knew I wasn't the only one, but this helped me realize I'm not the only one that would talk about it. For me, the biggest value of Viral is here's 4 men that are still alive and they're talking about it. We have to meet people where they are, so you have the ability to meet people where they are. We not only did this here, this is why iCCaRE is global. So although it was developed in the United States, but we also took it to Africa, and we've taken it to 2 places in Africa now. It was in Tunisia, and we also took it to Nigeria as well. And these are prostate cancer survivors, and I hope they are online and they're seeing themselves on here. But we have Mr. Adejaro as well as Mr. Dennis Olise, who tried this and just kind of thought, oh, it's time to bring it to Nigeria in different languages for us to be able to use as well. So the impact really going through this, so we only have one question for me because I'm going to go through the impact. Here you have a clinical problem, which is Black men experiencing the highest prostate cancer mortality. The gap in care that we found for this project is that a diagnosis, emotional shock, information overload, mistrust was not being addressed for this man. So we are seeing the actual implementation and translation of the work that we are doing. And the opportunities was for us to use digital tools and culturally tailored support for this man. And the solution is this Vera immersive intervention. And we've been able to establish that it is feasible and early outcomes are very positive, and we are now thinking about how to integrate it into clinics now. So Vera can do a whole lot of things from shared decision-making to building patient trust. Implementation considerations are really high in how we are going to integrate into different places, and we can scale it across health systems as well. It has great feasibility, yes, cost implementations. Dr. Ngufor is in charge of handling the cost to make sure that it's low cost when we put it out. We are working very closely. I'm not sure whether Dr. Christopher Williams is here. One of the good things that we have is that we have a urologist on our team. So we are actually working with Dr. Christopher Williams to make sure that it does not impact the workflow of the urologist. And he's just been, congratulations to him. He got an award yesterday because of the outstanding work that he does in giving us input into how this can be integrated into the urology clinic in a very seamless way. So right now, it's phase 2. We are doing our randomized clinical trials, evaluating clinical outcomes. Long-term survivorship impact will be next. Integration with AI-driven personalization into clinics after we do that, and then expansion to other cancer populations. Everywhere we go, people want it for breast cancer. They want it for colorectal cancer. They want it for all kinds of cancers. They want it in different languages. They want to add spiritual distress. So we're just telling people, let's just establish this as a clinical trial, and then we will be able to get that done. So I'm not going to go into this. Thank you. It takes a village. We have a global village. We have the iCCaRE village. We have the CHAP village. We have the Captaincy village. And we have the most beautiful village of all, my grandchildren.
Yeah. Dr. Odudna. Dr. Odudna, do you want to take questions or you want to wait till after?
I am proposing that we have other presenters, then we can have a comprehensive Q&A. So thank you, Dr. Dedino, for the presentation. I'm going to bring on stage right now Dr. Rosanna Dronka. She's going to share an outstanding research. We're going to see possibilities of having therapy at home. Thank you.
Thank you very much for inviting me, and thank you for allowing us to present our research on cancer care at home. And I hope that my slides will advance. There you go. So our project in iCCaRE was focused on understanding preferences and impact of home-based cancer care for Black men with prostate cancer. This is just a part of our team, myself, Senator Tony Hill, as our community project leader, Adam Case and Trevane Matthews Hugh are two early stage investigators who are very active in the project. I think they both probably have clinic this morning seeing prostate cancer patients, so I think they are forgiven for not being here. Kayla Falzone is back in the room. She is our program manager, and then our coordinator extraordinaire, Jesse, who's been really instrumental in getting a lot of our patients to participate in this research. So why is home care important, or why does it matter for Black men with prostate cancer? Well, I would say that for decades or forever, probably, we've built cancer care around institutions. People traveling to clinics, to hospitals repeatedly to get cancer care. And I think we're shifting that narrative now, especially after COVID, which I think, brought up the importance of being able to access care, whatever happens in the world. Now we're thinking about, putting the care around the patient rather than around the institution. When you receive a cancer diagnosis, you've heard from Dr. Odedina, it's a tremendous impact on somebody's life. It's really turned upside down. Treatment has a significant impact on our patients, but there is also, in a way, the unspoken or less talked to burden of cancer care. And that's the travel burden, that's the financial burden, that's the emotional burden for patients. And bringing the care to patients in their own homes and their own communities, it's not only about convenience. You've heard Dr. Kaninji could not have said better. Geography matters. The farther you are from the clinic, the less likely you are to receive treatment such as surgery and radiation, which is, a limited number of treatments. Think about treatments that you have to go for every week or every other week or once a month. It is even less likely that those patients will receive the treatment and actually complete the treatment. So bringing the care to people actually also improves clinical outcomes by allowing them to finish their treatment. Having the care delivered to their own home with the same quality and clinical oversight also allows us to reach patients to treat their symptoms and complications early. So we prevent some ER visits and hospitalizations and allows people to have better quality of life. So for us, we definitely saw all the benefits, but we wanted to make sure that we don't assume that patients want what we want for them. So actually, our first project in the pilot phase of the iCCaRE was exactly this, asking our patients or prospective patients, what do you think if we were to tell you that we could give you care in the home? What were the pros and the cons that you would think, are important? So to do this, we surveyed 50 patients with prostate cancer actively undergoing treatment. We also had a focus group with 5 of you in this room with 5 consumer advocates, prostate cancer survivors. And we also had in-depth interview with 10 patients who answered our surveys. So the focus group, I would say, was enlightening. It brought up, the themes that we actually thought, but it brought up other questions as well. Some of the cons, I'll start with that we didn't think about initially. The privacy concerns that actually informed how we developed the program, further down. Some people said, I don't want to have my cancer care in my living room. I don't want my church friends to come in and the first thing they see is the technology pack that you have there that the nurse can access. We didn't think of that. It was enlightening for us, right? Because we thought, okay, we're bringing the care to you that's, medically very important. So we actually took this to heart and we developed that technology pack in a collapsible bin that when you're done, you can put it in your drawer, under your bed, wherever you want. So there is no cancer care in your living room. For the patients who want to have it out, great. For the patients who don't, there is this option. The safety concerns, for patients, they wanted to make sure they know the nurse who's coming into the home. So we wanted to make sure that the same nurse or the same folks come repeatedly so people know them. So a number of themes, I think, that were extremely helpful as we thought about putting this together. The questionnaire really focused on, as I mentioned, the pros and cons. Do people appreciate more that being in the home, they are not in contact with all kinds of hospital-acquired infections, that they don't have to look for parking, that they don't have to drive, that they are being in their own home, in the comfort of their familiar surroundings with their friends? Is that helping the healing, as you receive the treatment? Or were people worried that if I'm not in the hospital, maybe I'll get less quality care? I'm not right there next to the nurse. What if I need an emergency intervention? I like to keep to a routine. I like to see the other patients share stories. So we wanted to gather this from patients. So please, like Dr. Odedina said, don't take photographs. We haven't published this yet. It's really hot off the press, our statistical analysis from pilot project 2. And we really heard from patients what we thought we would hear. Patients really were favorable, or at least neutral. Most patients favorable to receive care in the home. They appreciated the convenience, the reduced need for travel, being with their own family and friends. And very few, the scores, as you see there, low in the 2s. Very few people worried about privacy, safety concerns, about not having the same quality of care. When it comes to what treatment they wanted in the home, hormone therapy was clearly a win. People saw absolutely no need to travel to clinic to get either tablets or to get a shot every 3 months, that you can easily do it in the home by a nurse, or even sometimes patients by themselves, so they could do it. But they were also open to receiving injections, other injections, or IV infusions in the home. So in conclusion of our pilot, project 2, we found that there is a strong readiness to receive home-based care from our patients with prostate cancer, that they felt this addresses some structural barriers that really prevent them to receive the care that they need to receive. So this was very encouraging and led us to open our clinical trial to actually offer patients care in their own home. So this was very timely, because around the same time that we started iCCaRE and we started this project, Mayo Clinic actually launched the Cancer Care Beyond Walls program, offering care to patients in their own home. So I'm one of the physicians leads of this program. So it was extremely insightful for me to bring some of the learnings from iCCaRE pilot as Mayo developed this program for all patients with cancer, not only prostate cancer. So we started a proof of concept care in the home with 10 patients in April of 2023. You see here, most patients were colon cancer, 1 patient was breast cancer. And we told people, we're gonna give you care in the home for 6 months, and then you can return to clinic. At the end of the 6 months, 0 of the 10 patients wanted to come back. They all wanted to continue care in the home. So again, it was a reinforcement for us that we are on the right path by bringing this, by serving this need that we heard from our patients. Thank you. These are some of the testimonials, as Dr. Odedina, put them up, from Vera. You see here exactly the same. The emotional support, the ability to go about your life and not put your life on hold for cancer treatment, that was huge for patients. So we recently published our model of care, showing how we do it in New England Journal Catalyst. And I will just tell you a little bit on how we're doing it. There is a sophisticated logistics system that revolves around being able that everything that we do in the clinic, we do it in the home with the same quality, the same clinical oversight, so you don't see a difference from a medical perspective. A whole team that makes sure that the supplies and the medications arrive on time. We partner with home health nurses in the home that we train, so they deliver Mayo quality care And we have a command center that's staffed by our chemotherapy trained nurses and practice providers, and also supervised by physicians to ensure that they supervise the nurse in the home and they are there at all times for any emergencies. We have a paramedic network that if something happens, can go to the patients, and the patients have a tablet in the home that allows them to connect with their team immediately. You see here the big button. People have no problem with this type of technology, very user-friendly, allows them to immediately identify how to connect, show the schedule, who's coming in my house, for what and when, so patients can plan for that. There's a number of education resources, that are there as well. They can measure vital signs at home. They have ability to do blood pressure, pulse oximeter, thermometer, and they are taught on how to do this the day that they start in the program. This is a sample program you see here, the day before your treatment or a few days before. Actually, you have the technology installed in the home, all the education, provided. Then you have laboratory testing done in the home. Somebody comes, draws your blood. The blood is processed at Mayo Clinic, result in your medical record. And then by the time your treatment comes, you have a virtual visit with one of our APPs. And if everything is okay, they put the orders in. So the chemotherapy is being, or immunotherapy, or biological therapy, or hormonal therapy is being delivered to your home, where the nurse comes in and administers this safely. If you have any issues in between treatments, the patients find that's a lot easier to access us. They contact the team and say, I have mouth sores. I wasn't able to, eat for a couple of days. What do I do? We can intervene a lot faster. What we learn in the clinic, a lot of times patients won't say anything for 2 weeks. When they come, they have not had, much to drink for 2 weeks. Their kidneys are affected, and it's a whole downstream of problems after that. This allows them to reach out a lot sooner and allows us to bring fluids in the home, or advise patients what to do and intervene a lot faster. So now this program has grown. We have 5 active clinical trials. iCCaRE proudly is one of those trials, active right now and enrolling patients. We have 3 patients who are in the program, with the first patient to be treated next month, I believe. So this is the schema of the clinical trial. Patients with prostate cancer, Black men with prostate cancer receiving any type of hormonal, chemotherapy, immunotherapy treatment, biological treatments. They get the first 2 cycles in the clinic to make sure they tolerate it well. And after that, they go in the home for up to 6 cycles, and then they resume the treatment in the clinic. When we do this, we want to make sure that we ask patients, what do they prefer, the clinic or the home. We look at their clinical outcomes and a number of quality of life metrics to ensure that we hear the voice of the patient. So this is my last slide. I think bringing the care in the home is not only practice changing, is actually paradigm shifting, is really rethinking healthcare from a patient perspective, making it more flexible, making it more personalized, allowing people to have access to care, to the standard of care treatments, but also in the future to a lot of the innovations, to clinical trials, a lot easier than if they lived only around Mayo Clinic. One of our trials is actually open in the Florida Panhandle, 3.5 hours from here. We ship all the medications there. Our nurses don't go there. We partner with local nurses to be able to deliver the care. And we think that's the future, Dr. Kaninji, for rural health, being able to reach patients in all the communities that traditionally have had difficulty accessing care. So thank you to the iCCaRE team. Thank you to the iCCaRE support. This, really has not been possible without all of you. Thank you.
Great. Thank you, Dr. Dronka. This is outstanding. The future is here, and iCCaRE team is driving the future of healthcare. So we still have one more presentation to go for this session, and we're going to be hearing from Dr. Kim Babel Johnson, who will be presenting on behalf of Full Project 5 on their discovery on vitamin D. in advancing healthcare. Thank you, Dr. Babel Johnson.
It's definitely a longer walk than I thought it would be. Good morning. I am not Dr. Colon, the PI of this trial, nor am I Dr. Pupula, another one of the PIs of this trial. I am Kim Barbell Johnson, and I actually serve at Mayo Clinic as a new program director in the general internal medicine space in women's health for the last few months. But this has really been a love of mine to work with the iCCaRE team since I returned to Mayo Clinic a few years ago. And again, it's not my trial. However, this is a very fascinating area of science. And so because I do not have the privilege of sharing the data as a non-PI in this space, I wanted to spend the time this morning kind of helping to understand not only what we're doing in this trial, but why we're doing what we're doing in this trial. And it really kind of follows the conversations that we had yesterday as where do we go next. So with that, I hope that my slides are available. Oh, I guess they're available for me. No diva here, right? So again, the trial actually that Dr. Colon has had running in the United States that we use to inform the full Project 5 for the iCCaRE trial has similar aims. So the study is to find why replacing vitamin D or replacing vitamin D in men with metastatic or non-metastatic history results in normalization of their immune function, so normalization of their ability to mount an immune system. And the hope of that is to identify if vitamin D insufficiency or not having enough vitamin D contributes to more aggressive prostate cancers. And so while that's a mouthful and that's a lot, I think it's, again, important to understand that why we're doing this is because prostate cancer is not something that's distributed equally, right? So we know that neither access or biology of this cancer actually is well tested in the literature. So Dr. Colon and Dr. Pupula in this study is actually looking at the intersection of what we know about vitamin D and what we know about prostate cancer, and also trying to understand more about the inflammation, the tumor signaling, the ancestry-informed molecular differences between populations, and the structural integrity that we can find out in doing this trial in ways that we can no longer afford to ignore. And so that is why this collaboration was formed. And I'm going to actually be apologetic to Mr. Merriweather in advance because these slides are busy, but I'm going to try to water them down as much as I can, given the fact that I do want you to walk away with a little bit more information today. So the cancer burden and what we know about this topic is not new information. Prostate cancer carries one of the largest racial mortality gaps. It is not fully explained by access, and even with equal access systems, there are studies in the VA system here in the United States that showed that access actually didn't totally resolve the issue. Black men showed higher rates of metastatic disease, and when equal access was necessary and provided, we had the same numbers. But those studies do not replicate in many systems. And so we needed to understand, is access just coming to a place or having the access taken, like in the study that you heard with Dr. Dronka, brought to you. So this trial is not only a trial about vitamin D, but it's a trial about decentralizing access. And hopefully it will help us to, again, push what we know about vitamin D, but also look at those really germane host factors for inflammation, vitamin D, tumor signaling, et cetera, because now we can go to geographically to the sites of where our men are living. So this global study is important because in Africa, we know that prostate cancer is major, and we also know that there's a growing burden of prostate cancer disease in many low and middle income countries. We're focused with our study population because we know that men often present with advanced disease, and these diagnostic pathways are often delayed because of both treatment resources and the constraints of social determinants of health, like transportation, et cetera. Nigeria was especially important to us, not because of only because of the disease burden, but because Nigerian investigators and our seasoned principal investigators there have already helped us to understand that the tumor data sets, the information that we know about tumor, are actively reshaping science. And so the partnership is especially important to Mayo Clinic and important to the United States. It's important to prostate cancer survivorship. And this distinction matters because African men then are not simply an extension in this study of the United States study. Nigerian tumors reflect a distinct gene or genomic history. We know and we're learning from others that the environment is important, UV exposures are important, treatment realities are important. And so globally, the importance of the study is not just about vitamin D levels, but about understanding that relevant vitamin D agenda must include research that is conducted in Africa, not an extension to Africa. So the pathophysiology, and that's a word that just means how does this actually happen in the body in the science way, and how does vitamin D actually alter cancer biology? It should be the question. So when we're talking about this, you know, it's not like vitamin D is important, go to Publix or go to your grocery store and start taking 2,000 units of vitamin D. It's really important to understand why we're actually studying this. So in the biological form, vitamin D is not just a circulating molecule. The effect depends on where the local activation of this vitamin D and the vitamin D receptors are functional in immune cells and in tumor tissue. And that's why we're looking at that. We're looking at this very busy slide to understand how all the mechanisms of vitamin D, both in how it's impacting the cell cycle, how it's actually impacting cell death, how it's impacting the necrotic factors that we actually study here in our science labs, how it's interacting with other hormones, with androgens, the principal hormone in males, and these receptor signaling mechanisms. So it's really not just, again, about a study about vitamin D and vitamin D replacement, but the bottom line for us is that vitamin D is not a single pathway, and that we need to understand more about the system level impact and the tumor behavior where vitamin D is concerned and how it's impacting the inflammatory tone of our prostate cancer participants in the study. And it becomes more complicated because vitamin D is not produced with this clean narrative. You've probably already seen, as with many other areas of cancer science, that there becomes lots of studies that conflict with each other. And with prostate cancer, there's no exception. We know that vitamin D has been studied quite a bit. We know that we've seen large supplementation trials where vitamin D in repleted populations in men and women who don't have high levels of vitamin D are tested. But these studies have not shown a clear reduction in cancers across all studies. And particularly in prostate cancer, we have not seen a clear reduction of prostate cancer incidence. So some observational studies that we've seen have raised concerns even about very high levels of vitamin D. So the lesson that we're learning in this space is that if we ask the wrong question, we're going to get misleading answers. And so we've got to be at the table, because you heard that from Chelsea, right? If you're not at the table, you're where? On the menu, right? So we have to be at the table here in this space because, and see, last night, tonight, it all connects, right? Incidence in repleted populations is probably the wrong question, right? Just, is low vitamin D causing or contributing to cancer? That's not the question. The better question is whether deficiency, inflammation, and tumor competence for vitamin D signaling together define the subgroup that we're looking at, where the intervention matters. So this is a very complicated and layered study. And again, because of the, not systems, but because of the biology, the pathophysiology of how this impacts different people based on their ancestry is really where we're hoping to go when we get all of the information. And while I'm very hopeful, we don't have that data set, and that's why you're not seeing the data set, because we're still collecting all of our samples, and then at the very end, we will collaborate with the US samples and the other samples to put that together. So because I'm going to be out of time in just a second, I just want you to understand that blanket supplementation is not what we're trying to do, just replace vitamin D in men. We have lots of different trials that's showing the signaling that we're looking at. One of the most important developments that we continue to see is that the study populations that we've been really being informed, that has been informing our science, has been overwhelmingly white. And that means that we must continue to have very specific Black men in our study so that we can actually not only replicate some of that science, but actually develop science of our own. Our clinical trial design is important. And so this trial was designed to be global. It was designed to be a true partnership where those patients and the participants in the United States are having the exact same study across Africa in the partnership. And hopefully there will be sub-studies that can grow from this to actually help us to take other studies with the centralized clinical trial mechanisms that we're using in this trial throughout Nigeria. So I'm just going to cut across the field here, and hopefully, I don't even know where I am, but I think I'm almost at my last slide, and I think that's most important for us to, well, I'll just go back for 1 second just to say, give credit again to the principal investigator whose heart has been in this space, Dr. Colon Otero, for this trial. The study has been successful and continues to be open in the United States, and it's been a true partnership to work with Dr. Pupula and the team in Africa, in Nigeria specifically, to enhance this study. And lastly, I just wanted to say that for the population that we're studying, again, the prostate cancer is a very layered study. We're not looking at vitamin D replacement. We're looking at the molecular aspect of this disease and how immunology is actually impacting. And we just continue to ask that we do ancestry-informed, we're pushing ourselves for ancestry-informed answers. We're looking at next generation modalities for us to understand how this impacts chemotherapy and treatment and how this actually impacts patient selection and biomarker. So lots of evidence in this growing space. I believe these slides will be available to the scientists and to those who are interested. So I sourced information from 2021 to 2026 so that you can see some of the background that we've had. And just again, to understand that prostate cancer, particularly in African descendants, and then the global community remains a biologically compelling area of science for us. So thank you for the time.
Thank you so much, Dr. Babel Johnson. This is great. Now we have 30 minutes for questions. I want to thank the presenters for helping us manage our time. Honestly, I was thinking we're going to run out of time. So please join me. I think we're not necessarily going to bring them back up. They're going to be answering your question. If you have questions for Dr. Dedino, Dr. Kaninji. Okay, please, we're going to have to take question that is related to the home care, home cancer care. Please just let us know if you have any question. Okay.
Good morning. It's still morning. For the home care, is there like a mobile bus or something like that that goes to the locations, or how does that work?
Thank you, Dr. Oderina. Yes, there is a mobile bus. We're not using it yet. There's one that, a project that actually Dr. Oderina brought to Mayo Clinic in Florida. Right now, the care is actually the nurses, the home health nurses, locally based, are traveling to the patient's home. So we partner with a number of home health agencies, with Brooks, with Concierge, with Bright Stars. So right now it is really those nurses located geographically close to the patient. But for instance, for our Florida Panhandle project, we are actually also going to be using the bus for some locations where there is no nurse agency. And believe it or not, in the Florida Panhandle, there are many locations where even the home health agencies have a very limited footprint. So for those, we actually will be using the bus, and we have built in a questionnaire in that study to also ask the patients, what's your experience like in the bus? If you're gonna get treatment in the clinic, maybe in the home too, and in the bus, what do you think? Because we really think that in the future, that's going to be a way of, reaching people, parking in the church, or maybe not in the church, but in the Walmart, or in a place where everybody knows that's closer to them. Thank you for the question.
Another question. So would the bus, say if someone was having some type of health fair, would the bus be available to come out and be parked at the health fair so people in the community could see the bus and what the clinic brings?
Monica, this is for you.
So the short answer is yes. So we are doing community open houses with our mobile research unit, where we're going to various locations so that community members can become comfortable with the research unit, see what's on it. And so as Dr. Dronka said, we're also identifying studies as well that we'll be taking out. So you can find me, I'll connect with you, but I'll be here all day, and so we can definitely have a conversation about the mobile research unit and bring it out to your community.
Thank you. Okay, we have a question from Mr. Francois.
My question, just like maybe same little bit difference for this question. If your home care, if it's someone, I know some guy, he don't have wife, no children, he got prostate cancer. That's just me all the time to take him to the hospital sometime every 2 weeks. You don't have nobody. If you got a couple people, something like that, how program at home care can help him? You can have somebody like that. You can go pick up him when you get to the doctor, because I've been there. I know this guy, you don't have anybody. That's me. You can see a couple people, just like a couple men, be like that. You live some by yourself in the home. You don't have nobody. If you know someone like that, how you can to help him to able to assist him for the medical?
That's a great question. And actually, those are the patients that the program is built for, right? You want to serve the patients that need it the most, not the people who live, 10 miles from here and they don't want to park. So you want to really be able to help the patients who really need this. So you don't necessarily need a caregiver. If you're able on your own to interact with the nurses, to take care of yourself, and I presume that if you live alone, you can take care of yourself, then yes, absolutely, this program is for those patients. We have a patient who is blind, right? And we, I think, have a patient who is deaf. So we really, our team is striving to be able to help all the patients to participate into this program.
Thank you so much, Dr. Dronka. I do have a question myself. Yes, Dr. Dronka, I want to ask, when in the pilot project, were the caregivers interviewed with the prostate cancer patient?
That's another great question. No, not in our clinical trials right now, but that's actually our next project. We are opening a trial for brain tumor patients, and those patients have a lot of needs, a lot of difficulty, right, with mobility, with speech, as the disease progresses, the patients are more and more, incapacitated, and the caregiver takes more and more, right, of the role. So we feel that that's a great program to start interviewing the caregivers. In the beginning, we were so focused on getting the program going, helping the patients, and now we're starting to look at those important aspects: caregiver burden, bringing other types of care in the home, physical therapy, the wraparound care around the patient. So those are the steps we're following now. So that's our next trial.
Thank you. My follow-up question, would you be open, because you just mentioned that the full Project 1 have started recruitment, would you be open to have a small project or a supplemental project that will be considering the caregivers of those Black men that are working with for full Project 1?
That's an easy answer, 100% yes.
Okay, I'm going to connect with you. Yes,
you.
I'm actually working on caregivers, and I would be happy
Awesome.
to
See, that's why
do a
iCare is
supplemental
useful.
project on that.
Yes.
Thank you.
Dr. Drunka, great study. Thank you for being here. My question is more so, how does the program try to reduce barriers for patients to access or any other barriers for patients?
Very good question. So the program really is focused on, as I mentioned, addressing the geographical barriers. You know, that's one of the most important ones, reaching patients that traditionally have had a lot of difficulty, you know, coming to the clinic. Financial barriers too, because a number of our patients who are in the program, for instance, in the Panhandle, they mentioned to us that they are really using their retirement funds to pay for the travel and the overnight, you know, stay in Jacksonville to receive treatments. So being able to bring the care to them, I think, is important. As our program grows, we are now partnering with insurance companies, with Florida Blue, with other insurers to make sure that we create the pathway for reimbursement, you know, at parity or maybe even better than at parity to if you had the treatment in the clinic, because we argue quality of life is better. Patients complete the treatment. We avoid ER visits. Look how much it costs when a patient goes to the ER. It's $30,000. You pay a home nurse for, you know, a year with that, right? So we are telling the insurance, the payers that really we are saving you money. We're starting to work with CMS, you know, about this. So we're hoping to bring a care that is less costly to the patients, that, you know, has less out-of-pocket cost and leads to better outcomes.
I know that Dr. Dronka has a meeting and has to go. I just wanted to tell Dr. Dronka, before you go, make sure you take a picture at the iCARE booth outside. Beams, if you don't mind. We want all our iCARE PIs to take a picture outside, and if your community PI is around, you can take the picture as well. So while you're walking out, you will be followed by Beam so that she can take a picture of you outside. Well, thank you,
Thank
Dr.
you,
Dronka.
will do. Thank you very much.
With more question.
One more question.
My question is on health fair. In Jacksonville here, we have a large Nigerian community. We have the Nigerian community, we have a Cameroonian community, we have Ivory Coast, we have Ghana, you know. And I was wondering whether I can coordinate a health fair. We'll provide the venue. Normally, every last Sunday of the month, we have Nigerian chaplaincy mass in a church after Memorial Hospital. So we can use that venue that has a large park and see whether the bus can come out and people can come in and do their tests, all the tests. And from there, we'll be able to identify what everybody has. So I will be ready to work with Mayor as regards to that. Okay.
Yes.
Okay. So I will see you.
Yeah, so I'll connect with you and connect you
Okay.
with the team.
Thank you so much. Let's put our hands together for Dr. Dronka. So right now we do have Dr. Dedino, Dr. Kim Babel Johnson, and Dr. Kaninji. Please, if you have questions. Well, already took questions. We think you have more. Okay. He wants to see Dr. Dronka. Yes, any question for Dr. Dedino on the Vera? Okay, any question for anybody? All the iCARE PIs. Dr. Zhu, do you have your hand raised? Okay.
I do have a question about the viral. So this is very innovative technology. I think adoption of a new technology will depend on many factors in addition to the expense purchasing the device. Maybe it depends on the age, the attitude to the novel technology, acceptance attitude especially. So I wonder, can you share some opinions or thoughts on how to ensure the adoption is decreasing the disparities rather than widen the disparities? Thank you.
Great question, Dr. Su. One of the things to note, and I think that is the whole idea of the comprehensiveness of the way that we do the work. The work starts from the community. So first of all, we begin from the community, having focus groups, having in-depth interviews. So the VERA concept transition, and I'm glad that Dr. Young is here, from 2013. We just didn't wake up and say, here is VERA. So wanted to recognize Dr. Mary Ellen Young, who a grant in 2013, funded by Department of Defense, and we were MPIs, started with what does survivorship looks like? And this was a study that we started with over 10,000 Black men diagnosed with prostate cancer in Florida. I think it was about 12,000 plus. Once we got that, we started with a quantitative description of the men. And then we identified from those men, 10,000 plus, across not only across the different stages and demographics, across the whole of Florida. We were very deliberate. And we selected African American, we selected African, and we selected Caribbean men. And from that, we did an in-depth interviews, and the in-depth interviews was probably 4 to 6 hours. That was done by, a very powerful clinical coordinator. Another reason why we're so passionate about this, who we also lost to cancer, Mr. Stokes. So that gave us a lot of information about what we need, and Dr. Mary Ellen Young developed a grounded model of what is needed from prevention all the way to survivorship, all the way to advocacy. And based on that, we then went next to develop a strategy statement. So you can see that this is sort of like grounded in what the needs of the men were. So even after we did that, there was another companion study, also Dr. Mary Ellen Young, Dr. Kim Walsh-Childers, another grant that focused on scientific discoveries. How do you communicate scientific discoveries, and what are the models for you to be able to communicate appropriate things, and what are the determinants? So this study again looked at the determinants of how do we communicate health information and scientific discovery to Black men. We also built another model of that. So once we built that, we knew, I don't think there's any group like us that has systematically and comprehensively studied Black men in the United States. And when I mean Black men, all Black men, regardless of your country. And based on that, we came up with a model. We not only developed the grounded theory, you will see the model that I put out there that is very specific to each point. Once we did that, now we started having the dialogue. That's when Mr. Merriweather joined our research team, and we had other survivors, and we were having dialogues on the use of technology, and that's where the use of technology came in. And I can tell you, Mr. Merriweather was here, he was gung-ho against it. Mr. Merriweather was like, I can't even do QR code. What do you mean by 3D, whatever you mean? But we continue to, one of the things that you have to understand, yes, we know the desires of men, but we have to continue talking to the community. And I want to give it up to Commander Motri right there, because another thing that was done which was led by Commander Motri and Dr. Floyd Willis, there was a community town hall that was done at the American Legion Post. So I want you to kind of understand that this is so systematic and it takes a whole lot of effort. So we're talking about decades now. And I have never seen a community person really do a research-focused town hall as Commander Motri did it. It was so outstanding. I was there at the American Legion Post. And they gathered data for the people who are there, and we classified the data, and after gathering it, they separated the top data, and then we went into all groups and we ranked other 8. And let me tell you, we were thinking number 1, and Commander Motri, if I'm wrong, please correct me. We were thinking number 1 would be insurance, screening, bring this here. Number 1 was technology. It was like, what do you want to go rank that? No, it was technology. But I think having people now have the knowledge of post-COVID that we cannot just shut our eyes or bury our head in the sand and think, yeah, we don't want to do technology, let everybody else know, whether we want to do it or we don't want to do it. So once that data came from the American Legion Post 197, we knew we were onto something. That the people were saying, technology is number 1. We want to have access and we want to be trained on technology, and it was part of that. So once we knew that, we knew the VIA, like we don't have any choice. And our first idea, and we wrote a grant about it, that I co-wrote with Dr. Mary Ellen Young, which we didn't get. Thank God we didn't get that grant, because our first idea was really to put survivors in clinic, right? And Mr. Merida was on that grant. And we put survivors in the clinic, and when the men come, so we would know when the men are going to come, and we will know, the physician would have already told us when they're breaking the news, and we will have a survivor there, and we will ask you, do you want to talk to a survivor? That was the grant. We didn't get that grant. I mean, in hindsight, thank God we didn't get that grant, because I don't think it would have worked as perfectly well as the VIRA did. So long way of saying everything kind of came together. But another thing that I think I was very happy Department of Defense grant said was that once Department of Defense saw that we are infusing technology into our intervention, the Department of Defense then said that you have to address the issue of technology access as well as literacy. And that's when we did the tech hub, and we were able to do the tech hub and do a training program as well as make technology. So it's very systematic. And then I gave that long story because you can't do this work by simply not putting your ears. You have to engage the community, survivors, advocates at every step. And that's kind of one of the things that we were, systematically, it wasn't only the prostate cancer survivors, it was having the town hall led by Commander Motri. And it was the community saying, technology is number 1. Give us access, give us training, give us this. So yeah, and we continue to do that. Even after we did it, the alpha testing was done by survivors and clinicians, and the beta testing was done by survivors and clinicians too. But then we are moving into the point of the clinical trials, and the clinical trials is also going to have an implementation research aspect of it, where we're going to go back and look at the experiences. But I'm going to tell you, one of the things that made us know that this is a great thing is that Dr. Christopher Williams keeps on telling you, when is this study going to be end? Because people are asking for it. His patients are asking for it. Is it already out? Have they ended? And we're in this sort of strange place that we have to do clinical trials. We can give it to you, but people are saying we want it right now. And in fact, a couple of people say, can I buy it? Where are you selling it? So we knew that we were on the right track, but you have to put your ear to the ground. You have to keep talking. That was a long-winded way, but that.
Thank you very much. And we actually have Dr. Kimbabel Johnson here, who is the director of the Mayo Clinic MC Health Community Against Racism program, who funded the COTEC Hub. So Mayo Clinic is funding community health hubs where community members have opportunity to train on how to use technology for their health, how to use patient portal, etc. So Mayo Clinic is really supporting community technology in Florida. Do we have any other question? Any question for Dr. Any additional question for Dr. Kaninji? Okay, thank you. Yeah, question. Do we have question on vitamin D study? Okay.
For the vitamin D study, Dr. Bella Johnson, could you share with us, because, you know, a lot of times we really do not have an appreciation of what it takes to launch clinical trials in LMICs. Could you share with us some of the challenges that you've had to overcome and what it takes actually for global collaboration?
Thank you. It takes a village, literally, to overcome these things. But I want to just talk about why beginning is such a great step. To get to where we are with the global collaboration, it's really taken the community, it's taken the insight of those like Dr. Detna, who spent 3 decades in this space, creating trust for people to fund this kind of work and people to participate in this kind of work. And even with that, it still was a heavy lift. So Dr. Colon-Otero, prior to my return to Mayo, and Dr. Detna's coming to Mayo, who was really on board with those like Sandra Casanova, raise your hand, Sandra, doing this vitamin D work and doing it successfully. And then we actually had one of Dr. Detna's lab members help us to expand the study to be decentralized. That success put us on the map to where we were able to take this vitamin D work and thought, what if we were to collaborate and go to the source of where this pan-ancestry information existed? And with seasoned scientists in Africa who were trusted by Dr. Detna and thus by Mayo Clinic and the funding of the DoD, we were able to at least write this grant. No problem, right? We got money, let's go. Well, not so simple. So even while we had an open trial, it really took us back to understand really what the regulatory space was, and you'll hear some about that in our overview this afternoon. But understanding not only how to partner, like to make sure that the things that we are doing to ensure our science here is paralleled, to understand who the FDA equivalents are in that country, to understand the difference between socialized medicine and others, to understand all of the layers of not only research, but practice of community, to the point that we were considering things that were kind of bread and butter, like if we did a vitamin D study, where's the vitamin D going to come from? Is it going to come from the United States? Is it going to come from Nigeria? What happens when the study's over? Do we just like let them, find their vitamin D source on their own? What does it look like when you're partnering with very busy leaders in Nigeria, in the OR, when we want to have meetings at Mayo Clinic in the middle of the afternoon? So there were so many layers of this partnership that go unrecognized, but it's really because the heart of people are in the study. And while it took us a while to get going, and I want to just report that we are just about to start enrolling our first patients in Africa, it's taken us much longer because we wanted to do it correctly. We wanted to do it correctly, not only for the study integrity, but for all the partners who are involved, to make sure that if I'm getting paid, they're getting paid. If our patients are getting paid, their patients are getting paid. We needed to make sure that all of those things were very diligently done with high moral and high ethical value considered. And so, yes, there is a science of vitamin D. Yes, there is a science of immune markers. We need that. We don't need that. We, like the world, needs that, right? Because we're talking about something that really is unexplained and unknown, really, in terms of, does vitamin D help for prevention? Does vitamin D help survivorship? I mean, this is a very complicated area. So we definitely are contributing to the science. But whether it was Dr. Colon and Dr. Pupulu's trial or some other trial that Dr. Detna had chose to identify in this space, what we're really learning is how small the world is, and how, while all the socio-political issues that exist in the world may be compelling to talk about on the news, it's really important that as scientists and as patients and as advocates, that we just recognize just how small this world is and why we must be aligned in the best interest of the patient, like we say at Mayo Clinic, and that patient is every patient who might be impacted, particularly those who have an increased burden of disease such as prostate cancer. So Dr. Detna, I know that I tend to go down the road in ways that you didn't expect me, but I hope that I answered that specific question. Mhm.
I think as you're talking about the science of it, it just hit me. I know you're working on the paper, that I think you need to work on the science of transatlantic collaborations for clinical trials. We need a paper like that. Where's Dr. Kanije? You're the publication team. I think it's so important because the last year and a half has been painful. We don't have to sugarcoat it. But I think there are lessons learned that I think if we publish those lessons learned, I think they will be great. And I think just tackling it, the science of transatlantic collaboration to facilitate clinical trials will be a great one.
I take on that challenge with Dr. Kninjan, but I totally agree. The process is important. It's like, knowing your grandmother's recipe, but you know it in your head, and then you try to create the same thing. It doesn't taste the same in the kitchen. I think that there is science, and Dr. Detna created a Science of Community Outreach and Engagement Conference that's now in its 4th year, and it's because we didn't even know how to do this together in our own country. And I think really the lessons that we learned in the clinical trial space, which again is different than practice. Clinical research, clinical trials is different than practice, and for good reason, because we have a lot of vulnerable things that we need to protect. And so having 3 decades of experience, what will that look like 3 decades from now, right? So we need to actually make sure that the science is there, but you're right that it's published, and we need to take the time to do that while the world is not stopping. So thank you for that recommendation. Thank you, Dr. For me? Yeah.
My question is not a question. It's recognition to something that Dr. Odetna said about the quality of life through the point of prostate cancer diagnosis and intervention. I would probably go a little further when it comes down to grassroots. The support group is one of the main parts of the iCCaRE program that was developed through our leadership with Mr. Gainey and myself and other members of the American Legion Post. We started that support group, and one of the things me and Mr. Gainey found out as we host these support groups monthly, we find men that has newly got diagnosed coming by. And one of the wonderful things about this program is that it allowed us to interact with these gentlemen that comes to the post, 197. They'll come, hey, I just got diagnosed, Mr. Merriweather. I don't know what to do. And the exciting part is we've been trained. We got the information, and we have other survivors in there. And I'm trying to reach these gentlemen's advocacy. That means y'all are survivors. When you have these support groups, you're able to catch these young men that newly got diagnosed that don't know where to go, don't know what to do. And the wonderful thing about having prostate cancer is you live. You are alive. That's the wonderful thing. The bad thing is, am I going to die? The good thing is having young men that have survived to encourage other men why it's so important to get diagnosed. Why it's so important to know your family history. Why is it so important to know the side effects? And I see the tool in the toolbox is the support group. That's just one tool. We are not trying to be doctors. We're not clinicians and all this other stuff. We are just plain old survivors telling a story. And when we tell the story, we're telling iCCaRE. We're telling collab. One of the good things about having leadership at American Legion Post, Commander Moultrie, but the most important person I like at the Post is Quincy. I'm gonna just tell you straight up. Whether y'all agree or not, Quincy brings the, he brings the material. Well, I'm going with it. He brings the material. When we can't speak technically on What is this and that? Quincy brings a different role. And the reason I use that's his baby. That's his area. He goes to these posters. And I'm sharing with y'all in a sense that we have to use all of the tools. The gentleman in the back, he shared with us about the Nigerian community and the health fair. I apologize, I never heard of it. First thing I said was, y'all here? And I said, well, I reached over to Quincy and I said, Quincy, we gotta talk to him. When he have these health fairs, we need to know. So we can bring Mayo, we can bring the Post, we can bring the leadership, we can bring the material. In closing, I want to give you an example. Mr. Gainey and Miss Gainey developed this card. All posters should have copied this, which I don't know if the card says prostate health support group. It's got the address, the date, time, and then Mr. Gainey developed the information on the back. What I told Ms. Beams earlier is we need to take this same card, these 5 categories about the percentage, and put the products that we have on this card and put a barcode where somebody can find information about. This is what leadership is with Mr. Gainey. He had an idea. I took his idea. I apologize for stealing it. But I took his idea and I gave it to Ms. Bim and I said, this is what we need to do. The whole concept of this session and this group is to network, to share ideas, don't care who got the ideas. One of the things that is important in closing is that, how many of y'all women got a prostate? None. But y'all know someone who had prostate. And it's so important that ladies encourage your loved ones to get involved. The support group, I got another friend in here, I hope he don't mind, Quentin Jones, my church brother. He told me last night, he said, Meriwether, thank you for introducing me to this program. I learned so much in the advocacy training so that I can go out and tell somebody. That's what it's all about. Thank you very much.
Thank you, Mr. Meriwether. One last word from Dr. Kim Babel Johnson.
It's actually a comment because I'm so happy that Mr. Meriwether didn't have a question for me because I usually don't have an answer for Mr. Meriwether. but he did actually speak about community and just how you just never know. And I don't know if Daniel Lee is still in the room, but one of the most impactful stories that, yeah, is he? Well, probably. He is my biostatistician for Dr. Willis and my trial in mindfulness and cancer survivors, and we are so blessed to have him. But his passion in this space, actually, one of the most impactful stories of, that I can tell is that Daniel actually was doing these talks and, bring your congregation. And the minister in one of the congregations was there listening with all of the people who were at risk and actually had not been screened. That minister ended up being screened and actually being diagnosed. So do you remember that story? That is a story. I didn't make that up. I wish he was here to just give me the details, but I do think that it's extremely important that we recognize, the importance of bringing your brother's keeper. Well, one last thing. Dr. Detna talked about yesterday, and she dropped it like, a hot potato, younger men being part of this story. So my father had prostate cancer. My son's grandfather is a survivor of prostate cancer. And we are moving in the space, hence vitamin D trials for immune markers and very specific personalization of medicine. So while we have each other's story, this story, the train is already out of the train station, that we're personalizing these things. And so what we need to do is figure out, if you have cancer, how does that impact your son? How does that impact your nephew? And so some of that study, like, was he BRCA positive? Some of those things we know now, but a lot of things we still need to find out. So Dr. Detna and others are leading this space because I'm not a prostate cancer scientist, but I raised a Black boy. I'm raising a Black man who's 27 years old on tomorrow. So I need to understand, when does he need to screen? What does he need to do? Because his risk could be greater than just the average Black man's risk. So let's continue to understand that this is a train that continues to move and to get on board with the train. So, thank you.
Thank you so much. And thank you, everyone.
I think that's it for this particular session. We're going to roll right into, we have some pre-recorded sessions for everyone. So we have one from Dr. Kimlin Ashing and also from Pastor McCall out of City of Hope, out of Los Angeles, California.
Good day. I'm Kimlin Ashing, PI on the Project 3, creating a digitized survivorship care plan for Black men. So we're joined by Dr. Odedna as MPI, Pastor McCall, who's been instrumental in creating the document, as well as reaching prostate cancer survivors for the pilot, and also Dr. Moore, Dr. Ngufu, who provided expert guidance on the digitization of the document, and Dr. Bedell, who's an early stage investigator at City of Hope. So the objective was to create a prostate cancer survivorship care digital toolkit to address the unique needs of Black men and to improve their quality of life. So to achieve specific aim 1, we employed a mixed method approach to develop the toolkit, and using clinicians for the key informant interviews, partnered with PASS on this project, and they were really instrumental in providing interviews to inform the development of the toolkit. We also conducted surveys with prostate cancer survivors, and Pastor McCall was also instrumental in reaching survivors within the community. So the findings will reveal factors related to the negative impact on prostate cancer, on quality of life, including comorbidity, adverse treatment side effects, decision regret, connecting with other survivors, emotional and sexual concerns, unhealthy lifestyle, and the lack of survivorship care, planning, and symptom management and guidance from their clinical care teams. For specific aim 2, based on the data that we collected from specific aim 1 with the qualitative interviews with the key informants as well as survivors, we worked to develop the survivorship care plan, and also conducted a pilot of the survivorship care toolkit, and we also conducted the feasibility testing. So again, some of the themes from the qualitative interviews, 18 themes were identified from the 22 survivors who participated. These included distress, anxiety, isolation, erectile dysfunction, life stress and comorbidity, high proportion of co-occurring illnesses, treatment side effects were identified, inadequate quality of care, the need for advocacy, response included anger, frustration, lack of exercise, family and strain that occurred due to the prostate cancer and its treatments. Nutrition information was also identified as an unmet need, and survivors identified a need for social support and self-advocacy. So this slide represents some of the components as we started building the prototypical digital survivorship care toolkit. So it included infographics, described what the survivorship care plan is, had information that built out of the ASCO template that provided information about medical treatments. So the study methods to create the toolkit, you really gathered the information from the AIM 1 and all the qualitative interviews, and there was clearly a need for a treatment summary, follow-up care guidelines, documenting the comorbidities that's very common in the African American community, and documenting the treatments that were being taken for those co-occurring chronic illnesses, to really facilitate integrated coordinated care with primary care clinicians, but also to identify any potential treatment interaction. So very important component that's not been included in any other survivorship care plan or survivorship care toolkits or templates. So we identified from the qualitative, the list of survivorship concerns that they can rate, and then were encouraged to discuss these with their care team. We also included health advisories and resources, included a societal determinants of health section that included resources for the survivors to use. We also included a survivorship booklet that contained information about diagnosis through treatment and survivorship. And the toolkit also included survivor videos that included many of the advocates within the iCCaRE Consortium. The feasibility testing of the digitized survivorship care toolkit intervention included 1 to 2 virtual sessions of motivational interviewing, and we explored acceptance and utility of the toolkit with all of the resources, as well as the motivational interviewing session. And to assess the outcomes, we use self-report measure of health-related quality of life using the FACT-G and the European Quality of Life measure, as well as societal or social determinants of health, the stress scale. Overall, 139 prostate cancer survivors were enrolled. Most participants were diagnosed at stage 1. We did have a range of stage 1 to 4. Radiation therapy was the most common, with almost 50%. About 39% had surgery, and 54% reported treatment-related side effects, and 56% reported co-occurring chronic illnesses. So only 13, almost 14% endorsed that they received genetic or genomic counseling or testing. So most indicated that they actually did not. And so especially with men who are younger age, maybe under 55, it would have been important for them to receive, genetic counseling to really guide the treatment. What are the results for the European Quality of Life scale? So this was collected at baseline, and we'll report on the baseline outcomes. We see that, mobility, self-care, usual activities, pain and discomfort, as well as depression and anxiety were identified. We see that the visual analog scale that looks at quality of life, that the score range to 70, almost 72, which is a fairly reasonable or good quality of life measure. Post-intervention, we had 80 survivors who completed the intervention in the post-survey, and the visual analog scale increased to almost 75, showing a trend towards improvement in self-care, as well as mobility, and improvements in addressing pain and discomfort, and reduction in anxiety, depression, and burden in engaging in usual activities. So overall, the sum score rose from 7.2 to 7.56. So approaching clinical significance, And with a larger sample size, we may see a greater effect of the intervention. The results of the FACT showed that the scores on the functional assessment of cancer therapy, we had 81 completing at post intervention, and the 4 domains of quality of life were well assessed and within the range of the scores. So for the FACT-G scores, we showed that there was a trend towards improvement on all of the domains. The mean change for physical well-being was 0.67, followed by emotional well-being of 0.4 and social well-being of almost 0.4 and functional well-being at 3.5. The total FACT-G change score was 1.78, which approached clinical significance of 2 points, but didn't reach the documented 5-point change to show statistical significance in change and improvements. So we wanted to dig a bit deeper and looked at those who were in the lowest 25 percentile, so the lowest quartile of quality of life. And so we explored this by looking at those who scored less than 71 at the baseline. And then, so we identify a total of 15 survivors who fit this category. And we saw that the total change was 5.73, which is statistically and clinically significant. These survivors show improvement in social well-being with an average score of 2, followed by physical well-being 1.6, functional well-being 1.2, and emotional well-being almost a point improvement, showing that those with the greatest burden stands to benefit the most from the intervention. I want to take the survivors who complete.
During the break, we will continue to run Dr. Ashing's presentation while we take a break and while we eat. But we're going to push on to our next session, and that is going to be on the explanatory model of prostate cancer experiences of Black men. So we have Dr. Iwan Coburn, we have Dr. Bolajoko, and we have Mr. Badamose.
Good afternoon, everyone. I thought that Dr. Balajoko was going to present with me. We are co-PIs, and so I just wanted my co-PI up here. But I think for the most part, we are the co-PIs of PP6, and we are excited to talk to you this afternoon. And so don't want to stand between you and lunch. So PP6 is diverse perspective, unified support, the prostate cancer survivorship model for Black men prostate cancer survivors. Dr. Balajoka and I, we are co-PIs. Oops. Uh-oh. Nope, that's not it. How do we get to the next? Oh, so we're going, no, it won't go down. The green. Mm-mm. Okay, ready? No, you said 1 minute. All right. But what we're going to present this afternoon is the pilot of a pilot. So we have a pilot study for PP6. We're recruiting 50 men across the enterprise, so Jacksonville, Rochester, and Arizona. And what we've decided to do is to do an interim analysis of 10 men who have started to be a part of the study. So we are using REDCap, ideally PP6. We're going to use the iCCaRE Vera for platforming. That's the platform that men are going to use for their recording their experiences. And so while iCCaRE Vera is getting ready for PP6, we decided that we didn't want to wait until the Vera was completed. We want to get some preliminary data to get started. And so we've recruited 10 men, and then we asked them to journal for 4 weeks. And so what we're doing this afternoon is just presenting some highlights from the 10 men based on their survivorship experience. Okay, here we go. All right, we're ready to go. So PP6 has a very dynamic, diverse, and just outstanding team. Our community partner or community science is Mr. Charles Walden, seated over here. Dr. Ballage, yeah, very important. Could not do the study without our community partner. So he is our outstanding community scientist. Dr. Balajoka and myself, we are the study PIs. Dr. Osedu in Rochester. So once again, this is a multi-location study. Dr. Osedu is in Rochester. Mary Ellen Young is an excellent community and qualitative researcher. So is Dr. Osedu. Dr. Katie Redden, these are our clinician partners, and Dr. Childs. Umar is the postdoc and is working with Dr. Ingolfu on the avatars and the platform. Jocelyn Turner, she's just an outstanding public health community scientist, or I would just say public health researcher and scientist community partner. She helps us to recruit participants across the 3 locations. And then the study coordinator is Amanda in Arizona. I have to really give a shout out to the men who are part of PP6 because several of them was trained yesterday to be prostate cancer advocates. So these men are here to share their story, and they are really engaged in the journaling for the last 4 weeks. And so several of them was trained yesterday to be community advocates. So they're going to be going out to the community to share their stories. And so we're excited about that. Okay, so I'm just going to give you a brief background, don't want to bore you, nothing too much. So what we do know is that after prostate cancer treatment for Black men, the experience is very high, variable. So some men report feeling discarded, don't feel integrated into community, while others feel optimistic. And so that's a very variable experience. The objective of the study is to capture the experience of an ethnically diverse prostate cancer survivorship relative of men, so prostate cancer survivors relative to survivorship cure adoption. The novelty about this study is that we're looking at Black men and Afro-Latino men, so Afro-Caribbean or Afro-Latino men. And so that's the novelty here in terms of the population. In terms of the study design, it's a qualitative method. We're using grounded in theory. The study population is diverse, African-American, Afro-Latino men between the age of 40 to 75. We're doing research in both English and Spanish. We have 2 aims that are driving the model development. Aim 1 is model development. So it's a 30-day of journaling. So 30 days, 4 weeks using the iCCaRE platform. And then model 2, we're developing a model of validation. So these are in-depth interviews, Zoom interviews. So what we've done so far is our bridge analysis, and that's what we're going to be talking about today. So we did automated surveys using REDCap, and so men did structural journaling for 4 weeks. And so 10 men completed this
journaling, and so we're going to be presenting here. So here are some of the preliminary themes that we found. Once again, this is not the full analysis, and so it's just for 10, but we felt that we had some exciting things to present for the conference. So men discussed psychological reorientation. So after treatment, they had to kind of think about life a little bit differently. They talked about medical literacy and follow-up adherence, so follow-up to treatment, follow-up care, and also literacy. The men also talked about continuous versus transformation of healthcare routine, and so we're gonna give you a couple of quotes here, and then dietary awareness, and then informal what Mr. Merriweather talked about in terms of support groups. We're seeing that highlighted here, just in the early initial responses. So we're gonna, in terms of the support, men didn't necessarily want to have formal counseling. They wanted peer groups. They wanted those kinds of things. So we're gonna talk about those real quickly. So in terms of psychological reorientation, participant number 3 mentioned, I didn't realize how precious life was until cancer come along. Big thing. I felt participant number 1, I felt a strong sense of normalization and completion after his treatment. So these are prostate cancer survivors. In terms of medical literacy and follow-up adherence, participant number 1 said, I view the follow-up as a vital part of the treatment life cycle. So this participant is talking about not just treatment, he's talking about the life cycle, not just diagnose me, treat me. He's talking about the life cycle of his survivorship. Participant number 2 said, my desire was to maintain close follow-up to stay on top of any treatment and prognostic recommendation. So these men, this is going in contrary to the notion that people will say Black men don't go to the doctor, right? These are survivors and they're talking about their experience, how important it is for them. Continuously versus transformation of healthcare routine. Participant number 1, my primary focus was returning to my existing healthy habits rather than overhauling my routine. So they want to go back to healthy habits post-treatment. Number 3 said, I got to start eating better. So this is somebody who's reflecting on the way that they eat. I have to exercise a little bit more and spend more time with my loved ones. So he's looking at how important life is now, what he can do to transform and do better and to spend and prioritize his loved ones. Dietary awareness, I'll just read one comment. I see that we have 3 minutes, and so we just want to keep going. Increasing vegetables and fish intake, reducing soda consumption, and we've got some soda over there, but Lord have mercy, prioritizing water consumption. Informal, here's this participant number 3 said, people still don't understand what he's going through. And so these men are, they're survivorship, they're in survivorship, and they're saying that some people still don't understand what they've been through. By educating the man in my life about prostate cancer, I was able to find a sense of purpose and completion. And so this is where in the support group that Mr. Merriweather talked about is important. Men want to be a part of the process, and so want to highlight there. So in terms of next steps, so just briefly, we are in the process of developing the VIRA. We've been meeting bi-weekly with Dr. Ngofu. And so what we're trying to do is not to reinvent the wheel, but to integrate this PP6 with the existing VIRA for iCCaRE. And so we've been working very collaborative with Dr. Ngofu. It's coming along, and we would not have these outcomes for the 10 men and VIRA development if we have not been working collaboratively. And so and transparent. I think that's the big thing. So then we want to do full aim 1. So we want to recruit 30 diverse Black prostate cancer survivors across Florida, Arizona, and the Midwest. And then we want to do the 30-day journaling via the VIRA. Model 2 is where we're going to recruit, we're going to do the model validation. And so we want to recruit 30 Black men for in-depth interviews to validate the models that we're working to develop. And so those are the outcomes. Any questions? Should I stop here but yeah, all right. And we have a minute to spare, so.
Okay. We're actually gonna pause right here and take a break for lunch. And what we're gonna do is break for lunch for 45 minutes. So try to come back by 1:15, 1:20-ish so we can get started. So Mr. Badamose can get his presentation, and then we'll take questions for the both of you all after his session is complete. So write your questions down for Dr. Coburn, and we'll get them done after Mr. Badamose speaks after lunch.
Well, good afternoon, everybody, and apologies for interrupting your lunch. I hope nobody slips off on me. Yeah. I think I'll begin this by acknowledging the other team members whose pictures and names are not represented on this slide. The national PI is Professor Abidemi Omonisi, and the PM is Dr. Uche Unwoku, and we have for the 5 registries we're working with. For CAN registry, it's I think it's Professor Aminu Zachary, and for Calabar registry, it's Professor Ekanem, that's Imabong Ekanem, and Dr. Festus Igbinoba for Enugu registry, and Dr. Izume for Abuja registry. So this is a retrospective validation of AI for early prostate cancer diagnosis. This will explore rationale and pathway. We have just secured the 4 IRBs, so we're waiting for one, and so we don't have a lot of preliminary data, but you'll understand exactly what we're hoping to achieve. And so understanding the problem, I'm singing to the choir here, so I'm not going to belabor the rationale for why prostate cancer is a scourge and why addressing it as early as possible is the way to go. We built our models, different versions of it, but the latest version of it has been built with data sets from the Mayo Clinic platform. And what you see here is a representation of the data sets on which the models were built. For a model to be generalizable, we've got to make sure that it would function the same way across ethnic groups. And it will function the same way regardless of the geographic location of the users or patients, as the case may be. So for training data demographics that you see on your screen, this has been built with representative data sets from Caucasian males, male of African ancestry and Asian ancestry, but you would see the representation is significantly low for the other ethnic groups. And so it means that we cannot deploy a model like this. And we believe we cannot deploy a model like this in a totally different geography without localizing. And so, whereas if you look at Nigerian patient demographics, and now this is purely physiological, because as Professor Rotimi has clearly told me, Nigeria is not homogeneous by any stretch of imagination when it comes to genomic variations. But from a physiological point of view, you're talking about a homogeneous group largely. And so the question is to say, if we're going to deploy AI model for early prediction of cancer in Nigeria, model that's been built with data sets from Western Europe, largely from the US, would the model generalize effectively? Will the current AUCs or in terms of predicted positive value or positive predictive value be the same? And if it's not the same, what will be the variation in there? And if we understand what the variation is, can we identify what's responsible for it before optimizing the model to work in that market? So here's a study design. This is a retrospective case control study. Now, this study is designed because it's retrospective, our partners are cancer registries, and they're hospital-based registries. There are 2 types of registries. We've got a population and a hospital-based. And the reason for wanting a hospital-based registry is because we have opportunity for longitudinal data. So because a registry is a static repository, and it doesn't tell you the story. It just identifies the patient's current state at the point of abstracting the data. Now, But that doesn't help our models, because we've got to be able to see what led the patient, want to see the information that resulted in the patient being, before the patient was actually, before the details were imputed into the cancer registry. For example, the Gleason score, where biopsy was done, or the trajectory in terms of the PSA data point. So that's why our data source is CanReg5, which is used by cancer registries, and also hospital-based EMRs. And we're looking retrospectively from 2020 to 2024. For some of the cases, we're realizing we can get much longer than that, and which is fine. A sample size about 5,000 subjects, which is for the cases, and we're looking to get about the same number of cases for benign controls. Our gold standard in this case is the histopathology result, which is a biopsy result, like I mentioned, the Gleason score. Of course, from a data architecture and privacy point of view, we're ensuring that the process we undergo in this study is in line with what FDA would approve, and also in line with what GDPR would approve. So from a security point of view, we're ensuring that the patient data does not leave the site, and that we don't have a line of sight to the patient's medical records number, and there's no way you can unscramble that. So the case definition, what's our inclusion and exclusion criteria? For the inclusion criteria, we're looking at confirmed cases of adenocarcinoma, and also complete Gleason score availability. And where possible, we want to be able to see the pre-diagnosis PSA. And that's the reason, like I mentioned earlier on, why the register of interest to us is a hospital-based registry. Exclusion criteria is the autopsy-only diagnosis, or in this case, previous hormone therapy. Now, from a control definition, what's important here is we understand that there are cases of false positives, a lot of cases of false positives that we know of. And so from a model's point of view, we want to understand what parameter led the physician to think this was positive, and see whether that overlaps with actual true positive cases, and then understand the subtle distinction or variation that might be there. If we understand the subtle variation from a model, we can amplify that variation. So in this case, we're saying men with urinary symptoms, but negative biopsy. Effectively, it means men who were thought to be positive, but then after biopsy, turned out they were negative. Why? Just like I mentioned, to prove that the AI can distinguish between the benign prostate hyperplasia and actual true positive cases. From a team structure point of view, like I said, we've adopted a process or a system that allows us to be able to implement a study protocol that can travel, which means, when I mean travel, it means it will pass GDPR and FDA protocol and process as well. So this is what our team structure looks like. I've given the names of some of our participants, but this is exactly what we have. For each registry site, we have a clinical lead, which is a pathologist or urologist, and we have the hospital IT lead, and also the registrars for the data abstraction. And also we then have a lead engineer, lead data engineer, site coordinators for all the 5 sites. We have ML scientists and our cloud engineers as well. And of course, you have the principal PI and also the global PI. And like I mentioned, all this is underscored by the ethics and compliance resource. I'm just going to skim through this. This is just defining exactly what each role is, ensuring that we have DUAs before we proceed, and that there is total compliance. what we have done is for this study, we have a platform that we built specifically for this study and a public-facing website. And the reason is there's challenges we anticipate. Like I said, we are only about to begin now because we just have the Arabs. The challenges we anticipate, one of the challenges about the systems available in the registries. We're not sure of what sort of data sources exist. We know that for some of the cancer registries, some use CanReg 5, some use CanReg 4, some use Excel sheets. And so we don't know what they are, but we've got to be able to create a system that makes it possible, that is less technical. You know, it has to be a very simple system. So we have a platform that makes it possible for data abstractors to be able to upload patient data onto a virtual, onto a cloud infrastructure, but not ours yet. And so that we can do the pre-processing of that data and ensure that it's stripped of every PII and stripped of every non-compliant data input before getting onto our own cloud infrastructure. And even though this is ongoing, what we think is important is as we go through the process, we want to make sure that it's possible for participants and also for the iCCaRE leadership to be able to follow exactly what the progress is. So where we are so far, like I said, we received 3 out of 5, but as at this morning, I understand we're now at 4. So we have 4 Arabic clearance right now. Our infrastructure audits, the first thing we want to do is be able to audit the registry infrastructure and the hospital infrastructure as well. We have training planned already where we will train the data abstractors and train the IT team as well. And one of the things we hope to achieve with this and why we think the training is important is we just don't want to do a study and get away from it. We're hoping that we do this study and build capacity in that process. And that's the rationale for building the infrastructure so that multiple studies that occur will just benefit from having the infrastructure in place. And I'm hoping we'll be able to do more studies with Dr. Ngoefa and myself and be able to leverage the infrastructure we're putting together in place now. And we're hoping to be able to get into data collection and then the exciting stuff of data analysis. So that's what our implementation schedule looks like. We have an infrastructure and an onboarding process. We have data extraction and harmonization of data sets. We want to be able to see where there are variances, then we go into model optimization. We want to validate the new models that might be optimized as a result of the findings and then stress test them. And we'll be excited to share the results with everyone. I'm curious to see whether, because this study has been done across 5 geopolitical zones, I'm curious to see whether we would experience variations in the model performance across different geopolitical zones. Thank you very much.
Dr. Odedna, do you want to wait? Oh, I'm sorry. Oh, do we need a microphone? Commander Green. I got you.
Oh, I wanted to know, do you have any kind of documentation on the efficiency of AI detecting cancer? I noticed that on the film up there, you said they use AI to detect. What kind of percentage of correctness are you having from AI?
I don't have to repeat the question. Okay, I'll just repeat the question for. The question was, what's a current model performance for prostate cancer prediction? Currently, it sits at about 0.96. What that means is 96% accuracy. Sometimes that's not the only number you need to watch out for. You also need to watch out for the false positive and false negative rate.
Right.
And both are significantly below 5%.
I have a question as well. Since it seems like the premise or the basis for the initial screening in your study is lower urinary tract symptoms.
Is?
Lower urinary tract symptoms. And because most urologists don't associate that with prostate cancer, do you think that when you get your data that you'll have to somehow account for that? Because, like I said, most urologists don't think that urinary symptoms are associated with the majority of prostate cancers. We screen because they are screen eligible, or they have family histories, et cetera, et cetera. So how will you account for that? Well, or is your model going to be geared toward like non-urologists?
Yeah, and that's interesting because that's some of the challenges we're seeing with cancer. We're seeing that to CRC as well, with colorectal cancer, because the screening protocol for these types of cancer seems to be age-driven. And we're finding a lot of patients turning up with cancer much younger. So they're not even eligible to be screened to start with, which means even when the symptoms are staring the physician in the face, he will go, no, you're 26. There's no way you could be, you know? And so we're seeing a lot of people slip through that crack. Of course, changes, lifestyle changes or whatever might be responsible for that. So yes, the beauty of, and that's the beauty of implementing a sophisticated AI model, which is the training process starts being agnostic, which means I'm beginning to look for patterns, right? And I'll take an example and rush through it. If you take pancreatic cancer, the symptoms for pancreatic cancer, one of the many reasons why you have late presentation or diagnosis is largely because the symptoms overlap with benign diseases as well. Whether it is persistent abdominal pain, or you have weight loss that is unintended. But by the time you're merging that with jaundice, then it's way too late at that point in time. But with the AI model, we can pick that significantly way in advance because we find unrelated correlation that makes it possible to identify or project likely outcome when you correlate multiple unrelated symptoms together with symptoms being presented at the time of presentation and patient's clinical record in real time. Does that answer the question?
Okay. Yeah, thank you very much, Mr. Badimose. It's going to be a quick one. What we're trying to do in Nigeria, you're going back and pick only PSA from the medical EMR.
Gleason score as well.
Yeah, Gleason score, of course, that's at the PSA. Why, also trying to pick other blood work, the general blood work that people do in the lab generally. Perhaps that can help in these all related parameters that can improve what you're doing.
Yeah, we are picking that. What we've defined is what's the gold standard.
Oh, okay.
So you want to say what's the true positive parameter in question. So we've chosen that from all the analysis we've done, we're saying that using the biopsy results, the Gleason score is the most reliable gold standard, not even the PSA alone.
Just to follow up on Professor Rotimi's question, and that gold standard is for that population, or that gold standard is for the general population? Because what we're seeing that even, I think one of the good things, the beauty about consortium science and working across the globe is we are seeing a lot of differences, whether we are doing the diagnosis of prostate cancer here, in the Caribbean, in Africa or even in UK. So I think, I'm not sure whether the African populations is very well studied to actually know the gold standard within them. So I'm praying that part of what you're doing is for the AI to also guide in reaching this gold standard as well.
Yeah, that's interesting. And that's we thought initially that the PSA would be a gold standard. But then we're finding variations in how the PSA works with men of African ancestries versus other races. But what we've found that is credible is a biopsy result. Of course, that's interventionist, right? But that's why the biopsy result, which is the Gleason score, is the gold standard. We're finding that to be, regardless of whether it's Caucasian or Asian or African American or Africans in Africa or Afro-Caribbeans, the biopsy result is the biopsy result at that point in time.
Dr. Coburn? Okay. 4, or he wants to ask a question? We have one more question by Dr. Coburn. We have a question for Dr. Coburn? Oh, okay. I'm sorry. I forgot. Do we have any questions for Dr. Coburn? You have another question for Ms. Buck?
Since you said you're going to take into account all potential variables in it, why not also, where available, include digital rectal exam?
Say it again?
Why not, where available, include DRE, digital rectal exam, in your study?
For prostate?
Yes.
That's interesting. That's interesting. For the population group, that's interesting. We see a lot more for CRC. For prostate, it'll be interesting. That's an interesting input. Yeah.
Because it's a low cost intervention.
Yeah, let me jump in. So in our outreaches that we're doing in Nigeria, what we have seen with DRE report from the urologist is that the way they write it is very inconsistent. And then it's just now that you mentioned it, that I remember that we had that challenge. And for the first one, we just excluded it because it was just too complex to, unlike Gleason or PSA or so. And you as urologist, perhaps, Prof, I think we need to take that back home. I'll take that. Yeah.
Dr. Williams is saying, because if there is appropriate technology with the digital rectal exam, and sometimes we are hearing that the DRE maps do better than PSA. So I think that is an opportunity, and what you're saying, Professor Rotimi, is correct. Then there needs to be training in the reporting of DRE by the urologist over there, because if we are looking at actually be able to do prediction, we should include things that are likely to help us with that prediction.
Well, the question would be,
and that's the problem. That's the problem, because the full professors don't want to do DRE. That's the problem in Africa.
I mean, I was going to say that, to say, the question will be, what's available in the data set? Would we find, I mean, if we find that would be interesting. Yeah. But thanks for that.
Now, do we have any questions in the room for Dr. Coburn? Who we point? Oh, you did like that.
Thank you for sharing this wonderful outline. I have a very rudimentary question just on an epi scale. I know you have a large sample size planned for your case control study. Is there any reason, with the abundance of cases that you have and how easy it is to find cases, why you're only doing 1-to-1 matching as opposed to a 1-to-2 or a 1-to-4 to increase the power of your study in the ability to detect what you're trying?
Yeah, I mean, look, what you see, the numbers you see there is less than or equal to, so in reality, we would love 1 to 2. And I just, will be great. That's the ideal scenario, Our initial number was I think was 3,000 or 4,000 cases, and control was about 10,000. But exploratory information with the local PIs proved that we may not be able to get that much. So we revised that backwards. But yes, we would love the control to be a lot more, which is why we wanted to then increase or specify the control group to also be those who were perceived to be positive and turned out not to be. That's a way to solve for that.
I think that is our last question for this particular session. Thank you. All right, I think we're going to move on to the core integration session. Does anybody want to stand up, stretch, get a little wiggle break, or do we want to keep pushing? Keep going? Okay. All right. Well, our next speaker will be Dr. Willis and Mr. Merriweather, and they will talk about Partnership Engagement Services Core.
Good afternoon, everyone.
Good afternoon.
So this is Partnership Engagement Services, and we're going to spend just a little bit of time telling you what we do. I really probably am going to do all of my talking from this one slide, and then we'll show you the other slides. We'll talk about who our partners are. And so Partnership Engagement Services really exists to partner with the community, to increase the throughput and the connection for all of our pilot projects. So you can imagine both Mr. Merriweather, who is my co-lead, and I are quite excited about that. But the biggest probably and the most important point I would make that on the next slide, you will see the team members who come to the office each day and who are online helping to organize, I think, some of our community partners in this room. But the community partners, make no mistake, are the most important part of this team. And if we didn't know it before, we absolutely know it now. So all of the fantastic projects that you've just seen and talked about probably would not move forward if the community wasn't engaged and willing to participate. So we want to help with that, but we really want our community partners, Senator Hill, the American Legion Post members, the ones, our advocates, our survivors. We really, really, really want to stress how important it is for you to tell us what's important about connecting with the community, and at some point, take over that leadership. We want to help guide the process, but we really want you to drive it. And we'll talk about some of our goals and major accomplishments. This is but a sketch of the team. But as I just said, the most important members are those whose pictures might not be up here. What are our major goals? Establish standard practices and operational procedures try to infuse ourselves in the community. Once we get into the community, work with the stakeholders and make sure that we're doing what you want us to do. Raise awareness so that people understand why it is important to get a PSA and why it is important to follow up after you've had that diagnosis so you can have the best outcome. And to try to expand our footprint, both here and throughout the world. Just an overview of the last several years, total community outreach in excess of 3,000 lives we've touched, and you can read this slide, educational awareness programs, and the collaborative and capacity building, 23 different events to do just that. But true to what we've talked about earlier, Mr. Merriweather whispered in my ear a few minutes ago that he wanted to change the entire format of this presentation. When Mr. Merriweather makes a request, you just say, okay, because 99.9% of the time, he knows what he's talking about. So he said, I'm going to be the roving reporter. I'm going to be in the audience, and I may talk or I may ask people's comments. I say, yes, sir. That's absolutely fine with me. And I'll begin to turn the floor over to him by saying, I think repeating what Dr. Odetna said earlier, when we were trying to move this along several years ago, one of the first things we did at Post 197 was have a town hall meeting. And I'm going to leave that one for Mr. Merriweather. Well, it doesn't allow you to go backwards, but that's okay. We had the, doesn't work. Oh, here we go, okay. Yeah, so at the town hall meeting, we simply went to the community, gathered them, and asked them, how should we partner with them? Is Commander Moultrie still in this room, or did she go home? She had an emergency, but anyway, she was one of the very ones. After we had spent 6 months or a year working up there, she said, we really like talking about this, but we want screening. I think you'll get more people to listen to you if they come and they talk about something, and then you actually do something that they might want you to do. Absolutely wonderful. So you just have to try to follow the lead of our community partners. So that is really what we did. We pulled the town hall meeting together. That's where some of those numbers that we just talked to you about came about. They gave a number of initiatives and things that they wanted. It was 10, 15 things, and we cut it down to 10 things, and then we cut it down to 5 things. One of those 5 things, one of the top ones, was screening. And I won't belabor that point, but we've done all 5 of those top things. And it's all an effort to make sure we're doing what the community asks us to do so we can connect appropriately. So the town hall meeting led to this particular screening. We were a little, I guess, disappointed at one point because we thought we should have had hundreds of people there. But that gentleman right there, Dr. Christopher Williams, pointed out to us that maybe there were not hundreds of people there, maybe there were 30 or 40 people there, which is correct, about 32 people. But interestingly, we got 3 PSA samples on almost half of them. That's a pretty big deal, 50% of them. And I think it all started not with us. It started with Commander Moultrie and the partnership and engagement that we began at Post 197. It goes right back to who's the most important of these team members. Now, I want to stop because I would love to hear Mr. Merriweather's comments on that and whatever else you decide you want to do, Mr. Merriweather. I know you had people you wanted to get comments from or questions from.
Hey, thank you, Dr. Willis. What we'd like to do with this portion of my couple of minutes is get the advocates, the newly formed advocate opinion. We know we got the Bob that we take a picture, and I know all of us ain't gonna take no picture and answer no questions because that requires writing. And I know you don't want to write right now. So what we'd love to know is, being that you done been here a day and a half, we'd just like to get your opinion on what you think. This is not a survey. This is more of giving you the opportunity to wake up. You know, we done ate that food and we done fell asleep. And one of the things about the PES, Partnership Engagement Services, the programs that me and Dr. Willis and Quincy and Sheena and all us have done over the years is bring programs to the north side to try to get programs that you said at the health fair, the summit, the men's health programs that you want. Would anyone mind just giving us your thoughts, a few seconds, a few minutes or so on so far? Do you feel that what we are presenting, not on the barcode, but what we are presenting, keeps you so excited that you want to come back and go tell your neighbors why they should be a part of this partnership engagement? Anyone?
You have a minute and a half.
Hey, y'all got to take advantage. We got a minute and a half. All right, Mr. Quinn, what you think?
Thank you, Brother Meriwether. I'm very excited to be a part of this group. I was diagnosed with prostate cancer in 24, had surgery this past July, and at one point I thought I was gonna die. I was literally planning my funeral because of my relationship with AJ, who 15 years ago was talking to me about prostate cancer. Wasn't even thinking about prostate cancer. Why is he, all geeked up about prostate cancer? And now I see why, and I'm a survivor of it. But I went to the post. He invited me several times. I finally went to the post and then started going consecutively after that first meeting. Being with men talking about similar issues is the most important part of this process, especially for the survivors. But we need to talk to men like AJ did with me 15 years ago, who not even thinking about prostate cancer. We need to talk to them years, 20 years out before they might be diagnosed with prostate cancer. Because like the Barber brother said yesterday, you don't know anything about it. You don't know what to ask. You don't even know how to spell or pronounce prostate. So this is important to get the information out. Yesterday was just overwhelming in a good way. And AJ asked me, was it too much? No, it's not too much. I've never been in a room with so many medically highly trained professional people in my life. I'm not a trained doctor, but I'm starting to feel like one because of all this information I'm absorbing and want to be a part of it. I want to share my story because if it can help save somebody's life and make a difference, I want to do it. Always include in your training and conferences testimonies from survivors. Don't leave that out. And you have been recognizing survivors, feeling like the VIP, you're a prostate cancer survivor, and being treated like a VIP. That is very significant and important. You want to hear the survivors' stories. I sat next to a man yesterday who had a procedure that I had, a very important, serious procedure. And I'm gonna wrap it up. These side effects from prostate cancer surgery and removal are more significant than the actual prostate cancer surgery. Dealing with the side effects. And I'm sharing with a couple of brothers the side effects that I'm experiencing and are getting fixed. He didn't know you could fix that. Some of these problems that medically can be addressed and can be fixed. So that's why it's so important to keep talking to survivors and sharing their stories and putting us together to share the story and getting over these fears. A rectal exam to me is nothing compared to all the other exams that I've had to go through. Just telling people what I've been through is just scary. Did you think you scared of a rectal exam? Just like the doctor told me, I got small fingers, I'll be in and out in no time. I pulled my pants down, I said, my ass is yours, do whatever you want to do. You do it. Thank you very much.
Thank you.
All right. So in closing, I know
we are
was
at
just
time.
absolutely wonderful, Theodore. But in closing, we just want to close by saying we are planning this year's next health fair. And I almost want to quote Senator Hill, but I don't know that I would get the quote right. But essentially what you said, you and Mr. Merriweather, was that we really want to blanket the city about PSA and screening and get that notoriety like a lot of other cancers get it. No knock against breast cancer and colon cancer. They've got to be dealt with. But our intent is to make PSA awareness as high profile as possible. And there will be some things coming in the fall that will be very interesting. And I will close by saying they will not be a success unless our community partners help us. So on that note, thank you, Mr. Merriweather.
Thank you. Okay, our next presentation will be on translational research and clinical trial service, and this will be again by Dr. Kim Barbell-Johnson.
I feed a sister and they want me to talk after lunch. And it slows me down getting up here, but again, it's an honor to be here. I'm aware at the back of the room how hard it is to read a lot of these slides. So if you can't read the slides, raise your hand. And if it's not just because you're my age and you're 61, then I can't do anything about that, but great, I'm glad you can see it. I have the honor of co-leading the Translational Research and Clinical Trials Core, and it's one of the many cores that actually provide oversight. I want to start with a little bit of a story. Again, you heard my son is having a birthday, and I just want you to kind of reframe before you listen to the details of the TRACS core. My son is on Broadway. He's a Broadway-trained musical theater person, and when he gets up and performs, he just performs. When you go to the show, there's 2,500 people in an audience, 8 shows a week, and they just see him performing. What they don't see is all the times his mama drove him to UF for voice lessons. What they don't see is all the people in the village who had to help me. All they don't see are the Willises dropping him off to 4 different classes because I had to do his daughter. I had to take care of my daughter as well. It's the behind the scenes work that actually creates a success story that we see in front of us. The TRACS core is like the behind the scenes work. And so I just want you to be aware as you hear what we're doing and why we're doing it, because we allow our trials to be successful and successful stars. So the mission of our core actually is before you, and it's actually to provide rigorous and equitable community-aligned research in this space. I'm actually going to try to take this off, be like Mr. Merriweather. And so we, way before we started to do this work, aligned and decided what our mission was going to be and the work that we were going to do. So we provide clinical oversight. We provide trained and tailored research support. We provide monitoring and administrative assistance to the teams for the clinical trials that are part of iCCaRE. And then we support community engagement, not because we're COE, but there are regulatory laws that are federal and now actually in the state of Florida are part of executive orders that say, how do you engage? What can you say on your posters? So there are lots of regulatory aspects that we can provide support. We're not Dr. Yost who's telling you how to ask your questions, just how you can do that within the regulations of clinical trial research and the statutes of the state. Our accomplishments have been many. I just wanted to mention that a lot of the data that you see is some of the data that we've had and collected for our first year with a couple updates, but we manage and support 3 full projects, and we're available and have capacity to support anything that comes up in the clinical trial space. We've had 4 regulatory bodies that we've been doing significant work, better understanding and aligning with, so from the FDA to NAFDAC and some of the things that you've heard earlier. And we've had 100% success, and I think that deserves a round of applause for our goals, for our goal to core goals for last year. And we are modest in what we say we can achieve, so we like to be conservative and then produce. But what's most important is that we are the space that we are international. As a core, we are actually living between two different continents as we do this work. And so we've had some regulatory wins, which have actually been part of our 100%. So onboarding our Nigeria site was one of them. Managing the timelines that we've talked about with 6-hour time differences, because this is an international collaborative core. Understanding the strategic framework of that work that we're doing. So those have been our wins, and we're very grateful because it's taken a team to do that. We've also understand that we need to have regulatory wins because, like I said, there's work that goes behind it. We can't just create a paper. Again, this is something that I think is important for our members of the support group, for our citizen scientists to understand, because when you say they're doing their due diligence back at Mayo in iCCaRE so that this is not exploratory, this is not hemiatolax, you need to be able to say that with confidence. So I want you to know that we are behind the scenes doing our data and infrastructure work, and we're winning at those things. So when we had to go through all of the stages to get NAFDAC approval, where are you, Dr. Rotimi and Dr. Odetna, to get our FDA, imagine me saying I'm going to go call the FDA in Washington, D.C. Well, Dr. Odetna and Dr. Rotimi could do that in Nigeria. So we were able to finally get NAFDAC approval, which allowed us to move forward with all the things that you would kind of understand from the FDA point of view here. Sandra Casanova, are you still in the room? I'm not sure. But with Sandra's help, we've been able to really standardize how we're going to collect our information in our clinical research operations system, our REDCap system, and that is now completed. So we had many calls where we had teams in both countries understanding this, and we had regulatory people helping us to build that data management system. And then we've been standardizing our operations. So that one thing doesn't happen one way in one country and one thing happens another way. So we've actually now really developed our new and improved SOP with our new members and with our new liaison, Dr. Falaki Odetna. And so we just recently had that endorsed by the core, and now that it's endorsed, it will be fully operational, and we will be circulating that to the entire group. Then, of course, the biospecimen, a cold chain. And what does that mean to you? Do we just collect blood and send it across the ocean and figure out where it's going to go? Well, there's chain of custody and lots of different things that happen, not only in getting the sample right at the right temperature and the right tube with the right analytics that need to go into that. But those samples, we need to understand, are they going from a Lauren to the US, or do they have to go to Dr. Rotimi's house or Dr. Rotimi's university at University of Covenant University? Where's the airport? So we need to understand that we preserve what that looks like so that we can help our teams. And this is just for one of our 3 trials. So again, accomplishments that we're very happy to do. Again, we have 3 trials that we're responsible for. One is definitely, leading without us, because we have the expertise of having Dr. Detna as the PI of the space, but we still are here. We're still here at the VR Assistant for Prostate Cancer Education space, just understanding the full project, supporting those things that the tracks can do, and partnering in oversight for the oncological and behavioral science that's involved in that space. And we have continued to identify ways that we can continue to help. So our next steps are to be able to continue to support the testing as that happens, just making sure that the VR assistant evaluation and the metrics are happening in a space that is consistent with all of the other research that's happening at Mayo Clinic. With Dr. Dronka's project, which you all had questions about, and nobody had questions for me, but that's okay, that you all had questions about today, because that is the most exciting thing. It's so amazing that we have a project like that I don't know if you actually picked up on what she dropped down about. We're looking at that in the practice space. Can you imagine that we actually are doing 2 things at once? Usually you have research and you have to wait on the research to be done, and then the practice decides what you're going to do. This is so successful that we are actually in parallel developing these spaces because time is ticking. So as we do that, the TRACS core is actually making sure that we're providing the timeline assistance that they need, again, the contributions from the regulatory space, and we've identified, again, some more next steps. My project that I happen to be on the IRB for this one in terms of vitamin D supplementation, you heard a little bit about the science behind that, but this has really been the core work. I would say 75% of what we did in year one was really supporting the regulatory progress and the data and biospecimen infrastructure and the active issues for this trial. And I say that if I asked you, like, do you use liquid nitrogen or do you use dry ice to ship something, those nitty-gritty things, it's not my core, but the best part of being a core is that we have other cores that take care of that work and we can sit with, so we can be with a biospecimen core and deliberate and collaborate with these things. So while it is the TRACS core, we are finding ways all the time to work with our cores across the iCCaRE ecosystem to do the work that needs to be done. This was a tracker. The tracker that I just wanted to highlight that we are meeting our goals and we're measuring what we're doing. So this is not a feel-good space. The space that you see 50% is where we're focusing on making sure that we can improve, but we've got to have areas for improvement. If we came in 100% every year, we'd have no work to do. And then lastly, we have challenges and mitigation strategies that we create based on the things that we have. So again, the science that's behind that, I'm happy to report some of the things that actually was reported. The last two items that you see were actually reported as not yet completed, but they are now completed, but we continue to evolve the science where this core is concerned. That list is outdated, so I won't share with these names, but I do want to actually just recognize that my co-lead is actually Nigeria resident and lives in Nigeria and is directly supporting all of the Nigeria activities. Dr. Ojo, in her absence, I want to give her credit. I want to actually thank Dr. Cologne for being the co-lead for up until our recent switch. And again, thank Dr. Dronka for her MPI assistance last year. But Dr. Detner, we cannot move forward without you, so we're happy to have you on board as our MPI. And with that, I thank you very much.
Thank you, Dr. Barbell Johnson. We're going to move on to Methodology and Measure Services Core, and that will be with Dr. Mary Ellen Young.
I like the idea of holding this.
Yeah, good. Thank you, Dr. Bonner. So I'm going to be talking about methodology and measures and our core. And as I was listening to the other cores and specifically to Mr. Merriweather always, I was thinking the investigators in this room already know what MAMS is. So you're not my audience. My audience, I think, are advocates who may not have been trained in scientific methods or have exposure to the science. So what we do in our MAMS core is to help improve and bring to the highest level the science that's done across the projects. And so this is to help you understand what we're doing behind the scenes in working with our investigators and their teams. So we have a core. Dr. Yost leads our core. She's in Rochester and couldn't be with us today. Dr. Oladipo in Africa and myself as a consultant. We have other members working with us and helping us stay on track. I will say that Dr. Cannonjin, where are you? Yeah, there you are. He is our newly designated MPI liaison, so welcome to MAMS. Okay, so what do we do in MAMS? We are here to promote best practices across the research continuum, across the studies. And we're particularly focused on research design and measures, particularly things like surveys, questions, and the qualitative research that we do. We're establishing procedures. We want standardized operating procedures across the projects whenever possible that are tailored to iCCaRE and our goals. So we help with any of the studies that have surveys, questionnaires, qualitative questions, focus groups, those kinds of things to both develop the instrument and to help the investigators implement it, and then even to help interpret it, all right? And so we consult on basically all of the iCCaRE projects, except for the ones that are biobanking tissue, because we don't give them surveys or ask them questions, okay? So what we did in our phase 1 study was to establish our infrastructure and to advise the pilot projects. We learned a lot by advising the pilot projects and the pilot projects coming back to us, and a lot of give and take. In the beginning, it was, what is MAMS and why do I have to send them my questionnaire? Because I've already got IRB approval, which for advocates means it's already gone through and been approved, and then MAMS is coming back and saying, but wait, you need to change this, this, and this. So our investigators were not always happy with us, but we did develop a series of documents and resources for our investigators to use. So that's what's on the right-hand side of the screen. We have best practices for qualitative research. That's the interviews, the focus groups, the times when we're sitting down with men with prostate cancer and talking to them about their experience. That's part of what we help the investigators design that questionnaire and those practices to collect that data. We have worked on establishing some focus group best practices and focus group training, because a lot of people want to use focus groups to help identify and explore ideas or find out opinions or experiences. And so doing focus groups is a very specific kind of data collection where it has to be done right. Otherwise, you're wasting all those resources for those focus groups. We also help with designing survey questions. And this is Dr. Yost's area of expertise. And she is really, really good at helping to make sure the wording of the question is correct, the measurement, how that is scaled, the answers are scaled, and how they're used. She's really an expert on that. We also have some standardized questions. So we decided early on that we would like to look across the studies and say, so if you're going to collect data on marital status, let's collect data on marital status the same way across 10 different projects, so that at some point you may be able to say, iCCaRE served this many men of this marital status and this age and this income, and all the demographics being standardized. We have a recommended data set and some funneling, and that's our graphic here. So the blue questions are things that we think all the projects need to be asking in a standardized way. So things like race and ethnicity, age, education, overall health, quality of life, those questions can be standardized across the projects. Then the yellow is for questions that are used in many, many projects. And so we have standardized language and standardized scaling for those questions, things like socioeconomic status, social determinants of health, symptoms and side effects of prostate cancer. And then in the red, we have things that are specific to a particular study. And we will review those and help the investigator make sure that they're being worded correctly and those kinds of things. But that's just specific to one study. So here's an example of a standardized demographic questionnaire for year of birth, education in the US. We also have gathered data on education in Africa because the categories are different, and the Caribbean. So this is just an example of a spreadsheet that we have that's available to investigators. So when they say we want to ask about educational level, they can go and see how we're recommending that it be assessed. We have our standard operating procedures and our best practices and measurement tools for full projects 1, 2, 3, 4, and pilot project PP6. And we have 3 projects in Africa that are beginning in the beginning stages of feasibility and those kinds of things. And each project has a MAMS member liaison. So this is, we were asked for a metric. This is our metric. So these are the projects that we've been involved with. The ones that are full projects were pilot projects, so we were involved in those. And the new Afrocentric projects are ongoing, and PP6 with Dr. Coburn, we're on that too. So each project has an investigator embedded in that project. That means that we attend the team meetings, we understand the protocol, we understand the problems that people are having, what they want to ask, how they're going to get it, and we help advise on that.
You have 1 more minute.
I gotcha. And I'm on my conclusion slide. So the MAMS score has strengthened methodological rigor. So for our advocates, know that the science is sound. That's the take-home message for you, is that there are people that are dedicated to making sure that when iCCaRE does something, they do it right. And the other cores do that as well. But we're really proud of that in our MAMS score. This year, we'll continue to review new projects and work with the old projects and see our march to success. I have 3 seconds. Thank you all.
Thank you, Dr. Young. We're going to move on to Digital Health and Cancer Communications Core, and that's going to be with Dr. Ngufor.
Hello, iCCaRE. Hello, iCCaRE. Please don't sleep on me now. Okay, so I'm going to present Digital Health and Cancer Communication Core. We're still in the phase of figuring out our name because we don't really do cancer communication. It's another group, the media group, that actually does the real cancer communication. So, but anyway, this name fits us for now. So, but here's my team. So I'm the MPR liaison, and the lead is Dr. Blocker. Unfortunately, he's not able to attend, so I'll do that work for him. And we have our team here. Unfortunately, I don't have images for them, so next time I'll try to do that. But I want to stop here and, really first thank, our community MPIs, our community scientists, those that were involved with the VIRA. Because the VIRA is really one of our core
innovation in this core. Go back. The Vira is really one of our core innovation in this course. So I really want to thank Mr. Merriweather, former Senator Tony Hill, and Mr. Abiola, Pastor McCoy. And before that, I want to say, please let us stand for a moment of silence for Mr. Abiola. Thank you. So Mr. Abiola was really instrumental in the early phases of the Vira development. His perspective was so unique. And in fact, we saw that when we went to Nigeria, the survivors that tried Vira, the way his own approach, again, Vira is that kind of culturally grounded, and we were able to connect to that. That's telling us a lot. But so thank you again, Mr. Abiola. So our mission here is really to design and develop iCCaRE digital interventions and provide data science and AI services to support all of iCCaRE projects. So we are that center, provide data science, provide AI to everybody that needs it. But so far we have got just one group that has approached us for AI services, but we are happy to provide them support. So, as I mentioned at the top there, currently the Vira is really our core or our flagship innovation, and we are doing everything to deliver that product as fast as we can. There are stumbling blocks along the way, but those are the challenges that we will deal with. So, what is Vira? And I hope our survivors can really, if you have any question, what is this Vira? Why did we choose to go with Vira? And again, Dr. Odena did a good job to explain why Vira, the history, why is it coming. So basically, we have AI, extended reality or virtual reality that is culturally grounded. And this is key. We are not just building virtual reality product for virtual reality's sake. Others can do that. But we want something that is culturally grounded. And because this is part of the iCCaRE study. So basically, when I jumped into iCCaRE, it was like, okay, Dr. Odena has done all this work, and now there is that need of the technology, as she explained, but how do we deliver it? So initially, I didn't really know what was going to be Vira, what was going to be, but the Vira thing was something that I had back in 2017, but it was like, well, I want to be able to develop an application. But has anybody watched the Netflix show Alternate Carbon? This is a Netflix show. It's way ahead of its time, but basically humans and AI are co-interacting together like the regular real world. But that's when you immerse in that environment. But when you get out of that environment, you're in the real world. When you get into that environment, it's something completely new. And AI is there providing you with instruction on what to do, give you your schedule, give everything as you go. And I'm thinking, okay, well, maybe you were just recently diagnosed with prostate cancer, and the AI is going to tell you, well, if you want to scream the hell out, I'll take you to Mount Everest, and you can scream as you like. Or I can take you to a place where there's calm, and you can get or spiritual. So that was the concept. So I think, but we didn't have it there. We didn't have the technology there. So then this thing come up. The stories. We want to tell you stories. The gentleman will talk about the stories there. We want to tell you stories. So we decided that was the angle that is going to be the hit. So we got our prostate cancer survivors together and let them tell your story. And then we captured that in the meta-human. And then again, with the CAPCAS, we need to address social determinant of health. We bring in the way to address social determinant of health, emotional, spiritual, and all that, and all of these things just coming. So now the key innovation part of it, as Vira is getting a lot of traction, but there's so much that we haven't started yet. There's so much that this platform can deliver. And I just use the word platform, because the Vira has moved
from that initial concept, which is my next slide, to now a platform. This is a platform that will address the AI. AI is everywhere, there is here to stay. The core component of AI, the core component of giving you that education, giving you what is in your data, what is in your data that is driving that risk. Professor Rotimi, all your study is embedded there. Somebody can go in there and see what is driving my risks. What is making me have this sort of, treatment regret, or what are treatment toxicity? And all of that you can see within the Vera platform. For now, we have this storytelling to be able to address that social, emotional, spiritual need at the point of prostate cancer that we've developed and we are ready to deliver. First, do the pragmatic clinical trial. So I've already said most of this. Our core message here is, again, as I mentioned, it's not a conceptual now, it's a validated and it's ready for deployment. Now, there's a lot, I think we just mentioned here, there's so many things that go behind the scene that, it's really the key driver. For us to be able to have this application deployed and it's ready, there's so much that go behind the scene. But the beauty of doing all of that is that we are setting up a foundation, we're setting up a platform that any application within iCCaRE can leverage it almost immediately and deploy your applications. So the platform behind Vera is a complete scalable platform built on the Mayo Clinic cloud.
You have 2 minutes.
So everything is driven by Cloud Run, it's driven by, the application, so it's robust, it's scalable. But above all, this is one new thing, the Mayo branding is not there because iCCaRE is global. So you will not see the Mayo branding like what Mayo does. We like to brand ourselves, that's true. But for iCCaRE, you will not see the Mayo branding because of the global consortium. So now, Dr. O'Neill also talked about the, when you have a technology like this, we need to address how it's going to be adopted. So within iCCaRE, we also have the digital training or learning, and we have the co-tech labs where we actually be able to train, survivors, anybody to be able to use this technology. And this is a platform, again, that we're building, and it's going to be part of this Vera. Now, the session of the training of the survivors, the advocacy, that was also another interesting angle as I attended that vision also. But the reason why I attended was, as we are building out the Vera, we have the 3D and we have the 2D, which is my next slide. But there's also that component of, after you go through all of this, the next thing is become an advocate. Once you go through all that, and how do you get that advocate training? Bam, we have Vera, and we can incorporate all of that within the Vera, where you can go through that training, and all that is delivered within that platform. So I think that's what I was just talking about, the 2D part. Again, we have the 3D, and then we have the 2D. Now, the concept of the 2D is basically using social media. So we thought of, using YouTube style, Netflix style, and provide you with those interventions. So that's what we've done. So when you go to that platform, you'll just be like Netflix. All those interventions that the survivor done is just rolling. The recommended one by the AI is going to be provided to you, and then you can choose which one you want to go. You can build your storyline, build what you want to watch. It's a Netflix, but this is a beauty too. It's scalable. All the videos that we've taken in iCCaRE can be incorporated into that platform. And that's the vision. Arrange in such a way that you can do search. I can go there and say, provide me a video where Mr. Merriweather is talking about blah, 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 blah. And it's going to come out with that video with context, those voices, who is behind that, and all that. So that's the 2D part. But essentially, we want to be able to address the equity, whether you have 3D or you have no access to 3D, you can have access to 2D. but also there's access to the regular part, maybe just pamphlet and all that. So how about building out that agentic way where it can actually select which part to actually present Vera to you? That's some of the next concept. Thank you.
Thank you,
I
Dr.
still have a lot of minutes.
Look, I was about to stop you.
Anyway, I'm open. Do we take questions now? Or
No. After.
There's a panel, okay? Well,
Thank you, Dr. Shea. Our next presentation will be from Mr. Daniel Lee. He's going to be Data Management and Analytics Service Corps.
Good day. I'm going to look to see if the slides were updated. They weren't. Okay, cool. So I'm with Damas. Oops, sorry. The Data Management and Analytics Service team. It's a team we have team members in Nigeria, here in Florida, Texas, and we are, I don't want to say the nerds, but the analytical nerds. And I'm a senior biostatistician with Quantitative Health Services, but I like to call myself a data wrangler. So we have a data dictionary that consists of over 15 different surveys, studies that we have to where we have the ability to standardize all the information. If we're thinking about a knowledge score or a prostate cancer screening awareness score, we have that standardized across all these surveys. So when you're thinking of multi-site analysis or different ways that we can aggregate different data sets, that's where we come into play. Where if you're thinking about a sealed data set that we have that's focused on trust of the doctors around COVID period, if you're thinking about red meat studies, we have all the databases to where we can plug it in if you are focusing on survivorship or focusing on, readiness or participation, for example. Where do you like to get your research done? If you want to see the doctor, do you prefer it in the home or in the hospital, things of that nature? We have all those and able to do that. And really, a lot of people are saying scalability and reproducibility, and that's where we have it, where we're using SAS, Python, R, having notes notated to where other statistical programmers, other statisticians can come in and replicate everything that we're doing. Standardizing outputs. So one thing for me, whenever I'm looking at analysis, I can go super deep and it's going to take me a while because I'm thinking regression, but sometimes we have to take a step forward and look at just associations, simple bar charts, and having the ability to explain it to a high-level individual or someone who doesn't know much at all. So standardizing outputs, identifying key factors. We can go into p-values, test statistics, and things of that nature, but we have the capability and the power to explain it and to be able to present it in different areas. So this is a snapshot. So the ability to see different things that we have in-house already, where you see the familial cohort study, Ironman, Red Meat study, mindfulness, all the pilot projects, SEAL, Collab. It says MCAP, but it's MyCap. These are all just different participants that we have to where aggregated, it's definitely over 17,000, where it's not public data where you can just go and find it. This is stuff that we only have in-house. So one thing I like to bring up is the Ironman study that we have in-house, where a longitudinal study looking at over 3 years of data, maybe 17 different time points where you have survey information, and being able to make a wide, long data set cohesive with other data sets is something that stands us against others. So this is kind of the flow of how things go. A lot of respect and appreciation to MAMS who can help us and helps us, making sure that we're doing things on a consistent basis. But we'll have the meetings with the different teams, focus on the design, the hypothesis, what we're trying to test, and going on, looking at these data sets and producing just initially associations. What do we have and what can we do? Is this something that's research that we can write about? And one thing that I've learned is that all research is worth mentioning and telling the story about. I'm looking at, is it statistically significant? But just because it's not statistically significant doesn't mean it's not valuable research. So making sure that the analysis is clean and making sure that what we're producing, that we're reporting it all the time. So. Decided to show different visuals. So this is using two different data sets. One was a familial cohort study, and another one was, I don't think it was a SEAL data set, but another data set that we have. And it's just looking at screening uptake. And if you're looking at it, these are percentages that you see. So 70% of people who got screened, they had a family history of it. And as I think of different community engagements, people who are coming to those community engagement events usually know somebody who had prostate cancer or had prostate cancer. And as we see from this data, that told that story. 70% of the people who got screened had a family history of it, as compared to if you didn't have a family history, it was 34%. Condition of 36% saying that you may have thought you had prostate cancer, or you may have thought you had symptoms, 36%. But if you didn't have a condition or any symptoms or anything, 16% of people got screened. And that's a telling story. So basically, if I don't know anybody who has prostate cancer, the likelihood of me getting screened is low. Or not non-existent, but it's going to be low. And this is, I think, during the advocacy training, something I was hearing as well, where how can we get more people involved who are not diagnosed with it or know someone who's diagnosed with it. Me, myself, I'm 35, and until I started working at Mayo Clinic, I didn't even know what a prostate was or where the prostate was. So I'm assuming there's more others like that, to where there's 16% of people who don't have the condition or no family members who have it, raising this up to 36% or 34%, which is going to take educating. I think a gentleman said people who are 20 years younger than 50 or 40. So education at an earlier age and continuing it is going to make sure that people who don't have family history or conditions of prostate cancer will get screened. This is a spider web plot that kind of shows the different levels that we can go per study. It may be different for every single study. So the orange is a Diamond study, which is one of the, I don't want to say difficult, but a study that you can see the different levels and breadth that we can go on a study, where the study design is a high level 5, longitudinal, reproducing this data, statistical analysis, data cleaning, and data collection. So any study that we're working on goes through these steps, but the level of detail will be determined just based off the data that we have. Cool. So, and I'll say here and then go to conclusion, but so with a critical backbone of the data and the team, and we're definitely, anytime you have any questions about data analysis or want to study and plan about how in the future we can use other data sets to help tell the story as secondary data. So when you think of the other list of data sets that I listed, just think of those as secondary sources that you can use to supplement any full projects or pilot projects that you're working on. And Demas, we're atypical to the typical statistician, to where we enjoy doing the community engagement and being boots on the ground. There are other slides that I would show where in Houston, Texas, where we're partnered with MD Anderson for Pilot Project 4, but we've built that collaboration to the point where we're able to do prostate cancer screenings on site. And as people have said, whenever you're doing community engagement, they're going to ask, okay, this sounds good. We appreciate the education, but now what? And in Houston, now we're able to do PSA tests after the community engagement. MD Anderson was able to give us a lot of different vendors. Well, they can come and do tabling sessions. They can do other screenings, diabetes screenings, blood pressure tests, all under the name of iCCaRE. And that's shown the breadth that we can go. So, and I think, yeah, the next one's conclusion, but that's it for me.
All right, thank you, thank you, thank you. We're gonna keep pushing forward. Our next presenter is gonna be Dr. Damian Francis. He's ready, and we'll be discussing planning and evaluation core.
Good afternoon, everyone. All right, some people are working, some people are sleeping, so let's see if we can get some participation going. Now, in the evaluation world, my job can either be very easy or it can be very hard. If the team is doing a poor job, my job is easy. If the team is doing an okay job, my job is easy. If the team is doing an excellent job, my job gets harder. So let me ask you this. Do you think my job in evaluating iCCaRE is easy or hard? What do you say? You think my job is hard? Okay, we'll see, we'll see. The case is out, it's gonna be decided. You're gonna see if my job is easy or my job is hard. And one of the reasons we think it's easy if you're doing poorly is we'll tell you you're doing poorly and you need to do better. If you're doing an okay job, we'll simply say, everything is okay, continue doing what you're doing. If you're doing an excellent job, it becomes so hard for us as evaluators to find areas for growth opportunity and what you can do next. And we're gonna see if that's the case with iCCaRE. So I'm Damian, I'm from Georgia College and State University. With me are members of our Planning and Evaluation Core. If you're a part of the Planning and Evaluation Core, just stand up and give a wave. So some lovely people. There are a couple of people who are not here. Dr. Bassi is not here. Prof. Akoy is not here, but they are also members of our core. So we have two primary objectives today. We have to look on the overview of year one, the evaluation domains, and we're also looking at progress of each of the projects, pilots, as well as the cores. If you want to see progress tracking and benchmark in terms of did you accomplish what you set out to accomplish, there's a poster in the poster session that documents that, and I will focus on our 5 evaluation domains. So this is our, over here, over there, you'll see our evaluation framework. What I want to focus your attention on is to the far right of the screen, you will see 5 domains that's listed on the screen here. We're going to go through each of these 5 domains and see how iCCaRE is doing on each of these 5 domains. Now, to get to these 5 domains, we collect data from various sources across projects, across cores, and we do some external surveying in terms of our event surveys that are ran through our PES core or some of the things that are done through the media core. So in terms of our collaboration and quality of communication, based on this diagram here, and where are the AI supporters? Raise your hand if you're a big AI supporter. My first rendition of this diagram was with AI and it made a mess of it, so I manually did it. So hey, there you go, AI folks. So one of the things we're showing here is that the collaboration is wide across multiple sites, multiple countries, multiple institutions. There is a lot of structure in terms of what iCCaRE does. in meeting cadence, in terms of the communication channel, all of that is established. In fact, when I reflected on it, I think we have so many channels and communication tools that sometimes we forget about the tools and the channels we have, and we pick up the phone or we send an email when we have an absolutely sophisticated tool to do that kind of communication. So in concluding on domain one, we can say that there is strong multi-institutional community partnership. There is formalized meeting cadence and governance structure. There is high level of engagement with both academic and community stakeholders. As you have seen here today, there are a lot of community partners here, survivors, community MPIs. And so in terms of domain one, the evaluation summary is that we're doing well there. Domain two, performance of projects and infrastructure. Our objective here is to evaluate the performance of interventions and projects. And as you can see, we have core infrastructure readiness. This is through IRB approvals and regulatory compliance. There is also operational data platforms like REDCap, digital health tools, biobanking infrastructure. So lots of things that are in place to guarantee the success of iCCaRE going into future years. In terms of capacity building, a lot has been done in preparing the platform. As you heard, I've kind of went ahead in preparing this, even though I didn't see Prof. Che presentation. I've been calling Vera platform even before. Why? Because I read your report. And so we see it in there where there's this capacity building. There's also program delivery success, in that multiple studies have transitioned from the planning stage to activation stage, albeit there are some regulatory hiccups that are there, and we have some best practice recommendations where that is concerned. There's also centralized data management and analytics. So a shout out to Damas. They're doing well in terms of data integrity, monitoring, qualitative and quantitative analysis, harmonization of measures across projects, as was mentioned by MAMS earlier. We see that happening. Data collection tool, there's standardization of instruments, REDCap implementation, and again, data tracking and monitoring. So our key indicators are telling us that our cores are doing well, and therefore our projects are going to do well because they are supported by the cores. In terms of just some granular look at data quality, as you can see, REDCap for data capture used by 4 projects. And as you go down that list, you can see what the different projects are using in terms of instruments and or platforms. In terms of quantitative data analysis, again, another look at some of the projects that are doing specific quantitative data methods. You can see that from FP1 all the way through to PP6, what they're using and how they're doing that. Again, projects using qualitative design. Shout out to my qualitative folks. A lot of times as epidemiologists and data scientists, we love quantitative stuff. Everybody loves numbers. But shout out to Dr. Young, Dr. Keninjin, my qualitative gurus. 3 of the projects are using qualitative data and state that explicitly in their reports. In terms of our domain 4, scientific productivity, we see that iCCaRE has exceeded all the set goals for themselves. So again, they're not doing okay here. Are they doing poorly? No, they're doing what? Exceptional, which means my job is hard. There you go. So in terms of presentation, 45 in year 1, but this is where it gets interesting. Publications, 17. And I took a deep dive into the publications because what I wanted to do was to kind of map where these publications are. And because they're so early in the pipeline of publication, I am tracking, and I'll be doing this for the next couple of years to see how they get uptake into, for example, clinical practice guidelines, how they get cited by other literature. And so when we look at that, the lag time is about 2 to 5 years to see where your publications are going and the impact that they are having. Grant submission, trainees, all of that is happening above and beyond the bar set by the different cores and projects. So some key indicators for domain 5, impact on member organization or community. So you saw firsthand many of the health education programs, be it community screening, digital literacy, cancer awareness, community advisory boards engaged in the study design. We heard earlier that community PIs were involved in writing the grant proposal, and that is something that we don't often see in the co-led approach, and this is wonderful. Health equity initiatives, so targeted outreach for underserved populations is going on, digital health navigation, and education for marginalized communities. Want to give a shout out to, I think they call themselves COE, that table at the back, so shout out to Chelsea. Wonderful job in some of the health initiatives, health equity initiatives. Workforce development, training for students, early career investigators, advocates, and global trainees. Shout out to the advocates who were trained yesterday. So again, you see that this is not just talk, but it's real action. Global impact, we're engaged across the United States and multiple African regions. And of course, we're also in the UK. I'm happy to see that iCCaRE is growing. There is collaborative workshops, advocacy program, knowledge dissemination internationally. I think this is kind of one of my favorite slides in the presentation. Again, I saw Bims earlier, and I just had to give her a shout out. When we look at what the Media Corps is doing for dissemination or translation to the community, it packages all that the cores and the projects are doing, and we have seen that it has a 5-star rating on iCCaRE platform, which indicates that the content that we're sharing in our media YouTube, on our LinkedIn, that this is really high quality stuff. The planned episodes have been 2 times what it was planned for. So they planned to have 20, but they ended up with 42 episodes. The media reach, 15,000 views, LinkedIn engagement, over 20,000. And we are beginning to see translation to systematic reviews, clinical practice guidelines, and other publications, which is something we're watching for in the future. So in concluding, lessons learned, there is strong communication and collaboration. There is some delay sometimes in study activation and onboarding of new sites. Opportunities to strengthen these does exist, and we're going to look to see how we can strengthen that. There is a need for greater inter-project and core measurement. It is happening, but again, we have tools to measure how the projects are collaborating and how the cores are collaborating. We just need to capture that a little bit better. Navigating challenges with regulatory process and study activation requires multiple on-site or multi-site coordination, and there is significant growth opportunity that exists for measuring consortium networking density, meaning the frequency and the strength of the collaboration between cores and projects. And the last slide is that one of the things we want to do going forward is to assess the community, meaning our survivors and our community MPIs engagement in iCCaRE. One of the things that we have done well is to say how valued our community partners are, but we need to publish and show how when a consortium includes our community partners at every level, how that makes for more and greater uptake and impact in the long run. And so we plan to do that through using the active framework. This is an already developed framework by Sean Pollock and some others. And we're going to use that in iCCaRE in the future. And then continuous improvement of feedback loop for evaluation results and projects and cores, and then a social network analysis for documenting consortium impact. It's always great to have the timekeeper being a part of our core. Therefore, she's not giving me the time signal. Though my time is up.
Thank you, Dr. Francis. Our next presenter will be Ms. Bims. And of course, this is our Communications and Media Core.
Okay, hello, and I'm going to be doing communications and media. Unfortunately, Dr. Kim Walsh-Childers and Dr. Ibasi were not able to join me, so it'll just be me today. And just a little bit of background, here are all of the faces that are a part of the media core. Some people are in here. And at the bottom are some of our series. We have Vera. At the bottom, we have the Barbershop series. We have Men's Health Matters, To the right is Prostate House of Humor. Underneath is a music video, one of our iCARE songs by Sheena. And we have Stronger Together, and then we also have an interview at the bottom there. So our problem is persistent cancer disparities are driven by limited access to information, low trust within the healthcare systems, as well as poor engagement with traditional dissemination. And scientific discoveries often are not culturally translated, as well as they don't reach the target population. And where we have a huge gap here is that science is not the same as community impact. So our goal here for iCARE is to establish a scalable community center communication ecosystem that integrates science, media, technology, and lived experiences to meaningfully engage underserved populations in order to build trust and translate research into action. And through this model, iCARE aims to help bridge the gap between scientific discoveries and real-world impact by delivering cultural relevant, accessible, and multi-channel communications that not only inform, but also empower. And ultimately, this approach positions communication as a core that's driving health equity, enabling sustained engagement, as well as improved participation in research and measurable progress, eliminating health disparities across the African diaspora. And for the iCARE communication model, it is a multi-level integrated ecosystem. It combines media, community, technology, and science. And our main goal here is to, again, help build scalable, culturally grounded communication ecosystems that transform scientific discovery into community impact, as well as advance global cancer equity. So for our approach, we have this central hub, which is our iCARE media ecosystem, and it goes from scientific outputs into our social media channels, into community programs, iCARE TV, into our digital platforms like YouTube, and back into our scientific outputs, and it's just a continuous loop. And so for all of our platforms, we have LinkedIn, X, also known as Twitter, Facebook, YouTube, and iCARE TV. LinkedIn is where we have professional engagement. X is real-time amplification, and Facebook is community awareness. YouTube is storytelling as well as education, and iCARE TV is a narrative and trust building platform. So the framework is iCARE employs a multi-channel audience tailored dissemination approach that integrates real-time communication, professional engagement, community visibility, long-form education, and I have here just a little bit of statistics. I'll go through them later in the chart. But for LinkedIn, we post educational posts, research highlights, and consortium updates. Those are where we get the most impressions from. And for Twitter, we get the most impressions from campaigns, awareness threads, and daily knowledge series. For Facebook, we get the most impressions on flyers and event promotions, as well as reels. For YouTube, we get the most views on survivor stories, expert interviews, and clinical trials. And key methodology principle, the different platforms serve distinct roles. They all impact is achieved through intentional integration of all channels. And then so culturally tailored messaging, we are designing this for Black men in the African diaspora, as well as improving relevance and engagement. And our Knowledge is Power series, it's on Twitter. It just defines medical terms. And on Twitter, one of our biggest impressions and one of our biggest engagements we've ever had was, Dr. Odedina mentioned this earlier, the Movember Dance Challenge. About 2 years ago, we had 5 different iCARE CAP-TC sites in Africa do a dance challenge, and still to this day, each of those videos have the most likes, retweets, and shares of all. And the interactive, culturally relevant content drives maximum engagement. So communication integrated into research design, clinical trial engagement, and community outreach. And it's treated as behavioral intervention as well as implementation intervention. And so for the results, so for LinkedIn, we have about 298 followers. We have about over 44,000 post views overall. We have about 143 posts, a little over 2,000 reactions, and about 566 page visits. And here the key insight is that we have a higher reach beyond a follower base, and we have a strong content amplification. Now for Twitter, we have about over 52,000 impressions, about 4,600 engagements, over 1,600 likes, and over 813 reposts. And Twitter is highly effective for real-time engagement as well as campaign amplifications. And this is just a little chart of LinkedIn with all of our page visits, reactions, and then the same thing with X, just like a little visual. And so for Facebook, we have about 200 posts, we have 284 followers, and we have about 153 photos on there. And the key insight here is that visual and event-based content drives awareness the most. And lastly, for YouTube, we have about 1,260 subscribers, which was a 225 increase since last year. We have about 8,185 views, over 58,000 impressions, and about 632.7 hours of watch time total. And lastly, we have about 7,800 page visits. And our key insight for YouTube is that it is a long form content that deepens engagement as well as retention. So this is just the chart for the Facebook analytics, and then this is for YouTube. And then these are some screen grabs from our YouTube analytics. And here I just thought it was very interesting that majority of our audience on YouTube are between the ages of 18 to 38. I do remember somebody mentioning that it's very important for the younger audience to be aware of this. And I think it's really cool that that's the bulk of our audience. And then of course, most of our demographic are watching our YouTube channel on their mobile phone. I already mentioned the dance challenge earlier, but in total, all of the dance challenge hashtags created about 21,562 impressions overall, about 323 reposts, 289 comments. And what we learned here is that interactive as well as culturally relevant content drives us the highest engagement. And the Knowledge is Power series, again, it's just defining medical terms. And this is super important. We have about over 19,000 engagements, over 7,200 likes, and over 3,200 reposts. And our insight from the Knowledge is Power segment is that it is a micro educational content that sustains consistent engagement. And impact comes from integration, not individual platforms. Here's just a little chart showing that LinkedIn is credibility, X is for amplifying, Facebook is visibility, and YouTube for depth. And this is a chart here. I won't go through it because I know I'm a little pressed on time. Overall, our big goal is to increase awareness, reduce stigma, increase engagement, as well as improve research participation. And in conclusion, iCCaRE demonstrates that effective cancer communication requires integration of media, community, and science. Thank you.
Thank you, James. That was great. And now I think we're on our last core presentation, and that is going to be Dr. Reynolds. You are up. Dr. Reynolds is our Biospecimen Resource Core.
Thank you very much, everybody, for having me. Sorry, I couldn't be more in person, but I'm on clinical service today, but I'm glad I have a chance to present to you today. So our core focuses on two things for this consortium grant. We are trying to be a way to organize ourselves for all the tissue that's stored, any samples at all locations. The good news is I work at Mayo Clinic and we have a very robust way of storing samples and cataloging them here at Mayo Clinic. The bad news is the system is rather closed. It's to protect patients. So we did not have a system to reach outside collaborators. But necessity is the mother of invention. So we collaborated with the FedGen group. And the FedGen group is a research group in Nigeria. It involves Covenant University, OTA, Nigeria. And FedGen stands for Federated General Omics. And it's a research group that comes up with cloud infrastructure for various things. And one of the tools that they proposed for us is something called the iCCaRE repository overview, which would help us to catalog all the specimens that are being stored at other universities in the United States and in Nigeria and Cameroon and other countries in Africa who wish to collaborate. It's going to be an open system that will allow you to see where samples are in freezers, what the diagnosis is there any molecular information? And we did receive some funding. We were one of the, here, let me see the proper name of it. We were one of the awards, the Developmental Research Program Award. We got monies to fund this software. So here's an example of how it could look. This is the proposal where you could upload information about your biosample, and you could do a sample search and find this. And it would be web-based, so you can log in. There would be a dashboard. It would provide openness for collaborators. That way everybody knows what's going in and everybody knows what's going out, so it would be fair. I think what we need to do next is, after this platform is up and running, we need to validate it. And we have a novel idea of, instead of validating with confidential patient samples, we could probably use a few, but we plan on using generated AI to generate as many samples as we want to, of patient information, molecular data, just to see if the cataloging system works, see if we can find samples where we said we were going to put them. You know how it is if you set something somewhere you don't know where it is. We want to make sure that we can be able to find it. And the other thing that the Biorepository Core does is we provide pathology services to the cores that need it. So we are providing pathology services to Project 5, Dr. Colon Otera's core with vitamin D. And the other thing that we are doing is we are cataloging what we have here at Mayo Clinic in terms of Black prostate cancer patients. So I had a researcher from an international researcher who is a board certified pathologist in her country of China. She asked if she could do a research project, so I had her come here. I had a list of all the Black prostate cancer patients at Mayo that were consented. And what we did was I had her annotate all of our digital slides so that way we can make a prostate cancer tissue microarray. I don't know, does this have a laser? Oh, here. So each one of these samples represents one sample from a prostate cancer from a patient that was receiving treatment here at Mayo Clinic. Most of these come from resections. Each piece of tissue represents a patient. And that way, if you wanted to do any kind of large study where you need to do some molecular information, you can just cut off of this block and make a slide. And here's an example of what the slide will look like. So each one of these samples is a prostate cancer patient. I wish I could zoom in because I did tune in earlier and I saw the team that talked about AI and prostate cancer. Can somebody raise their hand who did that? Oh, okay. Well, I'll catch them afterwards. But what we have here is all these histologic samples on one slide. This could be used to train any AI model. And we do have experience with training AI models here at Mayo Clinic, Florida. We do have a platform that we've developed in-house that we could make external. But having compact samples like these would allow for many collaborations. And we've come up with a novel way of making these through digital pathology. I do plan on publishing our findings in a journal just because it was so novel. Oh, yes. Hello, sir. I saw your project about prostate AI, and I just wanted to, I was talking about our prostate tissue microarray. So each one of these samples represents a sample from a Black prostate cancer patient. And we have 3 slides that look just like this. There's a histologic sample on each slide, detailed morphologic information. We can that's scanned in. We can share that with you internationally. If you have any AI platform, we have one here we could collaborate with. But if you have one, we could provide a sample. We have 70 patients right here. We're continuing to grow this, and this would not have been possible without the iCCaRE DRM Developmental Research Protocol Grant. So with each one of these costs $1,000 to make. I plan on applying again this year for more funding, and we can continue to make cores. If anybody if any university would like us to make one, we could do that as well. But this will lead to future collaboration with wonderful projects that I've heard at this conference so far. Thank you.
Do we want to do the pre-recorded video? Okay. We have one pre-recorded video from the REACT course of Research, Education, and Training.
Hello, everyone. I am Dr. Catherine Oladoyinbo, and I am the co-lead for the Research, Education, and Training, the REACT core, and I'll be making a presentation on behalf of the members of REACT. This slide here shows the members of REACT. Myself and Dr. Ashley are the leads. Other members of the core includes Dr. Kwebe Bolajoko, Ms. Williams, Dr. Uju Aliche. Pastor McCall is our community scientist, while Professor Folakemi Odedino is our MPI liaison for the REACT core. Now, the REACT core has 2 training programs, and the main objective of the REACT core is to facilitate the development of well-trained early career investigators and community advocates who can effectively address cancer control, cancer prevention, and survivorship. And so to achieve this objective, we have 2 major training programs, and that includes the advocacy training program for prostate cancer survivors and other members of the community who are willing to serve as prostate cancer advocates in their communities. And also we have the training of the early stage investigators through the Global Oncology Certificate Program. So in our year 1 of the Research, Education, and Training program, we were able to train 7 prostate cancer advocates in the science of prostate cancer advocacy. We funded 4 pilot projects for the prostate cancer survivors, which they carried out in their different communities. They implemented these projects. And during the SUPCA conference last year, September, we had 20 of our prostate cancer advocates who participated in the conference. 2 of those advocates who were trained submitted an abstract. The abstract was accepted, and they made a presentation during the SUPCA. During the SUPCA conference also, we had a town hall meeting where we invited 50 community members from the church and from the mosque who also participated during the SUPCA conference. Also in our year 1, we selected 15 early stage investigators who went through the Global Oncology Certificate Program. These 15 ESIs were selected from 6 countries, and they completed 12 graduate credit hours. The ESI also completed a mentorship research program with the assigned mentors, and we had a completion rate of about 84.46% among this ESI. The picture here shows some of the participants of our early stage investigators who participated in the certificate program. After the completion of the program, we had some evaluation where the students were evaluated by faculty members, and also we had the students evaluating the faculty, also evaluating the course, and also we did an assessment on how they are going to use the skill and the knowledge that they have gained from this Global Oncology Certificate program. We also are currently doing some indirect evaluation, looking at in year 2, the program enrollment and growth of the program. We're also looking at faculty and student honors. We're trying to check out how many of our students are receiving awards due to the training that they received from the Global Oncology Certificate program. We want to look at the number of manuscripts that have been submitted, those that have been accepted, or those that have been published. We also do some exit interviews with some of our early stage investigators, and we also want to do some student employer satisfaction assessments and also do some placement data assessment. So for cohort 1, which we have concluded, some of the students are rounding up their projects with their mentors. We're going to have a graduation ceremony on the 11th of May, and we want to use this opportunity to invite all of us to this graduation ceremony. It's going to be a beautiful one. And the link to the graduation will be sent out very soon, and everyone is invited to join us in the graduation. So for the year 2, we received 60 applications for from 12 countries, and all these applications are presently under review. And in our year 2 also, we have recruited a cohort of another 50 prostate cancer survivors who are going to go through the prostate cancer advocacy training for cohort 2. Also in year 2, we had the 4 early stage investigators research retreat was held, and this was led by Professor Folakemi Odedino in January 16th of this year. We also had a clinician ESI research collaboration interactive meeting with the Mayo Clinic cancer director. 4 ESIs also participated in this. This happened on the 24th of March, 2026. And currently, another program that is ongoing under the REIT is the iCCaRE sponsored grant writing experience cohort, where we have early stage investigators going through a grant writing process. It's a 6 weeks grant writing process, and this is currently ongoing. So these are some of the programs that we have been able to achieve through the under the research education and training program. I want to thank you for your attention.
That brings us to the end of our presentation for our cores. What I think we're gonna do, you wanna do question and answer? Okay, we're gonna do 10 minutes of question and answer, then we're gonna take a break.
Yes, another round of applause for our presenters. Yeah. So we can see from the presentations that the cores are not just a backdrop in prostate cancer research. They are actually the engine. They help to determine our scientific advances in prostate. from ensuring that we have measures that are valid and reliable to making sure that the data that we collect are meaningfully interpreted to make sense, and then also for publication, getting us out into the world for people to see what we do in advancing prostate cancer. And so we'll take our questions now. Any questions? Okay.
Good afternoon, everyone. I'm Jocelyn Turner, and I first want to make a comment because in doing this work with Mayo, I am amazed at what I've learned from men. I had a gentleman that I reached out to about participating in the study with Dr. Cabran. Oh, did he slip out on me? Yeah, he slipped out on me, didn't he? With Dr. Cabran. And a gentleman called me on my way here this morning, Dr. Odetna, and he told me every one of his family members have died of some form of cancer. Just let that sink in. Mama, daddy, grandmothers, every sibling, every uncle, every aunt, every grandparent. And isn't that sad? He said of various forms of cancer. So I just wanted to mention that as a reminder to everyone that the work that we're doing is important and it must continue. And we must continue to have the conversations with people, even if they are not appropriate for the study that we're reaching out to, because he was not eligible, if you will, for this particular study with Dr. Cabran, but he is eligible for some study. Like I told him, like, where did your family come from? Is there what in the world happened in their family history? So I've always told people that when summer times are coming and people are planning family reunions, I tell people, make sure when you're planning your family reunions and doing your family trees that you write the disease next to the person's name that family members had when they died, because this is so important that families know that type of history, right? Okay, so that's my soapbox. Now my question for Dr. Young. Dr. Young, I noticed in your slide about the demographic questions that you were asking, it was noted that regarding educational attainment, most of the gentlemen did not answer that question. So my question to you is, are you going to rephrase that question so that they will answer it, and or the persons who are asking that question of them, give them some type of lead-in to indicate to the men why it is important that you know that answer? Thank you. Oh, I guess you need the microphone in order to answer the question.
Yeah, thank you. Thank you for that question. And I don't think that I had a slide that said they didn't answer that question. So somebody else may have had a slide like the, okay. But our data set that we have there, the spreadsheet that we have, is for investigators to use if they choose to ask the question that way. So there's some standardization. How they ask the question in the context of the question and in the context of their research question is up to them. We're just providing some standardized way of collecting that data. If it doesn't work, then yes, the investigator and the MAMS team can help figure out why that's not working and what modification what needs to happen. And a good example of that would be family income. That's a question. We've got a standardized question, but a lot of people don't answer a family income question for a lot of different reasons. And so one of the things I say is, why do you need that information? If you're just trying to categorize people as have resources or don't have resources, you really just need 2 categories. You don't need 10 categories and then a cut point. So it depends upon what your research question is and what your actual question is and why you need that information. And Absolutely, absolutely. And you know, in qualitative research, if we find that something's not working in the middle, we go ahead and change it anyway. So
Thank you so much. Questions? Dr. Border. Okay.
Hi there. I'm a clinical research coordinator and very excited to be here. I have a question about Vera. Is this something that just anybody can download that has a headset, or is it something that they would need to have special access for?
Thanks for the question. And again, the question was about Vera. Is it just something that anybody can download? So how we're trying to develop Vera as this virtual reality application, that first 3D part will require a headset. So the workflow, as Dr. Dina indicated, was first you connect to Vera through a mobile phone, for example. You go through a quick survey question, and once you finish that question, you're provided with a link. When you click that link will now lead you to be able to go through the Vera application. It's probably going to ask you to put on a headset, which probably already contains Vera installed. So as we're going through this first initial phase where we do the pragmatic trial to actually evaluate the efficacy, now the next plan is actually how do we build that product so that individual users can actually use. And the way I imagine it being in such a way that either it's covered by insurance or a newly diagnosed prostate cancer can have access where we give them that access. Once we give them that access, they have Vera, they can take Vera anywhere they go and use it anytime that they want. But you have to have it connected to a headset. That's it. Now for the 3D, or the 2D part, which is open for anybody, is basically you just open up a website and you have that access. But again, it's gonna require you logging in, which means we need to give you that approval. You have to create that account. So you go through the study, we create you an account, and then you can log into it. Just like you log into Netflix, you have to have an account. So once you have that account, you'll be able to now experience Vera. Thank you. Thanks for the question.
Thank you, Dr. Kufu.
Sorry, his response prompted another question for me. So regarding the virtual humans, do you find that people are receptive to it and does it matter by gender?
Excellent question. Excellent question. So the meta-humans, this was actually something that me and our external contractors, we weren't sure of. We weren't sure of how like users are going to relate to those meta-humans. But from the aspect of the gaming industry, we know kids that use the games. It's such a popular thing, something that is part of the world now. Maybe the older folks are not really related to game, but eventually the new folks that are all into game, they're gonna grow old. And this is part of what they do today, they play games. So, but one thing that made us realize that this is it was our first initial visual. I think I had it on my slide. I was actually planning to talk about that, but I get carried away by some other thing. The first visual when you have Senator Tony here, Mr. Merriweather, they all sat. That was the first visual that was done with such high level of quality. We have to bring it down because we need to upload it to the headset. So we reduce the quality. But that first one, once we saw it, even within the research group, everybody had goosebumps. We had goosebumps from that image. Just the image. It's not even actually experiencing it. It's just the image. Just the image of the men, how they were characterized and all that. And then when you play the video, the quality of the video, and that was it. We said that was it. So again, this was a group of researchers that involved both men and women, and everybody cried. You saw that first video. Could you, what was your experience? That first video that we saw?
Oh, having been on this journey with Dr. Adinna for a number of years, I started crying. And it was very emotional.
That was my experience. So and that's from where we saw that this is it. And again, I think even from research where we are not cancer patients, but from us seeing that, and I can imagine how prostate cancer patients seeing that and going through those stories, it just goes back to that story. How can we capture their experiences and their journey in a way that can relate to others? And then the AI is a part that is jungling around, providing that data to them in a much more consumable fashion.
I just want to add to that. I think that the success of Vera is built on the stories that we got in the original research that we did in 2014, where we did qualitative interviews that were very lengthy and very in-depth. And so Dr. Ngufo had that information to work with, and I worked with him when he was doing the video for the Vira with the men to make sure that the things that we learned from those in-depth interviews and the stories that people talk about, that's what we're getting. And those real stories were then captured in Vira. So it's not just that it's an artificial environment. It's like these are real people telling real stories. And it comes across that way, I think.
Thank you so much.
Any more questions? Thank you, Dr. Ngufo. Thank you. Okay, a round of applause for all the presenters.
Break time. Let's take a 10-minute break and come back, and we're going to push through with the rest of our presentations. Before you all get up, start stretching and moving out, moving around, we have another QR code that we hope you all go ahead and scan and complete our planning and evaluation core survey.
They just gonna pass the handheld around.
Huh?
They just gonna pass the handheld around.
Yeah. So we need one mic, and then we'll have one mic. Yeah, how many mics y'all got?
I got 4 wireless total. Do we need some in the crowd too, or?
No, it'll be up here probably.
Okay, so you're gonna have 4 up here. Do you want the moderator on this side so they can use the podium?
Oh, we're gonna have them sit down. The panel. Jada said only Meriwether and Gaines, but we got Misty. Well, you still got the Rotimi, Bolojoko, and yeah. So we're not doing the last panel then? Let's do a plan for it because she needs to see. Well, I tried to. Yeah. They were doing the advocate training. They I tried each person that came in. I tried to talk about it, but they really go to their go. My dad. Yeah. Mr. Sammy, I think he went to but she said she was gonna come, but she might not be going to so she said she wanted to go through half days.
Okay, everybody, we are gonna go ahead and get started. Save your conversations, write them down. I know it's a lot going on over there. Okay, everybody, let's sit down, let's sit down, let's sit down. Is anybody out? No. Okay. Jeffrey, you ready? Is there anybody that need to be in the session? Is there anybody down there that need to be in the session? Okay. Okay. Yeah. Look. Oh. All right. Okay, everybody, we are getting ready to get started back. Okay, everybody, let's take a seat. Let's take a seat. We are getting ready to get started with our next panel discussion. It's going to be on clinical trials and translational readiness forum. And the theme is moving interventions into multinational trials. We have Dr. Bolajoko, we have Dr. Rotimi, we have Dr. Williams, as well as Dr. Odedna. I think she'll be moderating, but she'll be inputting. Hey, hello, how you doing? And we have former Senator Tony Hill.
I think Dr. Rotimi has left us for a meeting. It's okay.
Thank you.
Question about the lights.
Can those lights be turned down some?
Yeah, they're too bright. Yes.
Right in the eye. Give me some shades.
Oh, they need them? Yeah, they need it for the camera.
They gotta keep them on? Okay. We'll squint.
Okay.
Oh,
okay,
man.
great. Yeah, we need black, so that we can look black on screen.
Start.
Are you ready? Okay, so welcome to the panel that focuses on what prevents global prostate cancer trials and how do we solve it. Today's panel focuses on a very, very critical question. And despite the advances that we have in science and the advances that we have in discovery and implementation, participating in clinical trials is actually very uneven. It's not only uneven in underrepresented communities, it's very uneven also globally. And what I mean when I say globally, I also mean in the United States as well. So global is not just international, global is within the United States and outside the United States. So we are joined by a very, very unique and diverse panel, starting from who I would call is the most important. We have an advocate and survivor of former Senator Tony Hill, and is known, Senator Tony Hill for a long, long time. We've traveled all over the world together, doing advocacy, Egypt, Morocco, and everywhere in between. So welcome, former Senator Tony Hill. And also next to him is another person that I've known for decades and who really has been a true champion in, when we say underrepresentation in clinical trials, he knows how to make sure there's representation in clinical trials. So Christopher Williams, and then next to him, representing, so we have the advocate, we have the clinician representing the science, is Dr. Paul for having one half a decade, which is still young. But she brings unique expertise doing research in Africa as well, doing research here as well. I know that, so I'm going to start the question. Dr. Christopher Williams was trying to run away from me, so I'm going to have the question from you. So, from your clinical experience, what do you think are the biggest barriers to enrolling diverse patients into prostate cancer clinical trials? And you would know this.
All right. Well, I'm glad you said that, this is global, really encompasses the United States as well, because I was kind of preparing for like international stuff. But I think anytime you're talking about underrepresented minorities in clinical trials, you have to go back to kind of a mistrust of the medical institution. And we were in the training yesterday with some of the survivors who are going to be advocates, and one of them mentioned that many times people use that don't really understand what happened. It was so long ago, and a lot of those things that happened while they were atrocious, there have been laws and things put into place now to prevent those things from happening. But still, the mistrust is there, so that's some part of it. So I think that's one thing that really can make it tough. Also, I think that we don't have enough minorities in medicine, and seeing somebody who looks like you does make a difference. There's just no way around that. Qualifications are definitely important, but there definitely seems to be a letting down of the guard when somebody sees somebody that looks like them, and they automatically feel like they can trust them a little bit better. And so representation there, I think, helps, and not having the representation makes it more difficult to enroll. Another big problem is access, especially for clinical trials, because with clinical trials, there's so many parameters that have to be met. There are time constraints, et cetera, and all those things are great, but people have jobs to go to. And so if they have a job to go to, they go to their job. So a clinical trial is not seen as something that's a necessity. It's more of a luxury. We'd like for you to participate in this, sir, but we do understand you have other things that you have to do. Also, there are opportunity costs too, because if they participate in the trial, then they're not at work, sometimes, so that's a barrier in and of itself. Also, it's another visit to come to as well. And with that, even if they're not getting charged for the visit necessarily, they're still putting gas in the car, so there's just costs that are important and that are part of the reason why we have low participation sometimes. But those are just a few things. I could go on and on, but I want to let somebody else talk.
Yeah, Thank you so much, Dr. Lawrence. I'm going to throw it to Senator Hill. We have had the perspective from the clinician perspective. What is your perspective? What do you think are the biggest reasons that patients hesitate to participate in clinical trials?
Well, first of all, I would say to you and say to the audience that clinical trials are so important because when you get into a clinical trial and you go into the pharmacy side of the house, then the trial would help the pharmacists or the pharmaceutical companies come up with the right type of drugs for you to take. Because a lot of times we get the drugs that the majority community has, and we don't have the drugs. So I would say to the audience that that's one. The other is opportunities. We don't have the opportunities that others have to get into a clinical trial. Normally, when we are diagnosed with prostate cancer, we're just trying to figure out how to survive. We get into a mode there, and I tell Dr. Odetta all the time, when I was in the legislature, she approached me because she was on the faculty at FAMU and said, we need to do something about this. And Dr. Williams, I will let you know that I did not realize that in 2019 that I'd be diagnosed with prostate cancer. So this goes back to what you're saying. But I knew what to do because I was trying to get the legislation passed, because I'm telling you right now, women are so far in front of us as it relates to this issue around breast cancer, that when they go to a primary care doctor, that's the first thing that comes off that list, that checklist they have. And so, but again, clinical trials are so important because then you can have the right prescription that is compatible to your situation that they could take. You know, it doesn't have to be from a majority community or another community, but it'll be coming because you were involved in the clinical trial, if that makes sense.
Absolutely. And as we go to the next question to our analytic investigator who has had a chance to do research in both in Africa as well as in the United States. If you have questions, please get your questions ready. This is not the spectator panel, so we want you to ask questions and we want you to contribute as well. But I'm going to ask Dr. Bolajeko, as an early-stage investigator, what challenges do you face when trying to engage in global prostate cancer trials?
I think the first thing, thank you for that question, would be access. You know, access in the fact that as an early-stage investigator, I would say access and trust. Nobody will trust an early-stage investigator. And when the conversation is being had, sometimes the early-stage investigators might not be there. So we need to ride on the back of a senior investigator who will do the introduction and say, oh, this person can represent me. Or I want her at the table, even if it means just listening. So that's good. So that, I know that for me and for other analytic investigators, to even have that person that knows the inadequacy and will say, okay, you can use my list, you can use something, and use you to contract. And that is a big one, because even with some of our colleagues in Africa, and like even with some of our senior investigators, the younger ones, even clinicians, need the same access to be part of the consortium that is being created, to be called to be part of that discussion, and also to have their sites included in the trial.
Yeah, it is interesting. We are hearing almost the same words, but with the same different meaning. We are already pairing access, providing the access through the clinic. Senator Hill is talking about access for the patient. You are talking about access for the senior investigators to help you access and fish down the bush, which is quite interesting. I'm going to go back to Senator Hill. How can survivors like yourself play a stronger role in shaping and promoting clinical trials? I mean, you have been a champion. You have been a champion in policy on the Senate. You are not stopped, and we thank God for your life. Even before you were an advocate, and after you're still a very strong advocate. So, what do you feel the role of survivors have, especially related to clinical trials, and how can you make us?
Well, I think, Doc, that when we're moving in these spaces, that we brand ourselves, and I don't run away from the word prostate cancer. I'm a prostate cancer survivor. And you want to use that platform, just as when you came to me to ask me to engage in this conversation around policy, right? And so I would say to you that those of us who have gone through this, I didn't have a trial per se that I went through, but I would tell folks that clinical trials are so important. The data is so important, right? I would even say, even at family reunions that we have, Josh brought up something that I've started using, is that when someone passes away, note what they passed away from. And a lot of times I run across people and I tell them, when is the last time you had your PSA check? And they said, man, that's interesting you said that, because my wife asked me that the other day. Hello. I didn't say J-Lo now, I said hello. And so I think, Doc, that the more we're involved in settings like this, the more that we're involved with Johnson, putting it out in the community. She has a podcast. I mean, we need to have, Dr. Willis, a day where we dedicate that to prostate cancer awareness, i.e., a run, or what's the new game now that they play? It's not the tennis. What is the other? Pickleball. So, I mean, our fraternity, which I'm a member of, we did something over at the park on Moncrief. And we did the pickleball, and we used that as a way of educating the guys in the fraternity. Now, Dr. Willis, we have moved it now to the Divine Nine, where we can have a conversation around that, maybe a softball game, Johnson, that we could do, that we can educate the public. Because most people, no pun intended, they still in the closet on this issue. And a lot of times we get in an argument, Dr. Odette, over 2 C's when we have family reunions. Dr. Williams, we get in an argument with 2 C's, right? How many people in this room know what the 2 C's are when we have family reunions? That's one. That's one. That's one. We get in an argument, Jocelyn, over 2 C's. She got the first one right. The other one is a cake. Who's gonna bring the cake and who's gonna bring the chicken? But nobody talks about why Big Daddy is not here this year. Hello? They don't even talk about that. We know he's been here. We saw him last year. We saw him year before last. He's telling all the jokes. but we got to make sure that when we having these family reunions, that we talk about the health side of it, right? Not get into an argument about the chicken and the cake. I'm on, Dr. Willis, I'm gonna stop right there because I don't get excited. But that's the thing that galvanizes us as a people.
Before we go to that, because every time I'm listening, I'm thinking of what can iCCaRE do, and I think this most probably will go to the test. So maybe iCCaRE can develop a family reunion toolkit that is available, that anybody can get those toolkits and take it as a part to their family reunion.
Hello.
It serves as a guide to help them start the conversation.
Hello.
So maybe you are part of PES too, Senator Hill. So just say that. Maybe you can help us lead that. Maybe that would be. We have a couple of questions. I know one is from Dr. Kaniji first. While we are going to go, Dr. Kaniji, I want Dr. Williams to answer one question. Yesterday, we got a very prestigious award. In fact, that is the first award that we're giving to a clinician for facilitating research for underrepresented minorities, because you do it so well and you do it so seamlessly. You make it look easy. I don't know how you do it. So can you tell us how can clinicians better integrate clinical trials into routine care pathways based on what you do in your practice? And your practice is so important because it's right there within the community, and you see a lot of populations that are marginalized and most probably are underrepresented. What is your formula? Again, we're trying to figure out what's your formula before we go to the question.
Well, I always tell people that, you just have to love the people first and foremost. If you love the people, you have to love the people enough to, enter into those difficult conversations and don't take no for an answer initially, not without educating the patient. Because I had men come in and, we broached the subject of getting your prostate checked, and they said, well, I was at another doctor and I told him I didn't want to do that, and he just let me go. And I said, well, you're not gonna leave today. I said, we're gonna talk about it first. You need to understand why this is important. Then if you decide you don't want to do it, fine, but we're gonna educate you first. And then, once they see that you care and you're trying to help them, and they understand the implications of screening or not getting screened, then they're more likely to allow you to screen them for their benefit. So that's one thing, just showing that you care about people, and that you're not just checking a box just to say you did it, or say that you told them about it and then they didn't want to do it, fine, let them go. You can't be like that. So it takes a little time. It's going to add some time to your visit to be able to have those conversations. I think that's the kind of the foundation of it, is that. And then as it pertains to clinical trials, though, obviously you have to know about them. And clinical trials typically are designed to answer like an unmet need or a question that's out, in medicine, or there's something that's not working that well. Can we address it somehow? That's what clinical trials are designed to kind of address. And many times the reason why people aren't told about them initially, like when Dr. Hill, Senator Hill was talking about, didn't hear about that initially, because it may not pertain to everybody. Many times the clinical trials are for when everything else has failed. And now what else is there that we might try? It might not be something that's proven, but it's something that we're trying to figure out if it will work. And then those typically are, big trials by big pharma in some cases. Sometimes they're by big institutions or collaboratives between institutions. And those can be a little bit more challenging to get somebody enrolled in than smaller trials, because you might not have a contact, for the iCCaRE things. Well, obviously I got a contact, I'm texting Dr. Odedno or, Jada or anybody that I know in iCCaRE. I got a patient who, fits the bill for this study. How do we get them enrolled, and how do we get in touch with them? And, so it just requires an extra effort. But obviously you have to know that the trials exist. And many times I think for like the big pharma type trials and things of that sort, you can go to like cancer.gov and look them up, but then there are 100 of them at least. And, but again, those are primarily for situations where the standard of care has not worked. And now what else is there that we can try? And then it can be kind of tough to figure out, what they might, what trial might be the best one for them to go into. So that's a challenge for the average clinician, because we just don't know all of these trials that are out there. And then to go searching through cancer.gov takes so much time. I think it's easier at a bigger institution because they might actually be sponsoring the trial, like a Mayo. They might have a trial, that's going on, and then they're trying to fill that trial. So a lot of different barriers there, but bottom line in terms of my success in helping iCCaRE enroll is because I love the patients, and I have access to iCCaRE to be able to get people enrolled.
Let me hitchhike on that thought with Doc about caring for people. A lot of times, Josh, and people don't care how much you know till they know how much you care. Hello?
And I just want to, we're going to go to the question now, but I just want to emphasize, one of the things that warms my heart is that what Dr. Williams is saying is just not saying for what, it truly does care. Because I've gotten calls in which we have an appointment to do a study with a patient, and he's saying, the patient is here, it's 5 minutes over, where is the clinical coordinator? And he messaged me. I have never seen a clinician that cares that much, that is showing 5 minutes late, okay, although the coordinator was with another of his patients, but he cares so much, like he follows everything up to make sure that those patients, so it's just not saying it, it's truly, and he knows when I see, I don't care where I am, when I see Dr. Williams, that's like, okay, we are. So I think what he said is so important. His care, his heart is on it, but having access to, also you have to have the access to the coordinators, to the PR, to the, where you are recruiting. We have a question?
Yes, ma'am. Dr. Odedno, I'm Carlise with the People and Blue Sox Foundation. I'm going to piggyback off of what you mentioned about a family reunion. We are in the process of having a pickleball fundraiser actually at the end of September. So since Senator Hill mentioned family reunion, you backed it up. So why don't we take our event and turn it into a free event and do education class instead, but still have it be pickleball if we can get someone to fund it. That way we get more people out, just like we were able to do in March at the symposium, and we were able to screen 21, PSA 21 on the 14th of March. So just an idea.
The idea goes directly to the person behind you, Dr. Floyd Willis, who is the chair of PES, and Mr. Merriweather. So Dr. Willis, that's why he's here. He got it.
Well, great conversation so far. We've talked a little bit about addressing some of the concerns that the populations that are disproportionately represented, why they should be participating. So let's talk a little bit about the other side of the equation, which is the pharmaceutical companies that may be behind some of these drugs. There are linking suspicions within these communities, and I hear that all the time. So what ammunition do we have to allay their fears that, yes, participating in this will not be taken as a guinea pig as it happened historically. But there is some lingering concerns that, unlike the physicians that they know that they care about them, some of the pharmaceutical companies may not quite have their interest at heart and will take advantage of them. How do we try to address some of those concerns when we talk to people in the community and that comes up?
So let me hit your hike on your thought there, Doc. My daughter was trying to get a position with a pharmaceutical company, and the person that was doing the interview did not feel that she brought enough to the table. So I would submit to you that the doctors that are receiving this information from the pharmaceutical company needs to raise that issue, all right? So she expressed that to me, Dr. Willis, and had she not had the conversation with that pharmaceutical company, because she's already working for another company, so they're moving back to Atlanta from Tennessee. And so the person that got her the job there is now working at another pharmaceutical company. So he threw a name in the hat. But that is one of the barriers that we have as it relates to advocacy from the pharmaceutical companies. They feel comfortable around hiring people that sometimes maybe have experience, but as she was telling me, that how am I going to get the experience if you don't hire me? All right, and she's very active in her sorority, so she brought a little bit more to the table in terms of them trying to do what they need to do in representing that pharmaceutical company. But Doc, that is one of the barriers that we have. But a lot of times we in iCCaRE, we might even want to have a meeting, Dr. Willis, with other Black physicians that folks come in to drop off their drugs and stuff to them, that we have to have a little bit more diversity, right? Because you see who my clients are, you see who the patients are, and so you know who that I'm dealing with as it relates to being a physician. So that would be my take on it in terms of the pharmaceutical companies, that if we're not pushing it and we're just receiving this, the drugs or whatever that they're bringing into the office, and we're not questioning the diversity of the folks, then that runs into a barrier for our community. And a lot of times our community lose out, because a lot of times those, like my daughter and some others, go to churches that are predominantly African-American churches. So it's a lot that they can bring to the table if given the opportunity. If that answers your question.
So I think that your question really was kind of really wanting to know, like, at the point of care, how you could address that. And I think that education would be important. You certainly can't lay away decades and decades of fear, but you can help some. You can talk about how, the things that happened in the past, no longer happen because of laws that are in place and things of that sort. But the other thing that I mentioned earlier too, most of the time the actual clinical trials are in place to answer a lingering question in medicine. The standard of care has not worked. So if you're offering a clinical trial, then you have to help the person understand, we tried everything else. And so now we're looking at what else could we try? We're not trying to make a guinea pig out of you, but there is something that's, being considered that may help you. We're not sure, we're not certain, but it may help you. My sister had stage 4 non-Hodgkin's lymphoma when I was at the NIH, and she was placed on the clinical trial. We knew that it was a trial, but it turned out that the drug that she was getting was rituximab. So she was one of the first people to get rituximab. It nearly killed her, but she's still alive 25, 26 years later. So, you just have to let them know that we've kind of come to the end of our rope with everything that normally works. And this is an opportunity to keep trying rather than just giving up.
Everybody knows all the students around Tuskegee. And I had a classmate that he actually works at the NIH now. He was really instrumental in the development of one of the co-curriculums. And in Alabama, of course, you have a lot of stigma about, whether or not you want to use a particular drug. Medical mistrust is huge because of Tuskegee. What he did was he did virtual town halls. He got in contact with young people in these communities, set up these virtual town halls for older people to mitigate all these myths that, okay, you know what, this is, Tuskegee around too. And that was one of the things he was really trying to mitigate, just completely kill, that myth and explain to them in the best language terms that you could the development of the drug and why taking it is so important to them. So I think just coming up with alternative ways to reach your target audience, I think that's going to be, these are important. So we don't want it to, have a barrier because we don't figure out what alternative ways to reach the population that we want.
Yeah. And as we're doing this also, I don't think we should think automatically people should trust, to be honest with you, because one of the things that scares me the most is to have this blanket trust and the person experiences something negative again. So we have to be careful how we plan to navigate people through this and make sure that we are navigating well, because each one of us could tell stories that we have also had in the healthcare system, despite how well educated we are. So I think in that sense, we that we are the one that is providing the guidance becomes the people that make sure that they are directed to places that are trusted and people that are trusted. And that's one of the things that we do very well within our program, and make sure that when people are being directed to, they're being directed to people that are not going to disappoint them. I know that you have a question. Is it related to this, or do you want me to? Okay.
So it's just a comment.
It's a comment regarding a study that I worked on. I can't remember which. I've done something with Mayo, but one of our community health workers in doing some engagement said that as she was talking, I'll never forget this thing down. And she was talking to about participating in clinical trials, was hesitant. And the person was going into the pharmacy to get some Tylenol because they had a headache. And so she told me that Tylenol was a part of the clinical trial. It's a drug that's very commonly used, and you don't think about it, right? And just by her saying that to that person at that moment, it convinced them, I better sign up for this, or put my name on the registry, if you will. So I believe two things that you all said, that it depends on who the messenger is that's having the conversation with the person, and you really have to find something about that person or that situation at that moment that would be relevant to that individual. And the conversation may vary from person to person, what you say, but based on the environment where she was at that moment, the person just happened to come up and was headed in the pharmacy, and she was asking the question, hey, can I talk to you about clinical trial registry, yada, yada, yada. And they said, no, I got a headache, I need to go and get this. Great, guess what? And she really put it together, and that person ended up being an ambassador, if you will, and telling other people after he went in and got his Tylenol, hey, y'all need to talk to her because I have ideas. So I think it really is relevant to who the messenger is and the message that you're relaying to the individual at that moment.
Yeah, absolutely. Well said, Ms. Turner. And it was the Community Research Registry. I think it was the early phase of the Community Research Registry that we are now calling the iCCaRE because we had this in the pharmacy with your support and you're one of the people. So that's good. We're going to move on to Maymoon. We have about 10 more minutes. So I want to start with Dr. Blanchard. Just talk to us a little bit on what would help you to contribute more effectively to global and multidisciplinary research efforts. If you're an early stage investigator, you're trying to be down the path, and you're trying to be the best of the best, what do you think would help you?
Yeah, one big one is mentoring. To be able to to effectively work in that space, I need a mentor who is in that space, who would be able to oversight what I'm doing and guide, because we cannot overemphasize the role of a mentor, because if I think I know what to do, there are certain decisions that if I'm able to articulate it to my supervisor or a mentor, he or she might say, no, don't go that route. Okay, I'm going to introduce you to someone. Oh, I did that in 2015 and it did not work. Then you know that no, don't do it this way. So really, I that's what I believe would work. And number two would be reviewers. Even before the grant goes out, people to help review. It's very difficult to have people look at whatever you have written. I know for me, I am privileged. But I know that some other early stage investigators may not have the kind of privilege that we have in our lab to have Dr. Dedi not look at everything. I mean, sometimes it's even in 24 hours. So reviewers, and like when I joined her lab, the first, I'll call it a thing. I would not want to call it a grant. The first thing I wrote and I sent to her, she laughed. She said, then she sat me down and told me the art, A-R-T, of writing, that you can have a great science, but writing that science is an art. And it's different. It's a skill that must be cultivated. And then I knew, then when she wants to send back our papers, she'll tell you, don't worry about the red stuff. But you should be grateful that even a computer is an electronic paper, that if it were written and you have to have everything in red pen all over, she shared her experience as well. So that's why I said to have mentors who can review, or other people. For example, we do have a lot of senior investigators in iCCaRE, and reviewing manuscripts, reviewing grants is key. So I would say that. I'll stop there.
Thank you. So we're going to go to Senator Hill. What message would you give to researchers that are designing trials about what truly matters to patients? Because a lot of times we want to make sure we are matching what patients really need.
Well, I think that if we continue to do advocacy in our communities, and we're serious about what we're trying to do in terms of prostate cancer, getting that out to people, whether it be op-eds in papers and stuff like that. I think that is another way to get that communication. But again, as Dr. B like to say, it goes back into mentoring, and it goes back to branding. If we can brand this out there like the women have done with breast cancer, I think that will take us to where we need to go. But as the gentleman said there, when he went into his primary care doctor, you can have my butt. But this is the thing that I find with guys is that they are scared of the finger, but now with the technology we have, you can have an MRI done. and the MRI would do the same thing that the finger would do, right? In terms of detecting detecting it. But I'm just saying, for those guys that are nervous about doing it, we need to get them in there some kind of way. So I just tell people most time, just get the MRI done, and sometime it will detect what's going on with your body. But just not going at all is not an option.
Williams, the last question for you, and we're going to take one more question from the floor. So if anybody has a burning question, it will be the last question from the floor. What practical changes would make it easier for urologists to refer patients to trials, not only nationally but also globally as well? We are in the age of decentralized clinical trials, and just kind of thinking about the practice of urology and the importance of referring prostate cancer patients. What are some of the things that you feel can be done to make people comfortable, to make urologists comfortable? And when I mean globally, I mean the urologists that are outside the US, referring, not you referring globally. I'm trying to think about, for you as a urologist, what will make you feel better to be able to refer patients for clinical trials?
Well, one thing that, like I said, is important is what kind of trial is for. Many of like the prostate cancer clinical trials are for when typical therapies have not worked, and now you're into a clinical trial because it's kind of the last resort, so to speak. And so those trials would be like on cancer.gov, and you can look them up. But the people who treat those diseases, like the medical oncologist, and primarily medical oncologists, they give chemo and things of that sort. Those are the people who would be enrolling in those types of trials. Urologists certainly can refer people to trials if they know. Again, there's so many trials out there for that setting. Metastatic prostate cancer, not responsive to hormone therapy, not responsive to local therapy, not responsive to immunotherapy. That it's just so many of them to know about. And I think that's a big barrier. I think that it definitely is true still, though, that people don't feel like they have the time to go that extra step and figure out what the next step would be for that patient, what would be the next best trial. But I think leaning on your cohorts who do that, like the medical oncologists who do those things, that's sometimes what I'll do. I'll say, hey, which trial is this person good for? You guys are sending people for trials all the time. This person has this, would they be a good fit? I think that would help for one thing. In terms of global things, there are a lot of barriers for global clinical trials. One of them being, many times, the pharmaceutical companies, they'll set up trials not where the disease burden is the highest, it's just where the infrastructure is the best. I mean, they're there to make it work. That's their goal, so if it's easy to conduct the trial, that's where they go. A lot of times clinical trials are in major medical centers. Internationally, you don't have as many as you probably have in the United States. And so if they're not parked at a academic medical center, then it's harder to get them into a trial too. So decentralizing it, like you said, would be very, very helpful. But many times they don't put in the budget to go out into the community and recruit, or to do the trial out in the community, conduct the imaging in the community, do the lab work in the community, all those kinds of things, because those things would really, really help to enroll. And even if they went out into the community to the individual doctors and made them aware of the trials that were ongoing, those things would help too, because being able to know what's available would also pull down that barrier for referral. So there are a lot of different things, but those are some that I can think of off the top of my head.
Yeah, thank you very much, Prof. Right now, the cost of doing the test is very expensive because the technology is imported. And so what we need to do is to train investigators in Africa or low resource settings to begin to develop the technologies locally. And the technology really on the surface will appear very complex, highfalutin, but the science behind it is not that complex, and it's something that can be domesticated. That's the approach that we have used to really cut down the price. And then the other part of it is that the cost of labor in the low resource setting is also not that high. And so that also cuts down the cost of doing this test. Give an example. I'm not sure how much immunohistochemistry is done in the US, but in Nigeria, it's less than 100,000 naira, which is less than maybe $50 or thereabout or so. So I remember talking with Dr. Matt Gates, and I mentioned how much it cost, and I was like, okay, really? And I said, yes, that's what it costs, because and the reason why it's even so is because the antibodies are often imported. So when you take that out of it, the cost of doing it is not expensive. So low resource settings need to own the science and the expertise. That is the more way to really bring down the price. Thank you.
So much. That is a great way for us to end this section. So thanks to the panelists, Senator Yield, Dr. Williams, Dr. Balaji, for
Yeah.
She only has one panelist. Yeah, the others. Just invite all the other ones.
Good evening, everyone. I have the privilege of moderating the panel for the most powerful people in this room.
I think
That's
visible,
the
like.
advocates. So join me to welcome Senator Tony Gill again to this panel. Let's be time together for me. I also want us to welcome Mr. Donald. Okay. I think all our advocates are not around.
We have a couple of advocates right here.
But we do have some advocates right here. I'd like to welcome Mr. Innocent Gugwe.
The one with Dr. Kanije.
Yeah, I don't think.
Sir, do you mind?
Oh, Mr. Quincy Jones.
Oh, are you going to sing for us? Okay, wait.
Please, let's put our hands together for Mr.
Today.
You're very welcome to this session, and I'm really honored and privileged to be encouraging this session. From the background of I'm an medicine investigator, I know that. And as a scientist, when I think about research, when I think about the community, maybe for some people, I think for investigators, we probably are looking at participants. You know, we're looking at participants, we're looking at co-investigators, we're also looking at NPIs. Like that's the kind of training I have received. And I have worked with My PI have worked with study coordinators in the community. I've worked with community investigators as well to see how even the science is, how we even develop research questions. You know, earlier today, Dr. Dedina was sharing how studying prostate cancer, and the question actually came from a survivor who asked that, what is the link between, the transatlantic trade? If the blacks in Africa have this and we are here in the US, is there any science? Is there any link? And at that time, she was not able to answer the question. But now today, she could answer that question. And she decided to answer the question. So my question to you, I'm going to give you all the opportunity to answer, is what does research impact? When we say research impact, what does that mean to you? Or what would you like to say about it? Or what comes to your mind when we say what is research impact? So I'm going to start with
That's a very tough question. And being a prostate cancer survivor, you want to know, how did you get it? What was in your background? What was in your family history, your DNA?
We can hear you.
What's in your DNA that would cause you to get it? And we talked about the clinical trials earlier. I was going through a medical procedure, and one of the researchers came up to me. I'm laying on the table getting ready to go in for surgery, and asked me to participate in a clinical trial. And I think we ought to explain more about what does that mean. Because you initially probably think they want your blood. And I found out it's not always about the blood. It's a lot of questions, answers, information, and that probably needs to be explained more. But I'm a part of a clinical trial because I want to help now. So the fear of, oh, you want my blood, whatever, it doesn't matter to me now because it's going to help me. It's going to help other people. I had, they used my blood to run against 70 different cancers. And came back and told me that I carried the pathogenic mutation that causes cancer. And I never knew that until I turned 66 years old and finding out that I've been carrying a mutation in me that causes cancer and never knew that. It's in my blood, and they told me to go tell all your family members to get tested because they may carry that mutation as well. So the research, the impact of it, getting that feedback to come back and say, you need to be tested on a routine basis for pancreatic cancer. So you get, the information comes back to you first. What am I carrying around inside of me? And if that's all part of the research, it's going to impact you first, and then you can share it with your family members, my son, my brothers, my sisters. You all need to get tested because a lot of times we don't think that cousin got cancer. I don't have cancer. But you're not thinking, wait a minute, we're from the same bloodline. So the more research and information you can find out about your family, I think that idea about asking those questions at the family reunion, I got a couple of them coming up. So we do need that kind of packet that we can share with the family and noting what did grandmother die from? What did mama die from? All that secretive stuff, we gotta move past that within the family and talk and be more open and honest about what's going on inside of us. And that research starts within your own family by you going back, researching your grandparents, great aunts, uncles, and all of that and tying it all.
Ditto.
Thank you very much. Yes, Senator.
Yeah, I think, yes, I think that research opens much more pathways to learn about um, the prostate cancer. I came to this conference. It was here that I learned that PSA is not what you should be looking at when you are getting a prostate cancer diagnosis. That was what I thought. I learned that the Gleason score was what drives the prostate cancer treatment. So if we have research, then we will be advancing on some of these other guidelines that we have been using to treat prostate cancer. So that's why I think research is very, very important, because anytime you get involved in the research, some other things will come up that will override whatever was done before. So I think research is very, 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 very important.
Thank you very much for your contribution. And my other question would be that as an advocate, how would you or how do you want to contribute to research? Like for Mr. Quincy Jones and Mr. Innocent, this is the first time attending this type of advocacy training. So from what we talked about yesterday and what you have experienced, including the dance. Yes, the family, the iCCaRE family. How do you think you're going to, how would you contribute and make an impact in research as an advocate and as a survivor? I'd like to start with you.
Yes. Having been here for 2 days now, I come from the Nigerian community in this Jacksonville. I happen to be a director in the association. So I'm going to go back to the association and explain exactly to them what I have learned here, so that they can remove all those fears about if you are going in for clinical research or trials, or you are going to be used as a guinea pig. Somebody is going to get your information and it will go all over the place. They are going to get all your information and all that. So I'm going to allay all those fears, so that if there is a research that is coming up, and I know, then I will be able to go to the Nigerian Association, go to the African associations in Jacksonville and say, "Hi guys, we need to participate in this research." Yeah.
Yes, I know you've been doing this before. Would you like to contribute on other means of contributing as an advocate to research, since we have advocates here and we do have some of us early stage investigators here?
So I would say to you that with the different Divine Nine groups, and then you got the Masonic family, but being a prostate cancer survivor is letting people know in the community, even at the churches that we attend, that when they have health fairs, that we put prostate cancer at the top, right? And that we join in with other groups, advocacy groups there that are doing stuff around that, around the issue of prostate cancer. But the key thing is that when you see someone and you know them as a friend, the conversation has to be centered around, when is the last time you had your PSA checked? That has to be a key thing that we need to put in our vernacular or jotting words that we use when we're in the community, especially if you're in a leadership role. I mean, even when you're invited to speak somewhere, you need to let people know after they do the introduction and stuff that I'm a prostate cancer survivor.
Thank you very much. And sir, I'd like you to answer that question as well.
Getting out and talking to the community, we can't be afraid to talk about this subject anymore. The men. Once you face death, so to speak, there is no more fear of anything. This is the easy part for me talking about this issue. The hard part is going through some of these medical procedures and the pain you have to go through after. But I don't even fear that anymore. I'm just willing to submit myself to whatever needs to happen for me to get healthier. Is this procedure gonna make it better for me? Talking about it is not gonna hurt. Is it gonna make me better? Yes, it is. And so I'm willing to talk about it, share my experience, the deep, dark, the ugly side, the side that you would talk about in public that people would never even think about talking about. I sat next to a man yesterday, had one of the same procedures I said, and told me he was in a deep, dark place for a long time and couldn't talk to anybody about what he had experienced. But he talked to me yesterday about it because I went through the same thing. So we gotta open up, we gotta share it. I got a son that I talked to about it. He's young, 22 years old, and he cringed, "Oh, Dad, I don't want to hear all of that." Just like that. I say, you got to get over it, son, because guess what? You could be experiencing this in the future. And I'm trying to share with you what I'm going through. So you got to toughen it up, be strong, and tell a story to everybody that want to listen.
Thank you very much, sir. And to the audience, you can actually ask them any question while I continue to ask my questions. Okay, Dr. Ledina. Okay, before we take Dr. Ledina's question, I've been privileged to work with advocates and survivors. And one thing that resonated with me was the fact that even though as a scientist, I see percentages, I see 1 in 6 will be diagnosed. But these are people. These are not just percentages. Even though we put everything, we summarize sometimes 3 years of research with just one table and a figure. And that's the summary of everything. But the truth is, these are people, these are people's parents, brothers, uncles, and I remind myself that these are people that we're talking about, and that our research, my research, should impact people, and that there should be a kind of human face in whatever I do. And that's why I'm very excited when we're having your input in what we want to do. So this year, I'm also privileged to be part of the Partnership and Engagement Service Corps. How do you think, as an engagement, iCCaRE outreaches, how do you think we can improve our impact in the community? How do you think, as iCCaRE, we'll be able to improve our impact? We're not just going to be talking about numbers, science, but that something that is translatable to our community. That will be, I would like to call it low-hanging fruit this year.
It depends on what kind of study we want to get into. if it's something like prostate cancer that we know affects the Black community.
We do prostate cancer in iCCaRE.
Yeah,
Prostate cancer.
okay. So I think the best thing would be to reach the community that is supposed to be affected by whatever study that you want to go into, because that's the only way that it can be spread. That's the only way you can remove all the myths. That's the only way you can remove all the mistruths and convince them to be able to get into whatever you want to do, the research, the trials.
Yeah, thank you. So Dr. Devin, can you? Okay, sir.
Yeah, this is such an interesting topic, and what I want, my question is, this is something that has really started about me for a while, and I really want to get the patient perspective on this aspect of research. And Dr. Blanton, just picking up the point, I work with data, and I know that that data is real, but that same way, it's not iCCaRE, another way as well. We push, so it is your data, and then that data will be with you. But sometimes what research doesn't really come out with what we expect. We saw earlier today in the vitamin D study, and I think we saw Dr. Johnson presented a slide that shows some sort of conflicting results about vitamin D. One study, the clinical trial shows one result, and another clinical trial shows opposite result. But so I really want to get from the patient perspective, what does that mean? How do we translate that? Again, we also talk about the iCCaRE package, family package. How will you translate something like that during that family year? Well, this research has come up, shows something great, and another research, the same thing comes up with, no, that's not. But it's research. And again, recently we did a study where, I built AI models and study that conducted a clinical trial on, oral anticoagulants. These are drugs that can actually avoid you getting stroke, bleeding. One clinical trial shows that these drugs are excellent, but the same another clinical trial on this equally same population, we say, shows no effective result with that same drug. And the AI model shows it's something different. So when you have those sort of conflicting data, I don't know if you ever have to pass through that kind of research. How does that mean? I don't know if from your perspective, and this goes to everybody on the panel, how do you get that? How do you communicate that to the fellow patient, to the family? Does that make any sense?
For me, if I'm communicating to my community, I will have to just get off those studies that have conflicting results, like the vitamin D and the other one that you expressed, and just go to the basics, which is get your testing. If you confirm that you have this, then just consult your specialist or the consultant, and then follow the procedures that they want you to follow to get treated. So the vitamin D thing is just an aspect of the treatment, but you should be able to follow your specialist and then go step by step until you get your treatment. Then I'm sure those who did the trials on the vitamin D are not going to stop there. At the end of the day, they're going to be able to resolve that issue to find out why they had conflicting results. So that's my view.
Does anybody on the team want to provide another view?
Well, I would just add on that we need to know our family history. We need to know. I ran into a gentleman and he told me his dad had prostate cancer, his uncle had it, and he was under some premise that he had to wait till he was 40 or 45 to get tested. And I told him, I said, if it runs in your family and your dad had it and your uncle had it, you need to be running to a urologist or your primary care doctor. Even when you go in to see your primary care doctor, before you leave there, you need to have a PSA done. You need to have the PSA test done. Early detection leads to directions.
Well, going through this advocacy program, and one of my fears in doing this kind of work for over 8 decades have always been, how do we keep people continuously engaged? I think, I've been blessed all my career that I've been continuously engaged with people that pharmaceutical here. But from your perspective, how do we ensure that we continue to keep you engaged in research, so that not as a participant, but as a research advocate? I'm going to ask all my questions. To Senator Hill, my question is the issue of policy, because you sat in the legislature for so many years, and there have issues when you are trying to pass a lot of bills and they did not go through. As things change, and nothing has changed with respect to the burden of cancer in Black men. Sometimes things are getting worse because now we're getting more advanced disease. We are looking at the potential public health problem now that has to do with cancer in younger people, younger than 50, that are more aggressive. What can we do in that area relative to policy? And my last question to you is, we're now talking about the uniqueness of creating a toolkit for family reunion, which is fantastic. One of the things that is very popular in Nigeria and generally in Africa is the African parties. The parties get the ashrabi and get a gift from the ashrabi. Is there anything that can be packaged? Is there anything that can be packaged and very well distributed during these African events, starting from the United States, that still might be similar to the toolkit that the family reunion has here, so that we're keeping the momentum going? So, Mr. Consortium.
When I was sitting in the audience, listening to all these professional doctors receive these questions and then try to answer them, and I was saying, boy, I'm glad I'm not sitting up there getting ready to be asked all those academic questions. But I'll try to answer your question. I've been kind of given a second chance at life. When I first heard about the diagnosis, and every day you hear about people dying from cancer, and then I started doing my own research and realizing it's a second leading cause of death for Black men. That scared me. And to begin to go through the research and meet with my doctors and get all the information I could and listen to my wife, who encouraged me to stop talking about dying and start talking about living. So if I can help somebody, if I can make a difference, I'm retired. I retired in 21, and the best thing that happened to me after I retired was have the freedom to do whatever I want to do, when I want to do it, how I want to do it, and who I want to do it with. I chose to be here from Sunday night to Wednesday afternoon. That was my choice. And some of the people who invited me here are gone and told me I could leave if I wanted to. I said, no, 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 no. I'm going to stay here till the end. And I want to absorb and suck up everything I can because it's helping me better understand prostate cancer and what I'm dealing with, and anybody that I can talk to and share this information with, I will. So that's how you're going to keep me engaged, keep feeding me opportunities, give me an opportunity to share my story, and we'll take it from there. If it's a time that, hey, I got to go to the doctor, I got to have a procedure, I'm on vacation, whatever. All the other times, I don't have any problem with sharing this information with the public, with my family, and anybody that's willing to listen, because I'm talking to a lot of Black men within my fraternity and within my family that are and friends that don't know a lot about this subject, and it's an opportunity for me to share it with them as a prostate cancer survivor. And I didn't realize, my doctor called me and told me he was all excited, one of my doctors, that Mr. Jones, I just wanted to know, I had to call and tell you that your cancer has not spread in your body to any of your other organs. And I was like, I think my response was just kind of flat, because I didn't realize how significant that was, that it had not spread. And later on, finding out once it spread, you are really in trouble then. It's putting that genie back in the bottle. And something else I was thinking of when I was sitting out there. Can you get a new prostate? Is that possible? You got transplants for all these other organs. Have anybody ever heard of doing that? Give me a new prostate, a clean one, and install that one in my body. Let's reverse that process. I just thought about that, hearing all this medical research and stuff. Get me a new prostate and reconnect it all so I can be somewhat normal again. But I'm halfway normal, because at the Mayo Clinic, two of them side effects, and I don't mind talking about it: incontinence and impotence. They can fix it. I'm halfway back to where I was before I had my surgery. And I meet with my doctor at the end of this month. He wanted to fix it with another procedure. They wanted to do two at the same time. They had to put a artificial urinary sphincter inside of me to address the incontinent issue. The men don't want to talk about that. Going through the pads, going through cases of pads, changing pads 5 times a day. They don't want to talk about it. I am so past that. And this is part of the testimony that the men need to hear. And I was able to talk to him about that at the Post. I'm not ashamed. I'm not embarrassed. This is my life. I'm halfway back to being back normal again, and maybe even better. There's that scripture, a proverb, God won't put you through anything that won't kill you. It'll make you better. And I'm halfway back to the way I feel being normal. And I will be reviewing with my doctor here at the Mayo Clinic. All my doctors are here at the Mayo Clinic. And that's a blessing for me because my wife has been working for Mayo for 20 years. Dr. Floyd Willis is my primary care physician. And I can say that publicly. Been my physician for right at 20 years. I can say that. He can't say that. He told me that last night. But I'm not ashamed about this subject. I'm proud to be a part of this, proud to be an advocate, proud to be a survivor. I don't want to talk too much, but there are men that need to hear it. The women need to hear it too. I cannot imagine going through this without a spouse. I just can't imagine that. And she has made all the difference in my life, and my faith has been elevated. My sister and I, my sister is 75 going through breast cancer at the same time I'm going through prostate cancer. And we had our whole church at the altar praying for us, crying, just crying, the whole church laying hands on us. And that's what helped get me through this, where I could have went into a deep depression like some of the other brothers are doing, hiding, shame, and don't want to talk about it. Thank you for listening to me.
So, Dr. Detta, I would say to you, if I had 10 or 15, when I got ready to speak, Josh, in front of the legislature, because a lot of the guys in the legislature was going through this and did not want to go public with it. And some have passed away, right? Two former speakers. One of them was from here in Jacksonville. But the thing, Dr. Detta, in terms of the policy side of it is, if we are really serious about iCCaRE and what we're trying to do, we have to have advocacy days at the Capitol. We have to go up there where the policy makers, and we have to take champions like this gentleman with us, right? We got to take the different groups that are doing stuff around prostate cancer and breast cancer. We got to take them to Tallahassee so they can see real people, all right? And then we have to also engage with our pastors that are secretly going through this, right? To say, now, I want to give y'all a heads up on this now. Don't go running up on your pastor telling him, Dr. Willis, you need to do this, and then he look at that book and you ain't tired. Hello, somebody. See, I tired at Macedonia, so I can talk to the pastor. All right, I can get on the agenda. All right, y'all don't want to talk, but this is the thing, Josh. I can get on the agenda because I tired, right? And Josh is tired. But please, Dr. Willis, don't go running up on your pastor talking about you need to do this, you need to do that, right? And you're not tired over there. All right, that's just a sidebar. But again, though, on the advocacy side of it, we need to have a day, and then we need to also let our community know that we're not going to sit in silence around this disease that is killing our loved ones. Right? We got too much information to be quiet. Right? And so that would be my take as it relates to being a former legislator. I am so glad that you approached me about this, because I got involved in politics working on the docks. as longshoremen. And I can tell you, we have a Cadillac plan, but most of the guys don't want to go to the doctor because they're so busy running behind the ships, right? And then when you look around, that the guy is not there, and you're trying to figure out, well, where is he? He's sick because he got prostate cancer. So that would be my suggestion to the iCCaRE team, is that we need to have a day that we go to the Capitol. And then we need to have our organizations doing resolutions to send to those policymakers, even when they have delegation meeting. If you can't make it to the Capitol, I know you can make it to a delegation meeting. That's where we sum up what we want done for the legislature. And so let me close with this thought with you. Reverend Jackson told me one time, Dr. Willis, just raise the issue and you win. We need to raise this issue.
Yeah, thank you, Dr. Odedino. By talking about the Nigerian parties and how we are interested in their shwebis and what you are going to take home when you are leaving after the party. So with that in mind, Dr. Balajoko, in one of our Nigerian independence and anniversary celebrations, yeah, she came up there and had a setup, a table with mayo on it, and had little things that people could take home. Yes, not apart from the literatures, even something like the jotters, and had me come in and tell the guys, Mayor has a setup here, go there and see what is there. And then we took the opportunity to put up a sign-up thing for people to sign up if they wanted to come for our prostate cancer roundtable. It was around the corner. And that little thing that we did, you'll be surprised that when I hosted the roundtable, we had more people coming in for that prostate cancer roundtable that we get when we have a Nigerian Association meeting. Yes, just because of that little thing that was done. So that is what we can do. You might look at it and say, oh, you are not giving them an umbrella or what have you. No, something as small as a jotter with the mayor, whatever on it, and they come in there, they ask their questions, and then if there's anything coming up, like we are thinking about the medical fair and all that, if it's coming up, then you use the opportunity to talk to them about that. And then one more thing that I want to add. Mr. Jones is lucky that he has his wife working in Mayo. I have a very close friend of mine who is a pharmacist like myself from Nigeria. I saw this guy going down. So I asked him one day, I said, what's going on? He said, my prostate. So I said, okay, get back to me. That he never did. So the day I went to my doctor, my specialist. After I finished him, I said, doctor, look, this is my situation. I have a very good friend of mine, a pharmacist, but somehow he's running away from me. So he said, you have his phone number here? He said, yes. I said, call him. So we called this guy. I told him, I said, look, I am in my daughter's office right now, and he wants to talk to you about your prostate. He said, oh, Innocent, I'm traveling to Nigeria tomorrow. My brother died and whatever. When I get back, I will get in touch with you. So he never did. Then he kept going down and going down. One day I called him, I said, look, my friend, what is going on? So when I saw him about a month ago or so, he told me he had gotten the surgery. So I told him, okay, you've gotten the surgery. But if we had, talked to me, talked to my doctor about a year ago, you might not have been been able to go do the surgery. That would have been taken care of at that stage. So prostate cancer is such that is not whether you are an illiterate or whether you are literate or something. It is just, especially among we Africans, because of the area of the body where the thing affects, it's very delicate. Secondly, I was just telling Dr. Bolajoko today, if we're going to run another prostate cancer roundtable, the wives are going to be there. If the wives are not there, we are not running no roundtable. Yes, they have to be there to listen to what is going on, because the men will hide all these things from the women. When you are going to the doctor, your wife should be there with you and be listening. If it gets to a stage where the erectile dysfunction thing comes up and you are thinking about the different things that you can use and what have you, your wife should have an input. But we as African men, we are so secretive, not just about prostate cancer, about disease, about so many other things. We're doing things we don't want our wife to know about it. And suddenly the man drops and is gone. And the wife and the family, they do not know anything about this man, So that was what I told Dr. Bolajoko today. We have to open up to our families, to be able to get a hands-on this thing that is affecting everybody. Luckily, prostate cancer is not the one that will just come up and kill you tomorrow. Mine, my PSA was like 4.5. I went there. They did. The doctor took took strips for biopsy. Out of 10, only 2 came out cancerous. And he was telling me, "Oh, I don't think you have any problem." I said, "No, there's a problem there." He said, "What do you want to do?" I said, knock it out. I'm not going to be fooling around because 2 out of 10. Yes, I'm not going to be fooling around. So that was why I was able to. The only thing I did for my treatment was to go for radiation, because the thing was just there, and they just went and knocked it, and I've been free now for 10 years. Yeah, so.
Thank you so much. This has been very, very interesting, and personally have learned a lot. And interestingly, I'm part of the PES. I'm going to meet with Dr. Willis, and then we got job to do. So I think I want to thank everyone who have asked questions and for making this moderated session a success. Thank you very much. And thank you, our advocate, for allowing us to, for sharing your experience with us. Thank you.
Thank you.
Okay, thank you so much, everyone.
Are you having more? Are you having more? Huh?
It's not that. I'm not giving you. To come in the last 2 days with us. Before we go, it's my favorite part is to show recognition, because without all of you and all of the busy work that you are doing in our projects and core, and the culmination of everything that we've learned over the past 2 days, iCCaRE would not be where it is today. So before we adjourn, I would like to show appreciation to the Consortium for Recognition Award to the MEMS Board. So in recognition of outstanding commitment to gain and support of the iCCaRE Consortium and its project, Dr. Young, you can accept this on behalf of the MEMS.
The next consortium for recognition award goes to the planning and evaluation core in recognition of outstanding commitments to the team and support of the iCCaRE Consortium.
I knew it was so bad. You get all the one that you represent.
Next up, the Consortium Core Recognition Award goes to the Media Core.
No, wait, please. Let's Prof. hand it over to you. Yes.
And as you all have heard today, this work would not be possible without our global team and all of our coordinators in Africa and around the world, really. So we would like to recognize our teams globally with the iCCaRE Global Impact Award, which has been awarded to the African Behavioral Research Center of the United States. And recognition of the talented global leadership in behavioral science and strengthening the capacity to reduce the burden of diseases of our country.
1, 2, 3.
And last but not least, as you have noticed today, we've had a display of all of the projects and pilot projects and everything that are being funded out of iCCaRE through Dr. Convention's hard work with his group. And so I'm actually going to let Omar, who is the senior program coordinator over our global projects, actually present the Best Scientific Poster Award.
Thank you so much. It's a good thing that we got awarded them because I've been involved in the keynote recently, so I'm not here. So the best scientific poster goes to the name the psychologist of cancer in Canada.
You don't know the story. That's the story. That's the story. Yeah, this is story. What is so good? Well, it's not good. It's not that good. It's a top line. Okay. Thank you. Yes.
All right. Last but not least, he could not be with us today, but we would like to recognize Professor Amansi with the Distinguished Scholarship Leadership Award in recognition of the leadership advocacy on shaping real poverty and advancing global efforts to reduce equity.
Hold on.
Are you gonna let that?
Yeah, so not special, because it's special. Okay, I'll leave it over here.
Don't be so blind that you say. Why did you call me?
All right. 1, 2, 3.
Okay, so this has really been such an outstanding. So I'm going to ask you, so going from poor to fair to good to very good to excellent to very excellent to exceptional on a count of 1, 2, 3. How would you rate the symposium? How would you rate the symposium? Thank you so much. This has just been, to me, the exceptional also. The science, the care, the clinicians, and the best of all, the survivors and the advocates. So amazing. Since yesterday, we've had a long list of what to do. So I don't know who's taking notes. I know Jeffrey and Jada, they're trying to bury their head as if I don't see them now. Oh, you have to go back. They're going to review everything, and they're going to, let us know what is the next step. A lot has been done, but I have to tell you what I am most excited about is the creation of a toolkit for family reunion and for Owambe. So there's no way toolkit for family reunion, and then we're going to think about the African group is going to be toolkit for Owambe, which is for the parties that we go to. I think that's going to be really exciting. Thank you so much. For the last people standing, where's the best place for us to take a picture? We also always take a picture of the last people who are standing. So we can, outside of here, maybe here because we can sit on the chair and then people can gather around. So why don't we take a picture and then we are dismissed people to sort of network and stuff. So let's move forward, take a picture, and I hope, remember, if your project teams and your core teams are not taking a picture, even if you're the only one here, from your core, go upstairs your local self, go take a picture. But let's take the final picture. Thank you, everybody, for taking on. Can we have the male child people let us